ਪਹਿਲੀਆਂ ਦੀ ਮੈਨੂਅਲ ਤੇ ਸੀ। ਅੱਛਾ ਉੱਪਰ ਮੈਨੂਅਲ ਤੇ ਸੀ। ਕੈਮਰੇ ਦੇ ਉੱਪਰ ਲਾਏਗਾ।
फिनाइल दी आई जाती है इन्होंने फिनाइल पाया और पानी मिला लेना पानी मिलाया दीदी तो कोशिश की थोड़ी यार वो मेरा पासवर्ड लगा ना और चक के ना इधर कट के लगी वो उत्थे दिखता नहीं किधर के उत्ते क्योंकि ना तुम दसा दे कबूतर जाते नहीं गे
I extend my warm welcome to Dr. Tanu Anand Ma'am, Scientist E, ICMR New Delhi. She is the Nodal Officer of North Zone MRU MRHRU. Ma'am, unfortunately could not join us in person due to some unforeseen circumstances. We will miss her valuable insights in person, but she is concerned with us online. Welcome, ma'am. I extend my warm welcome to Professor Dr. Joseph Jones, sir. He is the head Department of Clinical Hematology, CMC Ludhiana. Welcome you, sir. Now I welcome organizing chairperson of this event, Professor Dr. Neetu Kukad, ma'am, Professor and Head Department of Immunohematology and Blood Transfusion, Medical Superintendent, GGS MCH Faridkot. I now request Professor Dr. Neetu Kukad, ma'am, to please come on the dais for a formal welcome. Thank you. Thank you, ma'am.
Good morning, everyone, and welcome to you all. It's respected guests, esteemed faculty, and dear participants. It's with great honor and privilege that I extend a warm welcome to each one of you gathered here today. Your presence adds immense significance to these 3 days workshop capacity building program, and we are truly delighted to have you all here. I extend warm welcome to Dr. Tanu Anand, Scientist E, ICMR, Nodal Officer MRU, MRHRU scheme. Your expertise and dedication for MRU is truly inspiring, and we are grateful for your contributions. Your presence adds significant value to this gathering. Our esteemed ICMR Intent Site Clinical Trial Program Director Professor Dr. M. Joseph Jones, Professor and Head Department of Clinical Hematology, CMC. Your leadership and vision inspire us to strive for excellence. Professor Dr. Johan will introduce you about the scientific program of the workshop. Hopefully, it will be an engaging and insightful discussion. Our esteemed resource persons from ICMR, CMC and Guru Gobind Singh Medical College, Faridkot, who have graciously agreed to share their expertise and insights with us. Your presence and sharing of knowledge will enrich our gathering. I would like to welcome our valuable participants from GMC Patiala, GMC Amritsar, ICMR New Delhi, and GGS Medical College Faridkot. We are gathered here for clinical trial workshop, which holds tremendous importance for each one of us. Clinical trials are indeed crucial as it provides necessary knowledge and skill to design and conduct clinical trials, to understand regulatory requirements, to develop research protocols and informed consents, ethical considerations, and to manage and monitor trials, and finally analyze and publish clinical trials. As we embark on this remarkable journey together, let us seize this opportunity to engage, learn, collaborate for the betterment and the stepping stone towards progress, growth, and shared success. Once again, a warm welcome to each one of you. We look forward to your invaluable contribution and guidance throughout this event. Thank you and welcome you all.
Thank you so much, ma'am, for the warm welcome. We are here to start new things pertaining to the domain of knowledge and novelty, and no such endeavor can be successful without the blessings of Ma Saraswati, the goddess of knowledge, Ma Saraswati. The embodiment of grace in your divine light, we find our place. So, let's proceed with the Saraswati Puja ceremony to inaugurate this workshop. I would like to request our clinical trial director, Professor Dr. Joseph Jones, and our organizing chairperson, Professor Dr. Neetu Thukar ma'am, and all the resource persons to please come forward here for the inauguration ceremony.
Saraswati Mahapatra Mahamaya Varaprada Shriprada Padmanilaya Padmashi Padmavatraka Shivanuja Pustakabhat Jnanamudra Ramapala Kamarupa Mahavidya Mahabhatakanashini.
Thank you everyone, and please be seated now. I would like to request Dr. Tanu Anand ma'am now, who is there with us online, to share the objectives and scope of this workshop under DHR and ICMR capacity building initiative. Ma'am, please go ahead and enlighten us about the workshop goals and its objectives.
Requesting for sharing of my slides. So anyways, in case if not, then I would like to first thank Professor Dr. Joseph Jones sir for for taking time out as a part of Intent site from CMC Ludhiana and actually helping our MRU network strengthening their capacities for clinical trials. So, as you all know, when it comes to research evidence, randomized clinical trials are considered at the apex of the pyramid for generating hierarchy of evidence. Therefore, it is pertinent that being a hub of clinical research, which our medical colleges are envisaged in the part of Intent network, we build capacities for the faculty in these colleges through our MRUs so that they can contribute for generating clinical practice guideline. We are progressing very well towards development at the rate of 2047. We want to envisage as Viksit Bharat. Therefore, this is an apt time where medical colleges could act as a global powerhouse of innovation. What makes us different from rest of the world is the fact that we have a diversity which is very unique to the country. We already have an existing drug manufacturing infrastructure and there have been a lot of initiatives taken. Intent is one of them, and then there is National Biopharma Mission, DHSTI by DBT, and various other schemes supported by other ministries. Therefore, with that background, and of course we had learnings from the COVID pandemic as well, this unit called as Clinical Studies and Trial Unit was formulated way back in September 2020 when we were actually struggling to find out an important treatment or interventions to control this pandemic of SARS-CoV-2. As you may be aware, at that time, we had various trials, global trials that were generating evidence. The famous of them are solidarity trials, recovery trials, and many of the Indian sites were participating, but they were mainly coordinated by global hubs from UK, NIH, etc. So at that time, Ministry of Health and Family Welfare proposed that ICMR should establish at its headquarters a unit that would establish a network for clinical research and in particular clinical trials, and therefore the birth of Intent Network, which is the Indian Clinical Trial and Education Network, took place. So this, talking briefly about this, so Clinical Studies and Trial Unit and under is a coordinating unit for phase 1 clinical trials, where we have 4 centers as of now that are Centers for Advanced Research and Excellence for phase 1 trials, and for phase 2 and phase 3 clinical trial, which comes under Intent Network, we have now 75 such hospitals and medical colleges that are contributing to a robust clinical evidence. So the kind of scope of work for clinical studies and trial unit under Intent is to provide end-to-end solution for phase 1, 2 and 3 clinical trials. From ICMR headquarters, we currently do regulatory clinical trials for new drugs, vaccines, biologics, gene and cell therapy. And we also have a clinical investigation for innovative devices and diagnostics through MedTech Mitra, which is again one initiative in which ICMR along with CDSCO and NITI Aayog is contributing to support the startups and entrepreneurs with ideas to the product fit market fit product. That is the kind of conception we have. And Intent comes in the phase where the medical device or diagnostics is at the stage of clinical validation. So all this is contributing towards strengthening the clinical trial ecosystem in the country. So the unit is spearheaded by Secretary DHR and DG ICMR, Dr. Rajiv Behel, and the unit comes under the Division of Development Research headed by Dr. Taruna Madan Gupta ma'am.
Recording in progress.
Clinical Studies and Trial Unit is Dr. Aparna Mukherjee, and there are 6 scientists, and I also belong to that unit. Now coming to our very own multidisciplinary research units, so we have a network of 118 government medical colleges and 5 private medical colleges under this network. This is a scheme, central sector scheme sponsored by DHR, and this has been running for last this is the 13th year, and we are going to soon enter into the 16th Finance Commission for extension of another 5 years in the league. So this scheme, just to, you all may be knowing it, but just to reiterate that this was to encourage and strengthen the research environment, and MRU Faridkot is also one of the oldest among the league, and therefore we felt that such units which are hosting these workshops are at a platform where they can take it forward to the next stage of research, and therefore we also want to bring in other MRUs which are relatively newer to support. The ultimate purpose is to improve the health status of our population by creating evidence-based solutions. Currently talking about this workshop, we have certain objectives. You all as participants are faculty members, and many of you may have got some experience of undertaking clinical trials and some may not. But what we want this 3 days to be is to strengthen both your cognitive and psychomotor skills regarding development of protocols with study design clinical trials. And Joseph sir, with his vast experience of leading some of the great clinical trials, has contributed immensely to the program to achieve this objective. We would not be leaving you just after this workshop. Immediate outcome is a gain of knowledge, which we are going to assess through a pre and post-test assessment, after which there is a short-term outcome that at least one protocol from each MRU, and we also have participation from MRHRU Hoshiarpur within 1 month of completion of the workshop. And next year, when we have our extramural grand call, which is going to be in early January or late last week of December, we inserted some of you submit them to this call, and next year when the results are out, 1 year from now, we are going to see that how many of you were able to successfully achieve one extramural grant, and for MRHRU specifically, they would be seeking intramural grant if they go to ICMR, and they can go beyond ICMR as well. So with this purpose, I would like to convey my best wishes to all of you, and I would sincerely request all of you to please be engaged actively and ask as many questions, as many discussions, because there are 3 MRUs participating in this from Punjab, and we have MRHRU, so your settings are almost similar, but there is yet some diversity which you can learn. And to say more about sir, this would be the best time to actually learn from him and his team. And from ICMR DHR, I would like to extend the support that you would require after this workshop, because being part of the development research and extramural division of ICMR, I have always said that from both either ICMR or DHR side, if you require any help, my role is more of a facilitator rather than being a resource person also. So thank you once again, and I again convey my apologies for missing out this opportunity to meet you in person. Thanks once again.
Thank you so much, ma'am, for the valuable insights and encouraging all of us over here. Now, I request Professor Dr. Joseph Jones, Intent Site Director. Sir is heading Department of Clinical Hematology and Bone Marrow Transplant at CMC Ludhiana. Sir did his MBBS and MD from CMC Ludhiana and DM from CMC Vellore. Later, he did his MBA in Hospital Administration from BITS Pilani. After completing DM, he has set up the Hematology and Stem Cell Transplant Unit in Punjab, CMC Ludhiana only. Then later again he did his MBA in Health and Hospital Management from BITS Pilani. Currently the center has an active sibling and matched unrelated donor transplant program for thalassemia and other hematological disorders. His other area of interest is hemophilia care and heads the comprehensive care center, which actively takes part in teaching and training for other hemophilia centers in Punjab. I request Professor Dr. Joseph John to please come on the dais to introduce about the scientific program and the agenda of 3 days event with all of us. And he is the first speaker of today's event. And the topic is Overview of Clinical Trials in India and Global Perspectives, which will lay the groundwork of our current event. Thank you. Welcome, sir.
Good morning, everyone. It's a pleasure and an honor to be here in Faridkot, interacting with you and learning together. And we have 4 centers here coming in, and under the leadership of Dr. Nitu Cooper and Dr. Richu, who have coordinated this entire process. So I want to first of all tell you that this has to be an informal discussion and interaction, and there is no formality here. And when a lecture is happening, please stop, I didn't understand, and then move ahead. We may not cover exhaustively all what we have to do here, but whatever we cover, we will cover it well. I want you to take out the program sheet in your hand and have a glance through that before we start the entire process, so you know exactly where we are heading to in the next 2.5 days. And I hope over a period of time we will know each other by name. And so presentation on. So if you can look through the program on day 1 and day 2, day 3 on the top, you will see basics and foundations, statistics, design and protocol development. And the last day is operationalization and data and presentations from each of you. So what we envisage is essentially for you to get an entire overview of what is happening and make it a hands-on immersive experience. Some of the experiences or interactions will go beyond the duty hours, and you can do it online or over the dinner, and then come back and make your presentations on the final day, where there will be judges who will give you some remarks on how to modify that. As Dr. Tanu said, at the end of the day, you are expected to come up with a research idea and submit a grant proposal to ICMR. And there is a fantastic milieu right now we have, is they are willing to support, train, and then want to give you the money to do the study. All we have to do is to grab it and ensure that we do it in the right way. So today we will have, Dr. Tanu has just mentioned a few of the discussions. I will give you an overview of clinical trials, then ethical principles, study designs, then formulating a research question. But before that, the group 1 activity is slightly changed here. I wanted to use choose a journal first, and then we will go. This is the version 2, I think. Yeah, no problem. So there is some change there, but we will do the journal. So what we are ending up doing is, you will get a journal in your hand, which is, we are supposed to do the analysis, critically analyze that, and design your research question in line of that. Imagine you have all the funds, you have all the, everything at your disposal, and therefore you design one on yourself. So that's an exercise, and that may or may not translate to your ultimate project proposal you will submit to ICMR grant. But at least that will equip you to think through all the components. There is also a laminated sheet on your table that is also there, where I have written a life cycle of a research starting from say ideation to dissemination of the work. Today when we do the journal club, we will start from the end, that is the dissemination, and you are analyzing it critically. We may go back and forth, up and down here, but essentially all the 12 landmarks would be covered, partly at least, and then you can go ahead and do in-depth understanding of each of them. So, from that, we will go to the Google form which we have sent across to you, and it is good to know how and where we all stand. And in this journey of 12 landmarks I mentioned there, which you are seeing the life cycle, nobody is 100% professional there. We are professional doctors with amateurs in clinical trials and research. And if you look at it, you may think that the other person knows better than you. If you don't understand, it is likely that the other person also does not so. So don't be shy away from asking questions. The dumbest of the questions you may think, but that may give insight to many other people as well. So this is the intent, the form which we sent across to you. And if you see, that's the distribution of the institutions which we've got. Years of experience wise, okay, 50% of us are more than 5 years in some research or the other. That means there is some seniority level happening, but there is a good mix of people at less than 1 year as well. So we have, if you look at the age group sitting here, we have baby boomers to Gen Y or Z as well. Okay, so our understanding of things may be different, but that is a fantastic thing about research is nobody is there yet. So even the junior most may know more than the baby boomer or a Gen Z, Gen X, but it is a good interaction which we have today. The previous trial experience, 75% of us do not have. So it is fine. So many people are grounded here. So it is good to the other 25% who know something about the clinical trial. We want you to learn as much as I have prepared while preparing for this entire workshop. I have learned much, and I want to learn more during this interaction. So we are diverse. We can remove this from the top, right? This. Yeah, and this. Okay. So as a group, we are. I asked, how do you describe yourself, right? So people described diversely, but many of us are academicians, clinicians, scientists, and technical experts. Now, to get through a clinical trial, interestingly, they need all these components. It is not that one academician can do that. As a clinician, you may not have the wherewithal and the time to do everything else. So therefore, you need to have a team to interact with, and that's a discussion, that's the disciplines which we are here today. Then many of you are motivated by social relevance, and you want to see key health changes in Punjab, and you want to address non-communicable diseases, diabetes, lifestyles, dietary issues, public health burdens, etc., and with strong personal values, with honesty, hard work, and sincerity. So we have professionals identity rooted in growth and learning, and of course we have the reasoning skills, medical education, and many are novice in research. And we have a shared inspiration to do ethical and evidence-based clinical research. That's the ultimate aim which we want to do. It is not because there is fund available we must do the research. We must do the research because I want to change the health status of the nation, and therefore I will utilize the availability of the funds to address that question and do that. Sometimes we think the other way. I do research because I want to get promotion, or I want to get a salary hike, or I want to go abroad and do posters, or there is fund, somebody has to use it and therefore I will use. Rather, I would say, do that as a secondary objectives, and the primary objective should be I want to make a change, and what is that change I want to make should drive the rest of it, and then you don't have to worry about anything, you just drive, you just sail away. So we asked the research journey, and many of you have had the opportunities to do postgraduate thesis and write up some proposals, and have somehow the formatting is coming not correct in this. The word, the format. So it doesn't matter. We'll adjust that later sometime. But again, it's the same repetition there. And we would come to, this is an important question we asked. one healthcare policy change you hope to contribute through research, and that should drive our part. So you said you want to do, this is through the public health clinical practice improvements, but you wanted to do on prevention and early detection in screening, in gynecology, in cancer, metabolic syndromes, and diagnostics. So you may have come with your question in mind in your area of speciality, but when you sit together as a team and the ideation process happens, which is the first step of the life cycle, you may have to choose one and give in to the other person's idea now. But that does not stop you from pursuing your idea to submit a grant proposal. So if 5 people are in one group and 5 different areas of interest, you can be a co-investigator in the other person helping out in some aspect, but you can drive your own research separately also. We'll come to what are the opportunities we have in India in terms of choosing that. So many of you wanted to look at also the cost-effective pathways of doing equitable access and standardizing the care pathways. One nation, one treatment in hemophilia is one example of that, and some in orthopedic and trauma referral systems. Some of you talked about public health policy systems, improving the resource allocation. Again, a very prudent way of looking at how to address the cost effectiveness. That is what we need to do in terms of LMICs and financing models there. Again, capacity building, food safety. Some of you wanted to look at the human, animal, environment, health. Antimicrobial resistance is a huge thing we need to address, because that is again an important aspect. When I asked for the infrastructure, all of you have an institutional ethics committee. There is a research cell is available in 81%, and data management biostatistics are there for 57% of you sitting here. Now with the availability of the ICMR, I'm sure the biostatistics also can be taken care of if you talk to Dr. Tanu, and they would be able to help you out. Otherwise, MRUs will have biostatisticians available with you. So this is the number of ongoing projects which in your team. So essentially nobody is at zero. In terms of some research or the other, all of you have taken part in somewhere or the other, which is ongoing in your institution. This is the interest level of infectious disease, maternal health, non-communicable disease, hematology, oncology, mental health, and other areas. So again, see how infectious disease and ICUs can come together. Infectious disease and non-communicable hematology, oncology can come together. So you can have collaborative research which can happen. Key expectations of the workshop from you: almost everybody said you want to learn about clinical trial protocol development, how to submit it, and budget and grant writing, and randomization, trial and statistics, and how to do data capture and manuscript writing. I think all of this is touched upon in the 2.5 days. So challenges when we asked. This is a ChatGPT. I just put in all the challenges and said, give me a frequency bar diagram on the challenges people have faced. And study design and methodology and biostatistics seems to be the biggest challenge. And therefore, I would expect more of you to ask questions regarding that and get learning from each other in the process. What is this habit, 7 habits? So what is this? Who wrote this?
Stephen Covey.
Stephen Covey. So I would say be proactive. Start with the end in mind. Begin with the end in mind. What is the end in mind you have for this workshop? Anybody? From whatever discussions happened with Tanu Anand and what I discussed. What is the end in mind? Develop a protocol, submit it, and get the grant in the end. So that is the purpose of this workshop, is that ultimately we need to reach that. For that, many things have to happen. So if you have to reach the 12th life cycle of research, I will start with number 1. Okay, with that, we will come to the overview on the clinical trials in India. Somehow the formatting is off. Okay, So the objectives is to understand what the global landscape is. And I would want to quickly go through the slides without making you read as many things as required there. We want you to go through the ideation to policy and practice change. So I want you to once more go through that laminated sheet in front of you so that you have an idea about where we are heading. So every lecture we have, which we are doing, you should be able to go and say, we are here now. So that you know exactly how the life cycle happens. You can take a picture also. I will share the document with you later once I get to my laptop. So ideation to publication, essentially we are looking at how to get your reviews done, how to get your research questions done, how to do a protocol, budget, ethics committee, regulatory approvals, funding, how to implement and set up an MRU in your own center. If it is not done, recruitment. Now all of us have some idea in different grades in all of this. And the lectures may be zigzagging from one to the other. But again, once you look at it, you will be able to know. You can't go from one end to the other all the time. Okay. Yeah, so that's the life cycle I mentioned. So, okay. So why clinical trials matter? We discussed about that. We need to have generalizable evidence, and as Dr. Thanuvan said, this is the apex of all the levels of evidence which we can get, randomized controlled trials. So from an era, about 25 years ago, we went with gut feelings, and the senior most said, where is the evidence? So-and-so doctor said to do this. Was the rationale of doing certain things. And I tell to my residents all the time, consider me the biggest liar. And therefore you go counter check everything what I say, and you should be able to say it is in this journal published in so and so time, and therefore we are doing this. So evolution of clinical trial in the global timeline. So anybody knows the story of 1947 James Lind? Okay, so all the sailors were getting bleeding, and he then thought it could be this and that. He made 4 groups, and one person they gave some acid, some alkali, some person give and some person he gave this white citrus fruits, and they found out that the group who had the citrus fruits did better. But to implement that among the sailors, it took another 30 years. So that sometimes, hand washing, she mentioned about the hand washing story. Sometimes you may have this, she didn't mention that. So there was a time when this Ignace Simowitz, he suggested that people who washed hands and then went to the postpartum, delivery room, would have less infection. Poor fellow was told that he is to be going to the mental asylum, and he died there actually. Then came the Nuremberg Code. I'm sure some of this will be discussed. Why did we have to have Nuremberg Code? So Nazis got a group of people, Jews, in captivity, and they wanted to know what is the human level of freezing and unfreezing a human being can go through. So a human being, put him there, freeze him, unfreeze him, freeze him, unfreeze him, whatever they want to do, they did that. They thought they will make a lot of advancements. But when there is moral contamination in what you do, it is bound to fail. And none of these trials actually helped in altitude pressures or whatever, actually helped us to do that. But that is how in 1947, when the World War II was over, Nuremberg Code was there on who can be the participant. It's not even called a subject nowadays in a trial. It is a participant. It is not a subject. Then came the Declaration of Helsinki, Belmont Report. Helsinki was, vulnerable people, you have no money, get into the trial. That is also a problem. That is because you are vulnerable, you are exploiting the person's inability to pay to get into a trial which is not really required also. So like that, we have evolved and come a long way in holding up the medical ethics. What are the 4 pillars of medical ethics? Fantastic, good. Many people don't remember this all the time, and that is exactly what the ICH and the Belmont report actually said, that respect to the individual is the epitome of making a decision. There are a lot of randomized control trials coming in. There is globalization. There are various designs. We will come to those designs later. So, lymphoblastic leukemia in the early stages was only 20%. Right now, the outcomes are good, thanks to the German trials and the European trials. In the global landscape, the top countries who are doing trials are United States, China, European Union, South Korea, Japan. India is rapidly growing, and we have to see the Viksit Bharat in 1947. They want us to be there. If that has to be happening now, 22 years from now, we have to reach there. We have to start with all of you. And the global focus is now mainly on metabolic diseases. From communicable diseases, we have gone to non-communicable. So you need to think about, you have 10, 15 questions in mind, you want to answer them, then you can say which is the most feasible based upon the fund availability, etc. And therefore, metabolic disease and many of the thoughts you had are covering that. So now it is going to various types of trials are there, called the umbrella trial, the basket trial. Various types of trends are there, and that is something which you will learn while you are learning about the trials, and a lot of emphasis on real world evidence now. So even if you have a simple registry to be set up, it may be good enough. Okay. So this is the international level. If you want to register a trial, it is the clinicaltrial.gov. Why do we want to do that? Because you want, before you do a trial, go and see in India or in the West if somebody else is doing or not. Because that will tell you, are you going to ask the same question again and again, or you want to modify that question. So research is like the sand in the ocean. Somebody says, wo mera idea tha, wo chhin liya. It's like saying that you took my sand from the seashore. So it is that much ideas and possibilities are there. You just have to modify and say somewhere else you have to go and pick up that sand. The Indian perspective, the India's strengths are we have a large number of patients. So our number of patients are 5 times that of the Western, the US population. But they have 5 times the land than us. That's a different question matter altogether. But we have the opportunity to lead the world with the number of patients whom we have. And this is the CTRI website. So if you are doing a trial, I would suggest that if you have, once you get your concept in, go to CTRI and say what are the trials. If you register yourself, and even if your thesis you can do through the CTRI website. There are about 4,000 plus trials going on. You can easily, so the idea of asking you to bring your laptops are that you may please use this password, set up your laptop, take notes into the laptop, or search the websites side by side. That is a way of active learning and saving that in your groups. There is a regulatory environment, DCGI and CDSCO, ICMR, and then there are these milestones we just mentioned, Dr. Tanu mentioned, new drugs and clinical trial rules have come up. Online there are courses for this, for half an hour, 1 hour, sometimes they do. I did one such in 2020. They'll give you a certificate, it's a nominal money which you have to pay. I'm sure your research grants will help you to support that at some point or the other. So now it is fast, transparent. Earlier days there were a lot of problems regarding the recruitment of patients, just without taking a consent, or tell something else to the patient and do something else if he is illiterate. Those things has caused a lot of damage to the reputation of Indian trials, and that is why 2019 this new drug and clinical trials rule came up, and now it is much more robust, and there is a process of auditing entire process. Ethics boards have been strengthened because, see, we are a body which are self-regulated, except for certain external government or agencies which are checking it. When you say self-regulation, that means our own faculty, our own kith and kin has to, in that matter, the medical faculty is supposed to safeguard that the other person is not doing anything wrong. And therefore you have the ethics committee. So we'll have more discussions on ethics committee by Dr. Rinchu. And they also have to submit their guidelines and protocol to the ICMR and get the certification there. Trial designs in India, so if you want to do any study or a trial, what are the options we have? One is like we do the thesis with no money involved, we do observations. But if the money has to be there and if financial support has to be there, what are the options we have? Academic trials like what we mentioned. You write a grant to DBT, DST, ICMR, HCC, ISBMT are some of the hematology centers which we go, not, and you ask for, I want to submit this trial. Second is industry sponsored trials. That is another way of boosting your infrastructure. That is what I did. We never had funding in CMC from externally to start with. So you take a pharma sponsored study. That means a pharmaceutical company wants to market a particular drug, and they have chosen India as one of the sites or India as the only site. In India, they would have now given a letter of interest to various centers and said, do you have these many patients? Do you have XYZ? And you say, yes, we are interested in it. And then they will give you a non-disclosure agreement form. That means you should not share that protocol with anyone. You write that and then they will send a protocol and say, can you be one of the sites? Then that then funds your center for either a clinical trial coordinator or an infrastructure development or whatever. And there is a money associated with every patient completed per visit. Now, institutions have different rules. In institution of CMC, none of that money comes to the person. There is a PI fees incorporated in it for the time and effort you put in. For us, that money does not come to us, but it stays in the department. We have the freedom now to use it for subsequent studies or a traveling or employing a person within the department. So that is how you build some bit of academic industry sponsored trials. Now there you are not the key PI of the trial, you are a co-investigator, a trial site. You are not even a co-investigator. Your name may or may not come into the author list depending upon the number of patients you have recruited and how much of input you have given in the manuscript. So that helps your department to grow in infrastructure and human resource learnings. Then there is investigator-initiated trials. I like to do a trial and I want to use a particular drug, or I go to a corporate center and say, I want to do this trial, this is the cost, this is the budget, I'm not taking any money for myself, but this is the cost of the drug and this is the implementation cost, can you give us? So sometimes even the drug companies have such trusts available where you can write to them and say, I would like to do this trial, can you fund us? That also helps your infrastructure. There is no PI fees, there is no service fee associated with it, but you will get a person to be employed in the department who may do other things as well. So that is investigator-initiated trials. Then there is hybrid academic and industry partnership. We have a thesis, and in that thesis, I want to know this is standard of care and this is the new drug which has been launched and that is approved by the FDA. I want to do a study, I write to the company and say, can you give the drug free? Rest everything remains the same. Majority of the clinical trials in the West are like this. The insurance companies pays for the entire treatment. The investigation company will give you the drug free. And therefore it's a win-win situation for everybody. Then there is real world registration. That is something which is, I would say, anybody can start with. So like now Varun is initiating this hemophilia registry for Punjab. Every center registering hemophilia patients should deposit their reports of the patient. Now there is patient level data and the institution level data. There are two different things. Patient level data is Mr. Dhyanchand has come, his reports are this. That is a granular data, and many of those patients in each row will give you a lot of insights on the patient data. One is that. Second is HTC or the Faridkot, Amritsar has so many patients, Patiala has so many patients, so many are hemophilia A, hemophilia B, C, etc. That is institution level data. That may not give insights into what happens, etc. So you can start with diabetes, you can start with postpartum hemorrhage, any of your centers, and I would say that should be a parallel growth with all the MRUs you come, and I foresee Punjab leading the rest of the country if we can come together. In fact, this is it. These 3 medical colleges and ICMR, if other private medical colleges are interested in coming in, develop registries for every disease possible, either within the institution or collectively. And for that, databases to be created. I'm sure ICMR will help you to get software databases to be created. We'll have a talk on what are the types of databases we can have. So these are the ways in which the last one has no money required, but if you have any of the others going on, you have manpower who will help you. So I would say many clinicians are also here. From the clinician's point of view, there is always an angst. 8 hours, 80% of my time is spent with the patients doing rounds and tackling the firefighting the patient's relatives fighting. Where do I have the time to do the data entry? You have to create a system by which you have other people who will help. So you need to have a clinical trial coordinator, research coordinator, and unfortunately in India there is no courses in which you can go and hire a CRA who will come with the bunny knowledge. All you have to do is to get a person, train them, but that helps out in the long run. That is what we have done in our department. Challenges in India, our scientific challenges are there. Limited GCP training is there. GCP training is something which you can do online. So if some of you have not done GCP training, in some of the slide sets coming up, I will show you the websites in which you can do either a paid or an unpaid. There are free GCP training, and we make it mandatory for all the research team and all residents to go through GCP. It's very easy. Online training, you ask some questions, at least you orient yourself towards a good clinical practice. So even if you do not have a GCP trained person, ensure that all your residents go through GCP, all your nurses go through GCP. Tell them it is a good thing, you will get a certificate, you can put it in your CV. And you have to renew that every 2 to 3 years. Operational challenges are plenty. Slow patient enrollment, contracting delays. So basically, sometimes you may have to engage with another outsourced agency in which to do the test. MOU has to be written. At least ChatGPT will help you to make MOUs very well. You can fine tune it and say, these are the headings in which I want to address this MOU. Fine tune it, it is fast, it's like an assistant who will make and give it to you for making changes. Staff attrition is a problem, but if you train more, in fact these are things we must think about. Having a Punjab training for this data entry operators. Why can't we do that? We can do that. People will come join, they will make the pay also. I've been telling this to all my societies to do that. Only HCC has started this data entry operator training, and on a regular basis. I'm telling you that they are the ones who are going to make the changes in the research. You may have all the ideas, you may have the protocols ready, but unless that accurate data comes into that Excel sheet cell, you can't do anything about it. So getting them to train between the groups is one option. You can start with something, and in fact it can be a degree course for 2 years. We must do it in Faridkot as a course, data entry operator course.
So yes, we can do in the skill development.
Yeah.
We are planning to start this.
Yeah, please do that, because that will fill a lot of gaps in everybody's mind of getting that done. Ethical challenges, vulnerable populations, LMICs, and this is something which you have to ask yourself, am I ethically, morally right, and not just legally right? So, NBCTR is very good approval processes now they have, and as Dr. Tanuan said, they are ready to help you the entire machinery in ICMR is wanting you to do more trials and giving you money to do the trials. So please do that. I just put a Google search on what are the emerging areas where India can lead under these specialities. So you can start your ideation process from there. Your ideation in your area of research, you say, what is the leading things which are happening in the country in your area. Now, understand that if you do a clinical trial on your own, you may not be able to do more than 1 or 2 in a year or 2. Because that's a time it takes, an effort to drive a trial with recruiting people, buying machinery, etc. You are an all-in-all person in this. We do not have, we have to get into procurement of equipment as well. So that takes away a lot of time. But you can be a co-PI to your other areas, other teams. So that way you can be involved in many trials. Earlier there was a cutoff saying that you cannot be more than 6 trials you cannot be part of. Now that has been removed. But at least you need to do justice to the time you spend in a trial and the clinical work you have. So that is various specialities just for your notice. This is not, you can get the same thing if you put a ChatGPT query and then you can drill down to your level of interest. That is in ophthalmology, ENT, urology, almost everything including Ayush and hematology, it has come in. Now it is better. The speed was very poor earlier in terms of getting an approval, institutional ethics committee. The thought itself, you don't want to do the trial. Because I will write it and sit on it for 6 months, they will do a query. By the time you have forgotten what you have applied for. So all those are slowly removed now. It is getting better in the country. China has been fantastic. And if I look at hematology-wise, CAR cells therapy, which is the in thing now, China has now surpassed everybody else because of their drive and collective knowledge on clinical trials have gone up significantly. So we need to have the essential pillars. Essentially, when we do a trial, we should say who are the regulatory authorities who are going to look at you and ensure that your quality is good. So GCP compliant documentation will come across the ALCOA C, attributable, legible, contemporaneous, that means it should be live, and I forget O. Anybody can tell me what is O in this? Forgot. Alcohol. You can put a search, please, fastest finger first. You can tell your residents to write notes like this. Alcoa C. Fastest finger first. You can search, you should use your mobile, laptops, notes, real time, so that original. And last A is accurate. So when we analyze the data in the end, it has to be accurate, it should be complete. Then only you have data integrity to that. Okay? So anything you document, so unfortunately or fortunately in the trial scenario or in a research, documentation is the key. NMBH, if you apply to, documentation is the key, because the principle is, if you have not written down, it means you have not done it. Okay? And say what you do and do what you say and write it down. And data integrity is key, and Intel contribution is slowly improving now in terms of development and practice changes. The future directions are digital transformation and policy recommendation is what we need. Now, digital use in all these 12 domains, we'll have a session tomorrow on what are the, I may be able to scratch the surface only, and what are the digital tools you can utilize to improve the process. There is an e-consent process, and there is various digital things can be done in the thing. And for tomorrow, what you need to do, if you go home after this, and I said one of the begin with the end in mind philosophy, maintain a documentation quality, and that is something which government medical colleges may suffer with, because you do not have an HIMS or a documentation system. Time is a problem. You need to devise some way or the other to overcome that. Maybe have a research coordinator sitting outside. As soon as you finish, they should come with their card and enter it and then go, or enter it and then come to you. Some device you need to this based upon the research interest. You may not be able to do for everything. Understand the protocol deviations. Once you say, when in a clinical trial and there is an SOP, if we have gone again, the patient is supposed to come between 3 and 5 days of starting the treatment. He came on the 6th day. We still capture the data, but we have to inform the ethics committee for all the deviations. Like the child going to the father or mother and saying, I made this mistake, please forgive me. But document it. You cannot oversee that without having addressed that problem, because the more protocol deviations you write, the less chances that you deviate next time. It is like incident reporting or error reporting process. And we have to master the SAE reporting process, so that is another ingredient for clinical trials, especially pharma-driven trials is extremely important. You will get more opportunities to attend investigator meetings, especially if they get into clinical trials from pharma-driven industry. They have investigator meetings for every study which is there, and there the entire protocol is discussed thread by thread, and you will get to know more of that, how to go about it. Big and internal departments, CRC pipeline, and you have already your MRU. It's all about putting the right people in and taking it. So I will end my session there. Before the next tea break, I will request Dr. Ranjit will give a little. No, I've just left it there, so the continuity part of it, you can go with your official introduction. Thank you very much.
Thank you very much, sir, for very, very engaging presentation. Now we have with us Professor Dr. Sanjay Gupta, sir, Principal GGS MCH Faridkot. Welcome, sir. Sir is always there for motivating all of us, the organizing team, for the smooth conduct of this workshop. I request sir to please come on the dais to have a word of it, sir. Thank you, sir.
Starting with the name of the God, Waheguru da Khalsa, Waheguru di Fateh, Jo Bole So Nihal. Good morning, everyone, and very happy to see all of you, respected medical superintendent, Dr. Joseph, Dr. Tanu, my colleagues, and all the participants. First of all, I welcome all of you. It is a medical college, and I always tell that Rabindranath Tagore told the point that a teacher is a person who is still learning, not only the academic subject. Learning that related with the subjects, that related with the human behavior, related with the human life, related with the human environment, related with the economy, related with the change in the world, and all the effect on our subjects. So I congratulate to all the organizers for this clinical trial workshop, and best wishes for your workshop. Thank you very much.
Thank you so much.
Please
sir.
continue. मुझे ना बीसी सामने बुलाया मेरे को जाना है.
Sir,
Thank you.
thank you so much, sir. Our next speaker is Professor Dr. Ranjit Singh Meshan. Sir is Associate Professor and Head at BCRIC and Research Scientist in Department of Pathology, CMC Ludhiana. His area of expertise is molecular diagnostics, genetic research, hemoglobinopathies, and prenatal diagnosis. He has fetched various grants as PI from multiple projects from different agencies, and he has many publications in indexed reputed journals. please come forward for your valuable talk on ethical principles. Welcome, sir.
The outline of my talk is introduction, then international crime trial for after World War II, eugenics, the inhuman experiments, Nuremberg Code, Trustee Syphilis study, then Belmont report, declaration of Helsinki, and compilation of unethical researches followed by conclusion. So I thank the organizers to giving me the opportunity to come here and speak before you all. And the interesting thing was today we had a very nice breakfast in the morning, followed by a hand wash session. I went to the washroom to wash my hands, and for a moment of time I thought I washed my hand and I thought that I'll go away, next person can come and switch, like close the tap for me. Then again I thought no, I wash my hand, I close the tap, go back. And when I came out of the room, of the washroom, I saw a board saying ki, please switch off the, like, close the tap once you wash your hands. So this is, I think, ethics. So when no one is watching you, what you do. So I was sitting there washing my hands. I could have easily left without switching off the tap, like closing the tap. But then, right or wrong, good, bad. So here comes the ethics. When our moral says what we should do should be correct. So this is ethics, and when no one is seeing, what you do is ethics. We start with after the World War II, which continued from November 1945 to 1949. The world had its first international crimes trial at Nuremberg, Germany. After the name of this place, we got the Nuremberg Code on the ethical principles. So this international trial was Germany versus sort of the rest of the world. So why was it? Because during the World War II, the Nazi-controlled government had taken up a lot of atrocities on the downtrodden and vulnerable class of people in the name of research and good for the humanity. But actually they were torturing the normal people. So all this, what has happened during the Nazi atrocities, has been documented in a book, Doctors from the Hell. And this is the place where the trial was taken up, the Palace of Justice. So what the Nazis did was, in the name of research, in the name of good, in the name of giving health to people. So they took the support of science, so-called theoretical science, the eugenics. Eugenics said that there is no place in the society for the genetically defect people, the people with birth defect, they have no place in the society, and better garibi mat hatao, garibo ko hi hata do. So they thought that they have no place in the society, so rather eliminate them from the society, and eliminate in a very noble way. So what is the noble way? Use them in research, so-called betterment of the society. So what they did is, they picked up all the vulnerable people, the person with mental illness, terminally ill people, they picked up all these people, and Jews they picked up, which they were not very good with, put in the prisons, and all sort of fancy experiments were done on all these people. So they become the guinea pig, and they have no ethical rights. And they also gave up a formal poster, an advertisement saying how they are going to benefit by eliminating these people. So this is the amount which they spend on one vulnerable people. So if they remove these people, they will be saving this much of money. So the kind of inhuman experiments that they took up is, you can see, putting people into chambers which duplicated conditions of 68,000 feet. They will put people into gas chambers, let them die mercilessly there, take out their brains, and give it to the physicians for some experiment. We don't know what experiment. And a picture says there is a pile of brain tissue lying there in the name of experiments. Amputation of the whole leg up to the hip and trying to get it transplanted into another person. And they murdered 112 live Jewish inmates and defaced them for skeleton to study, say, anatomy. Injected gasolines into the human. So these kind of weird experiments they were taking up. So all this, they might say that it was good, but it was actually an unethical principle because they were king. Nazi was, Adolf Hitler was the king, and the doctors that time, they also thought themselves of king, and they thought that ethics is there. They can do whatever they feel like. So after this, after that international trial, so we came up with the Nuremberg Code that henceforth, if we have to conduct a research, medical research, clinical trial, so what we should do with what are the rights and what should be our ethical background, what should be the principle behind this. So we came up with 10 points of Nuremberg Code. So I will enumerate one by one. So the first, it says the voluntary consent of human subject is absolutely essential. So whoever is the participant in the trial, we should take the informed consent of the person, and no trial should be without in absence of the consent of the person. And to get the consent, we should explain what the trial is all about, how they can benefit or how they can contribute to the trial. Then the experiment should be such as to yield fruitful results for the good of the society. When we are involving a human or a participant in the drug trial or a clinical trial, it should be worthy that something good or fruitful will come up. It should not be vague, that just for the sake of doing research, we are doing research. Like sir said, if we have money, just go for research is not the thing. We should have purpose of our research. So it should be fruitful. And the experiment should be so designed and based on the results of animal experimentation, and a knowledge of natural history should be there. So right away we should not take a drug and put it and expose it humans to the drug. So we should have background study on the of the same drug or the molecule on animals, so that we can say that it's grossly safe for humans. The experiment should be so conducted as to avoid all unnecessary physical and mental suffering and injury. So because someone is consenting to be a participant in the trial, and because he has, on goodwill, he has consented, you should not unnecessarily open him to unnecessary torture. Suppose you unnecessarily don't ask for biopsy, invasive biopsies and all. So if it can be avoided, you should avoid it. You should not expose person to unnecessary painful procedures. No experiment should be conducted where there is a prior reason to believe that death or disabling injury will occur. If we beforehand know that this kind of death or extreme situation can arise, we should avoid the thing. And at least if the physician doing the research himself is involved, then we can think of. So we can cancel a study if we feel that extreme dangerous side effects or end effects are expected. The degree of risk to be taken should never exceed that determined by the humanitarian importance. Like the benefit should outweigh the risk which we are exposing the participant to. Then proper preparation should be made and adequate facilities provided to protect the experimental subject against even remote possibilities of injury or disability or death. So in case of any adverse effect, we should be there to help our participant to recover. So we should have a plan B to what to take up if some emergency comes up during the experiment. The experiment should be conducted only by scientifically qualified people. So whoever is taking up the experiment should be qualified enough. During the course of the experiment, the human subject should be at the liberty to bring the experiment to an end. So our participants, once they sign the informed consent, they are not bound to be with us throughout the trial, till the end of the trial. We expect them, but whenever they feel like they don't want to continue, they should have the liberty to stop the trial, to withdraw from the trial. They should have been given the option to withdraw the trial at any moment of time. During the course of the experiment, the scientist in charge must be prepared to terminate the experiment at any stage. If the scientist feels that the experiment is no more going to yield any fruitful thing in the future of the study until now, then he should be able to terminate the study then and there, and not unnecessarily expose the participants henceforth. So this was the Nuremberg Code, 10 Nuremberg Code points. So then there was an unethical clinical trial again, which continued for 40 years, 1932 to 1972. This is the Tuskegee syphilis study, and this was a grossly unethical trial from the beginning to the last end. So this was 40 years trial conducted by the US Public Health Service government. Who were employed was poor black people, the vulnerable, because they were poor. They were given, they were offered money to get into the trial. They were said that we will give you money, you get into the trial. So they were vulnerable. So they were sort of coerced into the trial, and it stood on the basis of lie. The basis of our trial should be truth. We should give the participants the actual data and the actual information about the trial, what we are expecting and what we are going to do with them. In this trial, they were told that they are testing some medicine on the participants. But actually, what they were doing is they were trying to see what is the natural course of syphilis. So actually, what they were giving was just placebo. They were not expecting any betterment of their participants. So what had happened? The key timelines are from 1932 to 33. They had recruited 319 black men with syphilis and 201 with normal controls. Even if penicillin was there, which could have helped cure syphilis to some extent, they were not administered this, and they were not given the opportunity to use penicillin. The ethical code by Nuremberg, which was already there, was totally ignored. They didn't give proper information to their participants. Then finally in 1972, this Jean Heller exposed this study publicly that this is an unethical clinical trial, and the study was immediately stopped in 1973. And later on in 1997, the US President Bill Clinton issued a formal apology for this unethical trial. And the final what happened is 28 deaths were due to the syphilis in this study. 100 men developed serious complications, and 19 children were born with congenital syphilis. So after this, the Belmont report 1974 was published, and this Belmont report gives an overview of what are the fundamental ethical principles that should be considered. So the basic ethical principles that should be considered are: we should have respect for the persons who are participating in the trial, beneficence and justice. Belmont report also made a difference between what is clinical practice and what is clinical research. And what is the clinical practice is, in the clinical practice we are actually involved with an individual. We are looking at one person, and we are trying to cure or treat that one person. But for research, we take up a hypothesis and we try to prove or disprove that hypothesis, whether we want to check our hypothesis whether it's correct or wrong. Then expected outcome, for a clinical practice, the expected outcome is sort of good, because we know what is the course of action, what is the course of drugs, which is already known. But in research, the expected outcome is not certain. Then the orientation is, we are in the practice we are focused on the individual patient, and in research we try to draw an inference from the study. So all ethical guidelines and principles are applicable to research, because in practice we don't expect this. So the Belmont report principles. So the principle respect for person. So it says that individuals should make their own informed choices, and extra protection for those with limited autonomy. So all the people who can give their autonomy, who can decide have a self-determination and can decide about themselves, should sign an informed consent form to be included in the study, and they should be informed on what is the course of the trial. So how to approach this? So we should get an informed consent from the participant in a language which is understandable by the participant, and we should always protect the vulnerable groups like the children, mentally disabled, and the seriously ill people. Beneficence means the physician or the one who is taking up the trial, he should have looked out that we should minimize the benefits of to our participants rather than giving any harm to our participants, and to ensure the well-being of our participants. So a risk-benefit analysis should be done priorly, and we should try to reduce the risk to the minimum as much as possible. And justice, fair balance of research between burdens and benefits. So burdens and benefits should be analyzed previously, and we should ensure the fair selection of subjects for a particular participant. What are the subjects that should be recruited? It should be fairly selected. In 1966 again, Henry K. Beecher, he published a paper, Ethics and Clinical Research, and he compiled all the research papers which were unethical on clinical trials. And what he saw is, what he published is 22 unethical clinical studies from reputed US institutions, and published in high impact journals like NEJM, JAMA, JCI. So these are high impact journals, but they published 22 unethical clinical studies which did not follow the normal ethical principles. And all these papers are published before the Declaration of Helsinki. So later on, this Declaration of Helsinki ethic principles were brought up. So Declaration of Helsinki first was given in 1964, and it has 12 principles which are adopted by the World Medical Association in 1964 to guide physicians in biomedical research. And it also gave the basic guideline for the Ethics Research Committee, which we often quote in our research publications. Like this study was a common line which we put when we submit our manuscript, that this study was approved by the Institutional Ethics Committee. So Madam will discuss it later on. So what are the research ethics guidelines is, we should definitely take the approval before starting the research. That it should get a ethical approval from the Institutional Ethical Committee. So this is the checks which we should put in, so that what comes up are ethical researches and not the Nazi unethical researches which we have seen earlier. So these are the points which the guidelines which Declaration of Helsinki has proposed. With this I conclude. The medical research is indispensable for the progress, health and well-being of the society and the humanity. It saves life and prevents disease and improves the quality of care which we give to our upcoming generation. However, scientific advancement must never come at the cost of human dignity, rights or safety. So ethics must guide research and not follow it. Thank you.
So just to get the discussion going, can you give me the mic? Feel free to ask questions. So we are now here and ethics and regulatory approvals. So to Ranjit, you stand here. Be here. Some questions I want to ask them. There are some chocolates here. If you answer the questions, you all have some candies kept there to give some ATP's boost. So basically, if somebody wants to do a study on retrospective analysis of patients who have had a complication of malaria, cerebral malaria and cerebral malaria, you want to do a retrospective study, do you need ethics committee approval?
Yes.
Why? Can you, please talk about. Yeah, you have a mic there?
No, I don't have.
Okay. Okay.
In the retrospective studies, again the patients or human will be involved, so we have to take the consent of that also. So if permission is required.
So consent is required?
Yes.
So it is a
But the
retrospective.
point is how we will take the consent of the patients, because we have the previous data.
So what is the next best option then?
Then
Don't do the study, do the study.
we can do the study by, I don't know exactly which.
Somebody was answering. Yeah.
Approval from the ethical committee is required in retrospective.
Correct. So now the question is that can you take informed consent of the patient or no?
Showing the confidentiality of the test.
Absolutely. So you can take a waiver from them saying that yes, we have said that we are going to do, and we will. So I want you to ask such questions in the discussions coming forward. Okay, so then only you will go back home enriched, and we will teach each other on what the practices are. We will break for tea now.
So you are asking for one interactive session?
Interactive session we'll come back and have. Let's go for tea and come back, and we'll have more space generated here. This is too congested.
Yeah, sir, we'll do that.
Okay.
Yeah, but consent the meeting.
Go ahead.
So tea break is there, and we all can have refreshments now. We will meet here after 15 minutes.
Which one? Internet is not working.
And when you get the new version, off. Yeah, अभी के लिए off.
You remember your numbers? Yes. So now, okay.
4. Sir is 4.
All right. 4. So now, all ones. This is table number 1 on my right, the front table. This is table number 2. That is table number 3, and that is table number 4. So before we attend Dr. Amit's session, can we please exchange the tables? All ones on table number 1, all 2s here, all 3s that side, and that is table number 4.
It is so flexible. It is so flexible. I can automate better.
Actually, I've been requested to talk in the mic because I forgot, and there is some Zoom thing also going on. I may not be audible to the people who are connected there, so we'll use the mic. So okay, let's go ahead hurriedly. Now we'll do the thing that we do in medical education also very commonly. We make a pair of 2, take 1 minute, and after exactly 1 minute, make pairs of 2 each, right? Regardless of which table you belong to. Pair of 2 to each, and you exchange your introductions with each other, and exactly after 1 minute, you introduce each other. So 1 minute for you to find out the introduction of your partner. Pair up immediately, please.
Same table?
No, not necessarily. If you are odd number on one table, you can pair with the partner on the next table. That's okay. Last 10 seconds, please.
I want all the faculty also.
There's a request for the faculty also to do that. Alright, time is up. We start with table number 1. Right? We start with the participants first and we'll move on to the faculty.
Good morning, everyone. I would like you to I would like to introduce you to Ma'am Dr. Sunny Chopra. She is Associate Professor in Department of Biochemistry from GMC Patiala.
Good morning, everyone. She is Dr. Jashan. She is Assistant Professor at GMC Amritsar. Very nice to meet you, ma'am.
Good morning, everyone. She is Dr. Neeta Goyal, Professor, Family Medicine Department, GGS at MCH.
Hello, good morning, everybody. I will like to introduce to you Dr. Monica Sharma.
Dr. Monica, please let everyone know.
She is Dr. Monica Sharma. She is a scientist in ICMR and ICH DR.
Please pass on the mic to table 2. Yes.
Good morning, everyone. Let me introduce you Dr. Vikas. He is Associate Professor Surgery from Government Medical College, Patiala. Thank you.
She is Dr. Ruby Agarwal. She is working as a woman scientist in ICMR in Safdarjung Hospital, New Delhi.
Ma'am is Dr. Manuja. She is working as an associate professor in emergency medicine in GGS MCH Faridkot. She has completed her MD, MBBS and MD anesthesia from BJMC Ahmedabad. Worked in critical care in Scots, Delhi, then transferred to DMC Ludhiana, and now ma'am has been working here and started the Department of Emergency Medicine here.
Can I request the participant to just stand up so that there is no confusion, please? Can I request the participant who is being introduced to stand up so that everybody knows who is being introduced? Now you are going to introduce sir, na? Sir, please stand up. Ma'am, you can take the stand now.
Good morning. I'd like to introduce Dr. Tebra Singh. He's MBBS from AIMS, and further he did his MCH also from AIMS in pediatric surgery. He started his career as a faculty in GGS Medical College, and then he moved to Patiala and started the department there.
I would like to introduce Dr. John Abraham, who is working as an assistant professor in the Department of Hematology at CMC Ludhiana. He's a graduate and a postgraduate of CMC Ludhiana, did his DM in hematology at CMC Vellore, following his senior, Dr. Joseph, onto his footsteps.
Thank you. Let me introduce Dr. Faridkot. She is an associate professor, Department of Obstetrics in GMC Amritsar. She is an alumnus, both MBBS and MD from GMC Amritsar. Thank you.
Please pass on the mic to table number 3.
I would like to introduce to you Mr. Kushal Jain. He is a technical assistant at ICMR and ICST with a background from biomedical engineering.
Good morning, everyone. I would like to introduce you to Dr. Agam Jain. He is from ICMR and ICHDR. He is currently posted at MRHRU, Bhunga, and he is a project scientist.
आप दोनों तो जैन और एक ही इंस्टीट्यूट से इकट्ठे हो गए, तो आपको अलग करना पड़ेगा। आप भी मैं इसके बाद आपके पास ही आता हूँ।
Good morning. I'd like to introduce Dr. Jashan Mitra. He is from GMC Amritsar, working as assistant professor in Department of Community Medicine.
Good morning, everyone. My teammate is Dr. Gaurav Sethi, assistant professor in Department of Psychiatry at GMC Amritsar. Sorry,
Good morning, everyone. I would like to introduce Dr. Kirti.
She is assistant professor in Department of Biochemistry at GGS Faridkot.
Good morning, everyone. I would like to introduce my teammate, Dr. Harinder Pal Singh. He is working as assistant professor in the Department of Internal Medicine from GGS Patiala. Thank you.
GGS, I introduce myself. I am Dr. Harpreet, professor in the Department of Herbs and Dietary, Faridkot, Medical College, Faridkot.
Thank you so much.
So it's my privilege to introduce Deepika. She is a technical assistant in ICMR and ICHDR New Delhi. So we are privileged and proud enough to have ICMR team over here and teaching us the nuances and being a part of our group. Thank you very much.
I would like to introduce Dr. Varun Paul, Professor in Pediatrics, GGS.
Good morning, all. I would like to introduce Dr. Pradeepa Faridkot, Department of Physiology, GMC Amritsar. It gives me immense pleasure to sit with her because earlier she was at Patiala.
Good morning, everyone. My name is Dr. Renu Bansal, working as professor in the Department of Microbiology at GMC Patiala.
Good morning, everyone. I would like to introduce Dr. Nitin Athwal sir. Sir is professor and head Department of Surgery at GGS MCH Faridkot, and sir is also vice principal of the institute.
Very good morning, everyone. It's a privilege to introduce a colleague, Dr. Himanshu, who is associate professor in the Department of Medicine at GGS Faridkot, and is also working as deputy medical superintendent in our
Can I now request the faculty also to introduce each other? And I will take the last call.
Okay. So I would like to introduce Dr. Rinchu Lumba. She is professor in Department of Biochemistry at Christian Medical College, Ludhiana. She did her graduation from this institute, Guru Gobind Singh Medical College, as a 1992 batch student. And she did her post-graduation from CMC Ludhiana and started in 1999 and completed in 2002. So thank you so much, ma'am.
Thank you for introducing.
Whom are you introducing?
Okay, I'm Dr. Vineet Jason. I'm a neurologist from CMC Ludhiana. With my MBBS, MD, DM from CMC Ludhiana.
So I'd like to introduce Dr. Neetu Kakkar. Neetu Kukkar. She has done her MBBS from Bellary, Karnataka, and then MD from DMC, and I understand that she did 1 year of internship in CMC as well. And after that, she is holding the medical superintendent post, MRU in charge, and DHS post for the entire Punjab. So very powerful person.
Now I will introduce Dr. Joseph John. He did his graduation and post-graduation from CMC Ludhiana, and he did his DM from CMC Vellore, and presently he is professor in head Department of Hemato-Oncology. And he is today the most strong, I think, resource person today, and he will be organizing this whole workshop. Thank you, sir.
I think many of you all know my name because I was putting all the messages and coordinating with everyone for organizing this event. So I welcome you all. I am Dr. Richu Singla, Scientist C, funded body of, I am working in MRU, Multidisciplinary Research Unit at GGS Medical College, which is a funding body of DHR and ICMR. Thank you.
Okay, so we have two Rinchus with she is without N and she is with N.
So sitting next to me is Dr. Rupi Arora, and she is now standing now next to me. And she is clinical pharmacologist in the Department of Clinical Hematology, Christian Medical College, Ludhiana.
Good morning, everyone, and I would like to introduce Dr. Ranjit, working as a research scientist in Department of Pathology in CMC Ludhiana. Thank you.
Good morning all. So I'll introduce Mr. Himanshu. He is a biostatistician in our clinical hematology department.
Good morning everyone. I am introducing Dr. Daman. He is a clinical research coordinator in clinical hematology of CMC Ludhiana.
Before I come to last but not the least, did you all observe something? Even despite the organizers' efforts to make you mingle with each other, you found your comfort zone. So leaving the comfort zone probably is the most difficult thing, and the whole intent of doing this activity was to make you conversant with each other so that we can find the ideas, discuss them and materialize them in a multi-centric study. We do that. But again, we tend to be comfortable with our own mates whom we know, so we have to come out of it. And now let me introduce your next resource person, Dr. Amit. He is Professor, Department of Pharmacology. He is my college mate, a graduate of 99 batch from GGS MC Faridkot and a postgraduate from GMC Amritsar in Pharmacology, now working as Professor in the department, GMC Faridkot. Sir, over to you.
Thank you so much, ma'am. So अपनी प्रेजेंटेशन को ले आए पहले. So आप लोगों की मिक्सिंग अच्छी हो गई है, बट अभी भी मुझे एक इशू है कि पटियाला वाले के साथ बैठे हैं, दिल्ली वालों के साथ बैठे हैं और अमृतसर वाले अभी भी अमृतसर वालों के साथ बैठे हैं, फरीदकोट वाले भी फरीदकोट वालों के साथ बैठे हैं. So but for the next 2 days you will require the coordination of this whole group. So your group will be fixed for the next 2-3 days workshop, and you will be presenting, preparing a protocol as in the workshop. You will be preparing a protocol which will be assessed on the last day. Right? So this will be your group who will be preparing. So all of you must coordinate. So another activity which we do in medical education is we decide 3-4 roles of every group. So I would like you to have some group leader, right, who will make and ensure participation of every participant. Second will be a timekeeper, because you will be given a limited time for your activity. So second person who will be a timekeeper. You can change these roles in each activity. It is not a mandatory thing कि एक group leader बन गया तो वही group leader रहेगा, या एक timekeeper बन गया तो वही timekeeper रहेगा. Second, timekeeper. Then the third important role is compiler, who will write down, because you will be preparing a protocol, so you will have to compile something. Whatever you are discussing, you will have to compile. So there one of you must be a compiler, and because there will be a presentation, might be intermittent presentation or at the end, so one of you will be a presenter, right? So these 4 roles, we are giving you 30 seconds to decide these 4 roles amongst yourself for this today's session, not for tomorrow, for today's session, or you can change it afterwards. 4 roles. You have to decide who will be the group leader, who will be the timekeeper. Timekeeper is the easiest person, so everybody will say I will be the timekeeper. So you just write down the name of your group leader, your compiler, so that it is on record. Another important thing for any activity is documentation. So you must record whatever you are discussing. So I hope you have discussed your roles. It is still going on. So we have limited time, kindly hurry up.
These rules are only for the workshop activities and not for the final submission of the protocol.
Yes. workshop activities, these roles, because your group will be presenting some activity in the end, and protocol is to be finalized. Right? So I hope you have completed the task given. You have decided your 4 roles in your group. Group 1 and 2 are still discussing. Hope so you have finalized. Okay. Just recording the proceedings. Okay. So can we start? So good morning everyone. मुझे नहीं है चाय पी के तो एक्टिविटी बढ़नी चाहिए आपकी कम हो गई गुड मॉर्निंग एवरीवन स्टिल स्लाइटली लो थोड़ी जाग जाओ चाय पी के सर आपकी आवाज आई बाकी किसी की आवाज नहीं है सो वी स्टार्ट So clinical trials, how many of you have some idea about this clinical trial? Because what I saw sitting at the back, the Delhi team, whenever you say a word, the Delhi team immediately goes to ChatGPT and starts searching for that particular thing on their laptop. So it is good, but remember one thing, what I have observed is AI is as intelligent as its user. So for using AI, you have to know the thing. You have to extract the information from AI. You can't without knowing the subject to some extent. Right? So we would like to introduce clinical trials to you. So why clinical trials? सबसे पहले तो we must know what is a clinical trial. Anybody? लिखा भी हुआ है. Anybody who can tell me? So there are the each and every word is very important. One, it is controlled. This you must be aware. It is every clinical trial is a controlled study, right? Because once the drug is launched into the population, drug is launched and prescribed by the physicians, it becomes uncontrolled because they prescribe it to every patient. But in clinical trials, it is a controlled research study. So mind it is a research study, and because it is a clinical trial, so it is on human volunteers. So each and every word has its meaning. This you must be able to understand. What are clinical trials? And why? As Sir has very rightly said, after ethical consideration, before going into clinical trials, we tested on animals. Now there is some advancements in those research. We have come up with in silico research, and there is a new thing which is coming up. Animal experiments probably will be very reduced or might be obsolete 10 years down the lane. So in silico research is now coming up. Every drug is being tested, a molecule is being tested to become eligible for a drug in computers, right? So AI is helping in that quite a lot. AI is working up day and night, right? Then once the drug, either you go in silico or you go preclinical studies, there is some molecules which are finalized that okay, these molecules, they are potential candidates to become a drug in a particular disease, right? And in addition to all their efficacy, those in silico and animal studies, they give us some adverse effects which come in the animals or which come with the AI. And they give us the pharmacokinetics of that drug. There are two aspects in pharmacology. One is pharmacokinetics and dynamics. Dynamics is this. Kinetics is this. Side effects, the benefits. Then, before FDA, even ICMR and DCGI is the regulator for this in India. So DCGI approval and market release, this clinical trials are required, right? And earlier, we used to say it takes more than 15 years. Now the time has slightly reduced. It will probably reduce more because of this in silico research, and the cost might also reduce, right? And these preclinical testing in animals is very important for us to understand. Without submission of this data, a company cannot file IND. IND is investigational new drug application, right? So for investigational new drug application, this data as well as the data for production. All those, they are running simultaneously. They have to be submitted, and then there is official review by the regulatory bodies, and then the drug is approved for clinical trials, right? After IND, drug is approved for clinical trials. After NDA, NDA is another term, new drug application. Once it is approved, then the drug is launched into the market, right? So this is how it happens. Once first the step is discovery, which is going on. A lot of drugs are being discovered. They are going on in silico. They are going on. A lot of research is being done on this first part. Few potential candidates, they come to this, and very few they come to this, right? And this investigational new drug application is followed by all the phases, phase 1, 2, 3, 4, which is the topic for today. Then this is the how it happens. Around 10,000 compounds, they are tested at the drug discovery phase. Out of this, say there are only 250 compounds. This is just hypothetical. It is not the actual scenario, but this is to give you an idea that from 10,000 compounds, there are chances that only one compound will become the drug, right? So this is the gravity of the situation right now, and this is important. Around 20 years back, another phase has been introduced into this, that is known as phase 0. So you can start with phase 0, phase 1, 2, 3 and phase 4. These are the various phases. After an investigational new drug application is filed and the process and gets approval for clinical trials, every drug has to pass through these phases, right? So we will start with first phase, phase 1. So phase 1 is the majority aim of this phase 1 is just to determine the safety, tolerability and pharmacokinetics and optimal dosage range. So this is the aim of phase 1. So it does not check about the efficacy of the drug, whether the drug is effective in that particular disease or not, right? That is why the participants can be healthy volunteers in phase 1. Because the goal of this phase is not to check the efficacy. The goal of this phase is only to determine whether the safe, as sir has very rightly said, first principle is do no harm, right? So for that principle to be achieved in clinical trials, this phase is conducted. Phase 1 of clinical trials. It can be on healthy volunteers and it can be on the diseased patients as well for the same disease, right? So this is important for us to understand. Why? Because the number of participants is very low. 20 to 100 participants are enrolled for phase 1 clinical trial, and it takes several months because various pharmacokinetic studies, various excretion, how this drug is metabolized, how this drug is excreted, they all are being tested. And safety, side effects and pharmacokinetics of the drug are tested in this phase. And only 70% from those molecules which are being tested for phase 1, very few they get to phase 2. Because the moment it is not safe, it is found in phase 1 that the drug is not safe, the drug is discarded, right? The further research for that drug will be over. Only if the drug is found safe, tolerable and it has optimal. Why dosage is being calculated? Because in animal experiments, the dose is given from the animal experiments, preclinical study. Dose is being calculated from that. But whether that dose is actually reproduced in the human beings, or it is producing some adverse effects, or it is less than the actual dose, that can only be detected by this phase, right? So if that same dose which is being approved by preclinical studies to be used in human beings, extrapolated, that is the appropriate word is extrapolated. So the dose calculated in animals is extrapolated to human beings. So that extrapolated dose, whether it is appropriate and can be used in human beings, is tested by in this phase. So once a drug crosses this barrier, then the next phase comes the effectiveness phase. So mind it, this is the phase where the actual efficacy or the use of that drug in that particular disease is being tested in phase 2. What is the difference between phase 2 and phase 3? I will let you know. The only difference between phase 2 and phase 3 is this: number of participants. In phase 3, more number of participants are being enrolled, right? And it, so एक चीज हमेशा याद रखना, ये वाला कंपोनेंट हर फेज में आएगा. ये वाला कंपोनेंट सेकंड फेज से स्टार्ट होता है, बट ये वाला कंपोनेंट फेज 1 से स्टार्ट हो जाता है. It is ADR testing is done right from phase 1, and wherever some problem is there with this ADR, there and then only that trial is over. कोई lethal adverse effect आ जाए. अभी आप लोगों ने शायद आज 1-2 दिनों पहले पढ़ा होगा कि SGLT2 inhibitors जो हैं बहुत commonly यूज कर रहे हैं. Internal medicine people, they might be using it very commonly. SGLT2 inhibitors. Now it has come to light from some study in Kolkata that those SGLT2 inhibitors are causing hyperosmotic diabetic ketoacidosis होता है, but they are causing बिल्कुल normal levels of blood sugar, and still SGLT2 inhibitors are causing ketoacidosis, and there are a lot of patients reporting there. So even after marketing, the drug is being launched into the market, still this part is still going on. This we have to understand, right? And this is just a preliminary efficacy, and only less than 33% of phase 1, phase 2 candidates, they proceed to phase 3. Very few, right? And this phase 3, this is वो preliminary testing phase 2 में हुई थी. Phase 3 is confirmatory and comparative, right? So phase 3 is confirmatory and comparative. So you can either compare it with some other drug, you can compare it with placebo, or you can use it as add-on therapy, whatever the trial methodology which you decide and design, that is done accordingly. This. So difference between phase 2 and phase 3 is this. We are doing the same, all those things which are being done in phase 2, but the participant enrollment is higher, right? It takes around 1 to 4 years for completion of this phase 3. Then there will be comparative efficacy. You all must be doing some clinical. Those who are faculty members, they might be doing some, they say drug versus drug B comparisons. So that is comparative efficacy. And safety monitoring is very important. So it is very important for us to understand that ADR monitoring or safety monitoring is important at every step. We cannot skip this. Then, so what happens after this phase? This is important. After this phase, successful completion of this phase 3, what the company does is, company accumulates all the data, company formulates a lot of pages and everything, a lot of files are there, and they file a new drug application to the regulatory bodies. In India, it is DCGI. So once a company is sending the data, so all the preclinical data, so there are important 3 things which are being submitted. One is the preclinical data. Second is all the clinical phase 1, phase 2, phase 3 data. Third is the manufacturing process, the conditions under which the drug is being manufactured at a larger scale, because once the drug is launched into the market, it has to be produced at a larger scale. What will be the safety concerns during production? What are the conditions, terms and conditions under which that drug is being produced? So that all that data is compiled and submitted to the regulatory authorities for a new drug application. And once the scrutiny of that application is being done and that application is approved as a new drug, this phase starts. Phase 4 is also known as post-marketing surveillance or long-term monitoring, right? So efficacy we have established in phase 3. No doubt efficacy is still being tested here, but efficacy, because here several thousand real world patients will be getting that drug, And this is important for phase 4. Again, we have come to phase 1. Long-term safety and identification of new uses. This is also known as drug repurposing. New uses are being tested. ठीक है, अगर कोई एक drug है, for example, you are using some drug, the drug has been tested in phase 3. आपको एक important example है, you must, मेरे को बहुत commonly use करनी पड़ती है, minoxidil. Minoxidil, when it was launched into the market, it was launched as an anti-hypertensive drug. But then the adverse effects of that drug came up. When it was launched into the market, many adverse effects they came up. So the risk-benefit ratio, as sir has said, risk-benefit ratio, risks were higher in patients of hypertension. So repurposing was done. But उसका एक adverse effect था, उन्होंने देखा, it increased the hair growth. So they repurposed that minoxidil into मेरे जैसे बंदों के लिए, ठीक है ना, जिनके बाल थोड़े कम हो जाते हैं, वो they use this drug, minoxidil. So this is identifying the newer uses of that particular drug. So it is phase 4 clinical trials. 2 minutes ले लो. So this is what is the brief. And so as very rightly sir has said, first thing which are very important in a clinical trial: informed consent, right to withdraw sir has already told, medical care and support throughout the study, and ethical board ensures the efficacy. Institutional ethics board. And in India, DCGI is the regulatory body. And you must understand this new drugs and clinical trial rules 2019. They govern the drugs and clinical trials in India. NCDT rules, right? And ICMR gives the ethical guidelines. ICMR, there are ethical guidelines given and updated regularly by ICMR. And this is very important for us to understand. Those who are involved in a clinical research, चाहे आप academic, because they have given another term, academic clinical trial. For that academic clinical trial, you don't require approval from DCGI. That you can do at your own. For example, the thesis which we are doing at our institute level, they are academic clinical trials. But very important for us to understand, every trial or every comparative study, every study, whether I would say observational study, it is, you can say retrospective study, prospective study, observational study, interventional study, whenever it becomes interventional study, it becomes a clinical trial. If you are observing just, it becomes observational study. If you are doing some active intervention, it becomes interventional study, right? So any interventional study becomes a randomized clinical trial, right? So you have to understand every study, कोई भी आप लोग कर रहे हो, थीसिस कर रहे हो, कुछ भी कर रहे हो, यहाँ पे रजिस्टर करना बहुत जरूरी है. CTRI, Clinical Trial Registry of India. हम लोग कह देते हैं CTRI है सिर्फ क्लिनिकल ट्रायल रजिस्टर करने, नहीं. Every study must be registered because उसका 2 एडवांटेजेस हैं. क्लिनिकल ट्रायल्स तो करने ही करने हैं, क्लिनिकल ट्रायल्स तो करने ही करने हैं, चाहे वो एकेडमिक क्लिनिकल ट्रायल हो, चाहे वो एक्चुअल क्लिनिकल ट्रायल हो, करना ही करना है. बट और भी स्टडीज करनी है, उसका एडवांटेज क्या मिलता है? whenever you go into some good publication, they ask your CTRI number. Right? So this, for that you have to register in Clinical Trial Registry of India. Right? And this is how it happens. You give CTRI registration, ethical approval, DCGI approval, then the trial is initiated. Once the trial is initiated, इसमें एक क्राइटेरिया है, कैच है, जो इन्होंने DCGI ने और न्यू ड्रग्स एंड क्लिनिकल ट्रायल्स में हुआ है. जो प्रॉब्लम थी पहले, वो क्या प्रॉब्लम थी कि अगर आप न्यू ड्रग एप्लीकेशन या इन्वेस्टिगेशन न्यू ड्रग एप्लीकेशन फाइल करते हैं, उसमें क्या होता था पहले कि उसमें वो प्रोसेसिंग होने में या उसका अप्रूवल आने में इट यूज्ड टू टेक अ लॉट ऑफ टाइम. सो अब उन्होंने एक केविएट लगा दिया है इसके ऊपर कि अगर 30 डेज के अंदर, यहाँ पे 90 डेज इज फॉर ग्लोबल ट्रायल्स एंड 30 डेज इज फॉर इंडियन क्लिनिकल ट्रायल्स. अगर आप DCGI को एप्लीकेशन फाइल करते हैं, आपको 30 डेज के अंदर कोई रिप्लाई नहीं आता DCGI की तरफ से, सो इट इज टेकन दैट योर एप्लीकेशन इज अप्रूव्ड. You can go ahead and proceed with your trial. 30 days window is being given by under these new drug and clinical trial rules, right? So आप एप्लीकेशन फाइल करेंगे, अगर 30 डेज में रेगुलेटरी बॉडी आपको रिप्लाई नहीं देती, so take it, assume it, your clinical trial is approved and you can move ahead and proceed with your clinical trial. Okay? Then e-provisions, compensation formula, this is very important. This you have to keep in your protocol. Compensation नया आया है compensation 2019 के regulations में. Then informed consent की audio visual recording, अगर आपके पास patient करना चाहिए, they have enforced it, given with force, right? And defined timelines जो मैंने आपको बताया, 90 days for global clinical trials, 30 days for Indian clinical trials, and they have expanded the definition of academic trials जो मैंने आपको बोला, जो आप लोग thesis करते हो, you don't require to submit your protocol to DCGI, it becomes an academic clinical trial. एक कंडीशन होनी चाहिए उसके अंदर, सिर्फ एक कंडीशन है एकेडमिक क्लिनिकल ट्रायल के अंदर कि आपका ट्रायल्स किसी भी कंपनी से स्पॉन्सर्ड या कंपनी की तरफ से नहीं होना चाहिए, और आपको जो ड्रग्स आप उसमें यूज कर रहे हो वो कोई कंपनी प्रोवाइड ना करवा रही हो, फंडिंग ना हो आपके पास कंपनी की, then only it should be an independent clinical trial that becomes a academic trial, right, without any funding. So this sir has already told, and we will discuss it in details as and when it comes. And this is important, e-consent and electronic data systems. तो आपके पास इसमें सबसे important जो भी हम लोग डिस्कस की, किसी ने बोला भी था कि आपका डेटा सेफ्टी, बिकॉज अगर आप ई डाटा कर रहे हो आपको सेफ्टी भी इंश्योर करनी पड़ेगी, तो कोई वायरस आके हैक करके आपका डाटा लेके चला जा सकता है, देयर आर चांसेस, so you have to ensure e-safety also, electronic data as well as electronic data safety also, right? So approval process, this will probably you will not be involved, those who will be going, their regulators will be probably involved in this approval process, and this is important. So every trial must be registered under CTRI. So why clinical trials? This is very important. हम लोग सब लोगों को आइडिया है एविडेंस बेस्ड मेडिसिन का. So for evidence based medicine, these clinical trials are a must. So first thing, which we have ethical principles, protects the patients from harm. As sir has very rightly said, there were many clinical trials which were without any consent of the patient. The Nazis used to do a lot of clinical studies without the consent of the patient, right? So this is, and this contributes, clinical trials, they always contribute to medical advancements, right? So any questions?
I'd like to ask, if you're doing some retrospective service, are they also supposed to be registered in the CTRI?
Yeah, you can do. You can register. There is no foundation to not to register.
Okay. Secondly, we talked about the drugs. Suppose a surgeon or somebody is evolving some new method
Yes.
to treat a disease like
Yes.
surgery.
So it comes under this ambit. New technique whenever you are doing something on the patients. So we are talking about drugs, but the word I used was intervention. So if you are observing, just observing, that is observation study. If you are intervening, चाहे आप पेशेंट को सिर्फ एक नॉलेज दे रहे हो, फॉर एग्जाम्पल एक स्टडी है कि हम पेशेंट को एक क्लासेस लेंगे, उसकी पेशेंट को हम नॉलेज एनहांस करेंगे, उसके बाद उसका इफेक्ट देखेंगे, दो वो नॉलेज देना भी इंटरवेंशन है, दैट बिकम्स अ क्लिनिकल ट्रायल, बिकॉज यू आर प्लेइंग विद द माइंड्स ऑफ द पेशेंट्स, प्लेइंग विद द माइंड ऑफ द स्टडी पार्टिसिपेंट. सो अगर आप सिर्फ नॉलेज भी दे रहे हो किसी को, वेयर एवर यू आर इंटरवीनिंग, वेयर एवर देयर इज सम इंटरवेंशन इन्वॉल्व, दैट बिकम्स अ क्लिनिकल ट्रायल. एक मिनट मैम, हाँ जी, कम्युनिटी स्टडी इन द सेंस, अगर आप वही मैं कह रहा हूं ना सर, अगर आप फील्ड में जाके सिर्फ डाटा कलेक्ट कर रहे हो तो कुछ नहीं है, बट अगर आप उसमें कुछ इंटरवेंशन कर रहे हो तो इंटरवेंशनल स्टडी बिकम्स अ क्लिनिकल ट्रायल. जैसे मैंने अभी आपको एग्जांपल दी ना, सर्वे, आप सिर्फ सर्वे कर रहे हो, दैट बिकम्स ऑब्जर्वेशनल स्टडी, बट आप सर्वे के साथ एक कोई एजुकेशन भी प्रोवाइड कर रहे हो और उसका इंपैक्ट देखने की कोशिश कर रहे हो, दैट बिकम्स अ क्लिनिकल ट्रायल. KAP तो मैम observational study है ना, you just are taking the data of the patient, you are not intervening. So intervention, any kind of intervention which you are doing becomes a clinical trial. So,
what is the expected outcome of the academic trial and the full-fledged trial?
so expected outcome is as per the protocol. Full-fledged trial, the only thing here is in full-fledged trial you have some sponsor, whereas in academic clinical trial you don't have any sponsor. That is the major difference, because these guidelines, 2019 guidelines, they have defined the role of sponsor, they have defined the role of principal investigator, they have defined the role of institute, they have defined the role of institutional ethics committee. So if it is a clinical trial, you have to have a sponsor. If it is an academic clinical trial, sponsor is not required. So if you have a sponsor, it will not remain academic clinical trial, it will become full-fledged clinical trial. That is the only difference. Outcome, everything, it depends upon your study protocol. It is not dependent upon anything else.
In an observational study, I have to collect samples. It is anywhere part of the manual. Less than become intervention, there was a confusion earlier. Mine is not an intervention.
No, it is not an intervention, sir. Yes. You are taking the data, na? You are collecting the samples, which is a part of routine treatment. You are not intervening with either the treatment or the knowledge or the anything with the patient. Just collection of samples is not intervention. It will not become a clinical trial.
So if you get a funding from a CSR for a study,
government sponsor is different, sir.
That's sponsor. So academic study, you can get funding from intramural,
yes,
or university.
Yes, from the government agency, because government agency is not does not interfere with your results.
Okay.
There
So
are chances. Non-government agency. From a government agency, if you are getting a funded, they are not sponsors actually. They are funding your trial. They are not sponsoring your trial. So understand the difference between funding and sponsorship.
So just to add on to your point, suppose we want to do emicizumab, low dose versus optimized dose versus high dose, and if you are buying from the pharmacy, a pharmaceutical company, and intervening, it is not a pharma-sponsored study. But if you're getting the drug free from them, then it becomes, and their DCJ approval is required. That's what is it.
Yes. Because if you are intervening, as the government is funding our project and we are purchasing it from the pharma industry, that is a different scenario. That is not sponsorship by the pharmaceutical industry. Right?
Suppose I want to do a repurposing of the study,
Yes.
of the drug,
Ji.
and the pharma is ready to give free of cost.
That becomes a clinical trial. That becomes a sponsor, and this pharmaceutical company, because the pharmaceutical company will gain from it. They will file a new patent, they will file a patent for that particular use for that particular drug. Because a government agency, if you are doing it, taking funding from a sponsored agency, that the pharmaceutical company cannot patent it. But if you are taking the help of a company and they are sponsoring your clinical trial, be it monetary sponsoring, be it giving you drugs, whatever way, they are sponsoring your clinical trial, then they have a right to use the results of your trial for further patenting or something. Your results can only be used with your permission by the pharmaceutical industry if it is a non-sponsored clinical trial, sponsored by government agency. But if it is from a government agency, company or some sponsorer or some CRO or something else, they have a full right, because there will be a disclosure agreement. You have to sign it before you are entering into that clinical trial.
So sometimes what happens is that some of the pharmaceutical companies give you a drug for a particular condition which is known, that is a PI, that's given in the PI only, that this is indicated. So they give you as a part of CSR. So they don't give it for like their benefit or something like that. So they take it as a CSR.
CSR is a full form, please.
Corporate social responsibility.
Okay.
So will that also qualify for getting approval?
No, sir. If they are giving you for something else,
no, they are giving for the same indication, but
Yes.
as a part of CSR, nothing else.
they are
just
giving
the drug.
they are giving it as a CSR. Agreed. I am not very sure. I have to go through the regulations again. What happens with this CSR? But as per my understanding, which you are trying to say, they are under corporate something, whatever your responsibility. They are donating they are donating, right? So donation does not come under sponsorship.
CSR gives them some benefits actually. ये एक documentation जिसे बोलते हैं ना झोल है basically. But if the data generated by CSR under the category of CSR can be utilized for, like sir said,
yes,
for any patenting or something, it qualifies for.
Yes, data generation and its use is important.
So not
there it comes.
The category gives them some other benefits.
Yes, CSR they get some tax benefit or something, whatever they want.
And there's one thing I just want to add now. Ma'am asked one question. कि retrospective studies के लिए till 2018, as sir mentioned, we could register with CTRI. But after 2018, they have stopped registering the retrospective studies. Now only the word is prospective before the recruitment of the first participant. So up to they do not even register 2018 के बाद से any retrospective data analysis. Any study which takes up the data which has already been generated, they do not register.
So where to register such studies where we use the retrospective?
They don't.
Ma'am, she is saying retrospective data and prospective data. You have not taken any data from the hospital.
It is like we want to do a prospective study.
Yes.
But for that we need the
retrospective data. For that, ethical caveat will be required, and you can register that study because you have not enrolled any participant even retrospectively. Ma'am, what ma'am is trying to say is, पहले हम क्या करते थे, स्टडी खत्म कर ली, खत्म करने के बाद याद आया यार CTRI तो रजिस्टर ही नहीं कराया हम लोगों ने, तो वो खत्म करने के बाद वो रिट्रोस्पेक्टिव स्टडी थी. बट यू आर ट्राइंग टू से कि आपने रिट्रोस्पेक्टिवली हॉस्पिटल का डाटा यूज करना है पिछले वाला, उसके लिए आपको केवीएट चाहिए और उसके लिए आपको ICMR इसमें रजिस्टर करना पड़ेगा. To first taking the permission of the concerned body.
Approval is needed, but there is no registration required under CTR.
Required, it is not mandatory.
Just taking up the generated data and trying to make some sense out of it as a part of study, but not intervening now henceforth. It doesn't require any registration, but it requires IEC approval.
Ethics committee approval to because IEC does not usually give you approval, they just give you a waiver that this study does not require institutional ethics committee approval. That is a certificate which has to be given by the IEC. So any other questions?
Then it will be like observation study. If you're using retrospective data, suppose
it is always an observational study. It is always an observational study. Yes. Yes. For using the retrospective data, it is all, you are not intervening there. You are just taking the data and, as ma'am has very rightly said, making sense out of making some sense out of that data. So that is retrospective study because you cannot intervene retrospectively. Right? So it is just an observational study. It is not mandatory to register your study, retrospective study into CTR. CTRI is clinical trials, be it academic clinical trial or actual clinical trial. It is a must to register on CTR. Clinical trial registry naam hi hai uska. Yes, any other questions? So I hope I was able to clear some of your doubts and some knowledge enhancement from my side. So thank you so much.
Thank you for very informative and interactive talk. I request you to please introduce our next speaker. Sir, please introduce our next speaker.
So I would like to introduce Dr. Vineet Jason. He is MBBS, MD Medicine, DOM Neurology, 3 degrees. He is positioned as professor in neurology and a lead consultant neuromuscular in Neuromuscular Center of Excellence in the Department of Neurology at CMC Ludhiana. He is India's first and only accredited Duchenne Care Center by the World Duchenne Organization, this center, and he is there in India's Book of Records for largest superbike rally to raise awareness on Duchenne muscular dystrophy in 2023. He has established telemedicine department for CMC Ludhiana in 2020, and he has won a Young Investigator Award at World Stroke Congress in 2024. So I would like to welcome Dr. Vineet Jason for the next one.
Can we all just stand up? So we'll just do a little bit of exercise, okay? So can we all just squeeze our fists tightly, all of us? Lift up your hands.
Let out the stress.
Yeah. So now what we'll do is we'll breathe in also as we go up with our hands and breathe out. Okay? So squeeze tightly. Breathe in. One last time. Squeeze our fist tightly. Lift it up. Throw it down. Jiggle around. Little bit of it. Maybe turn around a little bit more. Move your legs, stand a little bit. Okay? Come, please sit. Can you see this? Okay, so good afternoon. I'll try to go through this. I hope you'll be able to understand as we go. It's very simple.
Sir, front arrow zoom. चलेगा सर.
Okay. So, thank you so much, Dr. Jain, for helping me out with my presentation. So, I'll just a quick outline of what we will go through in the next 20 minutes. 20, I'll try to keep it short. Is study designs. Okay, these are just clinical trial. We won't look at observational. It's all interventional. So, randomization, some basic principles on randomizing. Just an overview. Okay, we won't go too deep into it. Blinding. How do we blind? Okay, what is single blind, double blind, what is triple blind, quadruple blind, and quintuple blind. कुछ नहीं करना. Double blind बहुत अच्छा है. Okay, and then there's of course very important thing called probe. I'll briefly just mention that. And I would expect you to have some key takeaways. Okay, and we have some chits there. We'll call out and we'll ask, what are your key takeaways from this talk? Okay, so it's randomized. Okay, not pre-planned. Okay, so what is study designed for clinical trials? There are simple, large, randomized control trials. Okay, that is one study design. We have small, we have seen with 100 patients, 150, these are small ones. You know, in the hierarchy of evidence, what is on the top? Have you seen that? Niche aata hai, case reports, case series. Very good. Meta-analysis. Okay? But GIGO hota hai na? Garbage in, garbage out. Agar meta-analysis is of those studies which are low quality, and RCTs kitne 150 patients पे 100 पे 70 पे है, तो उसका जो meta-analysis भी कराओगे वो उतना कोई खास नहीं होगा. It won't be as robust. So then you have a role for a single large randomized controlled trial which will settle the issue once and for all. Okay? So there is that thing above meta-analysis, and above that is meta-analysis of large randomized controlled trials. Okay? I hope that makes sense. Alright. What is the next trial design that you can have? A factorial design. I will briefly tell you about this. It's factorial design. When there are two interventions that you want to study, especially when they have an interaction with each other. Maybe it is a synergistic, it may be detrimental. You want to study two different interactions. This is the design you can use. Okay, it's very simple to do. It's also very efficient because in one go you can study two. You know, because why go through the whole process of collecting data again? Then you have cluster randomized trial. Have you heard of this? Yes. Is this, yeah? Some of you have heard of it. So cluster randomization is like 2 unit है department में. You know, एक professor को एक तरीके से इलाज करना है, दूसरे को दूसरे से इलाज करना है. But that's just 2. Cluster randomization, you can even, you know, do villages, whole villages, where there's a chance that the control arm will get exposed to your intervention arm, and so the result will be skewed. So you want to keep it separate. It can be hospitals, it can be floors, it can be days, it can be different weeks. So it can be done in different ways. We'll touch it briefly. Then this is not really a study design, but you would have heard of superiority trials. Have you heard of superiority? जब दो दवाइयाँ हम चेक करते हैं देखने के लिए कि one is better than the other. But आजकल दवाइयाँ इतनी अच्छी आ गई हैं, उससे अच्छा क्या आएगा? तो हमारा स्टडी फेल हो जाएगा. So then you do non-inferiority. कि उससे कम नहीं, उसके बराबर का है. And if there are certain gun which are better convenience-wise, cost-wise, then our drug will be more popular in the market. That's how non-inferiority designs are designed. And then you have adaptive trials. Okay, adaptive trials are trials which are concept in which you can build in a priori certain rules. That if this happens, then we will do this. We will just briefly touch all of them. So when do you do simple, large, randomized control trials? These have to be done when there's a very important question you want to answer. It's very expensive. This is not cheap. Multi-center. You need multiple centers. You need a large sample size in thousands. But it's a significant question you want to answer with a very hard endpoint. It may be mortality. You want to study, does this drug help with mortality? It is not for small, endpoints which are difficult to study, but which are very clear. Also, the effect size, should be modest. It is not something that is very obvious. If it's very obvious, why do you want to do such a large trial? It is a question which is unclear, you're trying to answer. For that, you might want to do a large randomized control trial. And of course, it has a large, it is a question for the nation. India needs to know this. We have to have an answer for our people, for our population. You would then plan that. Okay. So, small randomized control trial, for example, you see here that you have small random. Okay, this point doesn't work on the screen. Okay, so you have around. You can use this mouse. This gives me numbers. Okay. Okay, so small RCTs around 1000 show that the magnesium in acute myocardial infarction had a relative risk reduction of 50%. Okay, p-value is significant, right? That is 0.05. We have statistics, so but it's significant. But then when they did a large study, okay, 58,000 patients. Wow, can we do that in India? I think we should do. There have been studies. There have been studies with 20,000 patients. Create registry at 20,000 patients. So it showed that instead there was excessive mortality with magnesium. Which one will you trust more? I think that's very simple. So we should have as many large, but it's very expensive. So the question should be significant enough to us, okay, and simple enough to do. Otherwise, very difficult across centers. And then here you have in small RCTs. You show that perioperative beta blockers, okay, anesthetic scale was important, actually showed lower mortality versus control. This is 80% relative risk reduction. I'll show you that slide. Whereas here with the large 8,000 patients, it showed excessive mortality. Totally opposite. This was a guideline changing study. As soon as it came out, they took it out from the guidelines. Don't give beta blockers. Okay, so that's the impact that the research that we do can have for what we want to see. When you look at this, what do you see? Here you have treatment A with how many patients are there? 100. Yeah? So the total study is around 200 patients, right? And what do you see here? How many had MI? 1. Here? P-value was? Okay. And here you see second trial, 4,000. And how many patients here? 4,000. Total of 8,000 patients. And how many endpoints here? MI 200 here? P-value again? But look at this. The relative risk reduction here, it looks very impressive, doesn't it? 90% reduction. Okay, whereas here you see it's only 20% reduction with a confidence interval between 5 and 37. Which study would you trust more? But दोनों the p value the same ही है. So yes, okay, sample size and also the events. Look here, you have 1 there and 2, and here the events. If you were to add 1 or 2 more events on this, what would happen? Okay, you have a drastic change. Okay, you add 2 more events to this, you would have the p-value would change to 0.13 from 0.02. Just 2 more events बढ़ा दो. So a significant change. That's what we call a fragile p-value. Whereas in the 8,000 you add 2 more events to that, the p-value will remain the same. So it's robust. So this is the advantage of understanding statistics, why large trials are important, and that's the point I wanted to make, why we should have large randomized controlled trials. Simple eligibility criteria, it should not be complex. Okay, it should be simple consenting process, because we're multi-center, one center can't do this. It will be all of us put together, we can do it. Okay, and then minimal data collection. You don't want huge CRFs, koi nahi bharne wala. It should be very simple so that it can be done, and treatment should be practicable, and measures should be used in routine practice. Alright, coming to the next design, factorial design. This is a 2x2 factorial design. Here you see that there is one treatment A, and then on this arm here you see that there is treatment B. Both of them are being studied in the same population. Okay? इसका sample size कैसे calculate करोगे? जिस भी drug का study के लिए आपको ज़्यादा sample size चाहिए, that becomes your sample size. The lesser one will anyway get studied. Of course, there's a catch to this. If there's a synergistic effect of the 2 drugs, then it gets multiplied. There's a way to do that statistically. But essentially, if you have, and especially if there is a interaction between these 2, this is the only way you can study this. There is no other design by which you can study 2 drugs simultaneously for their synergistic effect. Okay, so it's also a very interesting design we should be aware of. Okay, so advantages are increased efficiency. You don't have to spend money twice. You don't have to put so much of labor at work. Nothing has to be done. One shot, you will get your answer. Okay, the only study which will show interaction. Limitation is that they must have different mechanisms of action. Okay, and they have to be able to be used together. Obviously, only then you can study it. Okay, so there is a limitation to this. Coming on to cluster randomization, I already discussed briefly. This is when you have clustering. So you can imagine it could be a neighborhood, it could be villages, okay, it could be, like I said, wards in a hospital, okay, it could be clinics, OPD practices, where you're clustering those patients who are going there and randomizing that whole set into one cluster, and then you randomize it. This has a statistical analysis which is slightly different, but the essential part is so that you can prevent contamination. You can prevent crosstalk. Okay, that's how you cluster. For example, we had a study called head-host study. In that, what was happening was all the patients, in stroke patients, you wanted to see if their head down is better or 30 degrees up is better. So that the whole hospital was cluster randomized. So it was all head up, that whole hospital. So the nurses had no confusion. Every patient with stroke, head has to be up. Whereas in the next hospital, patients will be head down. And then of course they crossed over later on. That's a different thing. But so that the nurses don't get confused. Because you see uska niche hai, iska upar hai. So there's contamination. So to prevent that, you can do this. Okay? For system level and behavioral change interventions, where contamination is very common, where you can't use a placebo, you would want to do this design. Okay? Coming on to quickly onto adaptive trials, this is our traditional design. What do? You design it, you conduct the study, and then you analyze it. Right? Very simple. Fixed. from the start it's fixed. What is adaptive? It's adapt. You adapt. What do you adapt to? You conduct it, you review it. But this is not, ऐसे रवि मन किया change कर दिया बीच में. No, it is set a priority. When you start it in the design itself, you say, if this happens, then we will do this. Or if this happens, we will do this. Okay? So you review it, and then you adapt it, you conduct it, and then you go on for analysis. It's a very important way. I think I have a slide here. For example, you have, these are different types of adaptive design. Here you can do a sample size reassessment in the middle of the trial. Okay? You said after 6 months you will analyze it. You do a sample size reassessment can be done. You can look at response adaptive randomization based on the response of the patients. You can do that. You can do adaptive enrichment design. You can see which are the patients who seem to be benefiting from that particular thing and add those patients in. So there's a lot of new things in design which will help us to do our get meaningful output better. This is just a crowded slide with all kinds of. You can even have phase 1 and phase 2. Like usually phase 1 trial hota hai, फिर phase 2 hota hai, फिर 3 hota hai. There's a lot of time that goes. But in this adaptive, you can actually do seamless phase 1 to seamless phase 2 going into 3 in that same setting. Okay, once we have information, you can proceed into that. Alright, coming to the second part of my talk here, that's randomization. How many of you felt the way the chairs were reorganized was random? 1, 2, 3, 4 bola, 1, 2, 3, 4 bola. Oh yeah, random. Yeah? It was very simply done. Simple randomization. It was stratified. Okay? So it's a stratified random. Huh? So what was happening before the randomization? All of you were sitting in your groups. And then what did we do? We did 1, 2, 3, 4. So is there a bias? There's no bias. Okay, so it is important to understand that there is a bias, right? In the fact that when you entered into the room, you were sitting in your own groups. There was no random, you just came and picked any chair. ऐसे तो नहीं हुआ ना? अमृतसर वाले, पटियाला वाले सब लोग अपने साथ ही बैठे हुए थे. उनमें फिर 1, 2, 3, 4 करके थोड़ा सा स्प्लिट मारा हमने. But वो 1, 2, 3, 4 ही था. You know, there was no limit, there was a limited number. So even this is not actually random. There is a bias. Okay, so that we have our outcome, which is it's mixed. It's still not mixed. we failed in our goal. But it is still not truly random. What is randomization? Why do we want? We want randomization to eliminate selection bias. There are lots of biases. How many biases are you aware of? Reporting bias. Okay? So many biases. Wow, that's a lot. How many biases do you think are there? 10, 20, unlimited? Okay, that's infinity. How many do you think are there? Koi number bolo. So very interestingly, Oxford has published, okay, if you want, I can give you the reference, it's on the website, there are 250 biases recorded. Okay, 250 biases. Started with just 30 in 1970s. Abhi tak 250 ho. Alright, okay, fine. So you want to eliminate selection bias, right? What does randomization do? Randomization essentially for the two groups, it balances them out. Two known and unknown. Please remember that. Randomization is very, very important. How you do randomization is also important. We won't touch that so much. But randomization is the principle. It is randomized controlled studies. Okay? It is to balance out known and unknown confounding and prognostic variables. Okay? So it forms the basis for statistical tests, for equality of treatment. Okay. Now when we randomize large numbers, all your confounding factors then even out. But if it is a small sample size, there is a tendency for it to get biased. Okay, there will still be a bias. It will be more on one side, more on the other side. The rule is at least 300 or more, even 800 and more is even better. Then you don't need to do this. Simple randomization will work. Both your groups will be balanced. But if it is less than that, then you need to do further techniques where you would do either stratified randomization. You can stratify them by age, you can stratify them by gender, you can stratify them by the disease severity, so that both the arms will have equal distribution. Okay? Don't do more than that, because then it becomes too much and your statistical power reduces. So a randomized experiment is essential for testing efficacy of the treatment. Okay? Efficacy. I'll go a little faster, I know I'm short on time. How do you do random numbers? You do random numbers by, you can use simple statistical textbooks, give you random numbers. Computers, you can use SAS and R. You can use Excel sheet. Have you ever used random on Excel? No? Okay, so Excel will be there. Okay. Some people say it's not truly random, but you can talk to the statistician after this, he will tell you how to make it truly random on Excel sheet. Okay, for smaller studies, because if you don't want to begin very large, it's possible. Okay, it's very easy. And then, why do you want to randomize? You want to make sure that they don't differ in any systematic way, because the conclusion will be totally off if they are different. है ना? ज़्यादा sick patient को आपने अपनी दवाई तो नहीं देंगे, ठीक वालों को दोगे, और फिर कहोगे कि चलती है हमारी दवाई. Okay, but you want to balance that. Okay, and allocation concealment. Okay, maybe we'll just move through and go on to this. So you have simple randomization. If you said simple randomization, block randomization. This is also very interesting, in which you pre-select the block size. Why is this important? Suppose halfway through you're not sure, whether the arms are going to be balanced. You have two treatment arms, A and B. So you will do a multiple of 2. So you'll have a block size of 4. A, B, B. So what happens is, after every 4, the block will start again. So you can be sure at the end of the study, even if it is 60 patients, 40 patients, your both sides will get the treatment. Is that clear? Is that anybody want me to repeat this? So you have two treatment arms, A and B. After you've done, say, 15 patients, it could be, by just doing a toss of coin, for example, that around 9 patients got treatment A and only 6 got treatment B. And then you had to stop it for whatever reason, or that was your sample size. Then it is totally skewed because it was not balanced. So here you do block randomization. You fix the block size, which means after every 4, they will be always A, B, B will be there. How you do that is that I won't go into the details of it, we can talk later, but basically you randomize the blocks. Okay? Okay. And then you have stratified randomization, which I have already mentioned. Okay. Coming to the third part of my talk to you, and that is blinding. Okay? What is blinding? Blinding basically prevents an ascertainment bias. Okay? And it protects against sequence. Okay, just leave it. What is blinding? Blinding is the participant does not know whether they have got the treatment or they have got the placebo, or the participant is blinded. That is a single blind. What is double blinded? The doctor also doesn't know, or whoever was not, that whether they got this. What is triple blinded? The assessor. Now, who is the assessor? Assessor is the one looking at the outcome, which can be at a later date, whether the outcome was there or not. Now, sometimes it's okay in pharmacological trials, you can have placebo and all. Surgical trials में क्या करोगे? Surgery करके टाँके लगा के छोड़ सकते हैं. How do you do it? So there are certain studies where it is very difficult to do this placebo-controlled trials. For that you have a design called the probe design. Probe essentially is, in which it's open but a blinded endpoint study where the assessor is blinded. So even if the patient is aware or the doctor is aware, the assessor who is assessing the outcome can be blinded. So that is also a very helpful tool in contexts where we are not able to do double-blinded studies. It's not easy to do double-blinding. Now, it can. It depends on the design. It depends on what you are trying to test. Okay, so you can do probe is also one design. I'll probably skip this. You can have my slides afterwards, so it doesn't matter. You can go through this. Basically, what are the things that you would do? The participants, if they are blinded, why, how does it benefit? Trial investigators, how does it benefit? Assessors, if they are blinded, how does it help the whole study? Okay. Coming on to the last part, controls. Okay. Not only do you need to plan who you are intervening on, the controls are equally important. That is why it's called randomized controlled trials. I know we call it clinical trials sometimes, but the actual term would be randomized controlled trials. Okay? So you want to get good inferences. It's critical. Your choice of controls will decide your final inference. If your controls are poorly selected, then it will not have good generalizability. It won't be true for the population. Okay? And you have ethics. Okay? Whom would you select as your controls? You want to prevent bias from a recruitment point of view also, so that your recruitment completes early. Endpoints, publicity, scientific credibility of the results, acceptability by regulatory authorities, for good conduct of the trial. Okay? Why do you want good controls? Is to allow discriminatory potential. To be able to look at whether this test drug really made a difference or not. Okay? So, you also want, what are the different ways? Natural progression. You can have controls of past patients. You know some rare diseases. You know that you can't, if you're testing these patients so rare, ethically there's a problem that you're denying that patient the opportunity. So you would want to look at natural history controls. You can look at observer or patient expectations. Okay? Other treatments that the patients may be taking. Then there is concurrent control. Concurrent is simultaneously. The same patient population is being given, who is being given the treatment is getting is also controlled. Same treatment, same time period, similar in all baseline and treatment variables. Okay? And then you have placebo concurrent control, in which you give the patient a placebo. You give them something, but it has no drug in it. You have no treatment concurrent controls. It's the same group, but they are not giving them any treatment. There is dose response concurrent control, in which you are studying variability of doses. Okay? Then you have active or a positive concurrent control, in which it may be another drug. Okay? The patient is being given another drug. And then you have external control, like I mentioned, historical cohorts, historical natural history data, which may be there. So coming back to what did we discuss today, I'm coming to the takeaways. Okay? You have design, randomization, blinding, and control. Okay? So I'll give you just maybe 30 seconds. Think of what are the takeaways that you have from today, and then I'll ask randomly. Okay.
Sir, one takeaway is if you want to get the results, we have to do large studies. and we have to keep it a blinded trial.
Two takeaways. What did you gain from?
Clinical studies and the if biasing is there, blinding is there, and probe studies.
Any questions?
Okay, thank you. Please give me a hand. Yeah, go ahead.
Sir, when you were discussing,
Yeah, go ahead.
yeah, sir, when you were discussing again regarding sample size reassessment, so is it permitted under CTRI trials? So these are adaptive.
So adaptive clinical trials, in adaptive clinical trials, when we go in itself, in the CTRI, when you're going to submit it, you will mention it as part of your protocol. So it is not something that we are changing at whim because अच्छे से हो नहीं रहा, patient आ नहीं रहे हैं तो हम उसको adapt कर रहे हैं.
Can it be like, sir, upscaling and downscaling both?
Yes. So if you're, that is why you have the, there's a whole DSMB, everybody is there who looks at your studies. And so adaptive clinical trials is a whole talk in itself. But you can actually even stop the study for futility or even for efficacy. And that's also fit a priori. When you start in, you have said that we will do it at 1 year, at 6 months, we will look at it again, and then we will decide which way we are going to go. But that decision is also fixed before we start the trial.
So again, for like non-inferiority trials, when we are for non-inferiority trials, when we are checking against a gold standard. So aren't we depriving the patient of the gold standard and using the other drug, like only on the basis of phase 1 and phase 2 when we are looking for the phase 3 trials?
Well, I mean, both are going to get treatment. One is getting a treatment, the other is getting the experimental therapy, which you believe is good. That is why you are studying.
We know that the gold standard works, so we are depriving the patient of the gold standard.
But there is a benefit in this, so I do not do too much because I was in a rush, but in non-inferiority, essentially, you know, both are almost the same, efficacy-wise. It's not the efficacy you're trying to prove, otherwise you go for superiority. Here you are saying efficacy is the same, but there are other benefits. It may be cost, it may be convenient dosing, one may be intravenous infusion, the other may be a tablet. You know, so those benefits stand to help society at large. So for that you are doing this study. So it's not because you feel it is inferior. If it's inferior, you should probably not do it. But you know that it will be at least non-inferior.
Okay, so I know that lunch is coming in. This I want it to be a little more interactive and more questioning types. So you know the word impartiality, right? Why do we use the word impartiality? There is an inherent nature in human being to be partial, either to themselves or to somebody else. And that is when you look at the entire trial, the biases, the randomization, the regulatory authorities, everybody is doing that because we are inherently doing things to your own advantage rather than being impartial or neutral. That's the human fallenness is there. So now I want you to, from this session, I want you to wear the hat of a Sherlock Holmes. You are going to look at the journal from a point of view, they are wrong completely unless it is proved otherwise. That is the whole concept of null hypothesis, right? Falsification. So unless you deny or disprove the falsification of the null hypothesis, scientifically we don't take it as truth. So with all the armed with all the knowledge which we have, the phases of the studies and the types of clinical trials, we'll come into a few things. And this is where we are now, looking at the trials. Now I had sent the 4 trials across to you. Now I would have wished all of you sitting in the same group, in a mixed group, but the whole intent, the intent of the intent was that you would go back and come back with a proposal. So after the initial discussion now, I would request you to form all groups, and you will have to have your presentation and all ready from that point of view. Okay, so how do we go about it? Why critical evaluation matters is because all studies are not reliable, and you will be astonished by the number of studies which are not reliable. And it will help us to understand it better, and it is important for the evidence-based medicine and poorly designed. Look at this study. Why most, this is PLOS publication, okay? Why most published research findings are false, and they look at that 80 to 99% probably are not true. And that is what he said about the playing with the numbers. You can actually make a p-value. Now, unfortunately, all or most 95% of the doctors are driven by this P magic. Okay, do not be fooled by the P magic. Look at various other aspects also, the effect size and the confidence intervals as well. So that is where the whole process is coming to, and I will show you that. And please understand, somebody has put in $1.5 billion or 2.6 billion, he mentioned, $2.6 billion for a clinical trial, and he would try tooth and nail to ensure that this is the go-to drug, right? And that is where we have to come in and find out and say what are the possible reasons, and that requires certain amount of statistical understanding, not necessarily having to do the statistics yourself. Okay, so I would, this is my modification on some understanding and of course some searches on 7-step evaluation framework, and we insist this for any journal club in our department. We do this, and I will go to the details of each one of them. Now, we will hear the name PICOTS, and you would be doing PICOTS regularly. Any study, any thesis study, anything, start with the PICOTS, then you are unlikely to go wrong. Okay? Then you look at the internal validity, what is the statistical and clinical significance. External validity is very easy to look at to start with. And then this is the last bit I want you to look at. So what? In the end, ask the question, so what? You have done a fantastic study, you got so busy with the methodology and the study and how it is done, and you are failing to ask this question, so what? And because we have a paper which is published in any JM, it says to cure something you have to give 3 crores worth of treatment. I would say, fantastic, good, keep it there, we can't use it, right? So what? So what should it be changed in my practice is the question we are going to ask the question. I want you to start thinking in that lines. You open a journal, or the journal club, first look at what is this journal, what is the impact factor, where all has it been done, who is the sponsor, who has written this journal article, and who's the funder. So for example, this is your paper which you have got today, and I have randomly, you would get that one of those journals in your study group. NEGM has an impact factor of 78.5, and when you present your journal clubs, I want you to go through this framework. It is easy for you to read it, and it is already sent it in your emails. So that Google Doc you can share with your teammates and use that same framework to understand the journal. Once you understand the journal, you choose a similar research topic for your group. So that way you have reiteration of the journal, how did they do, and you're making a study in that line. Okay, so this journal is funded by Bill and Melinda Gates Foundation and WHO, so the funding is from outside, so reasonably good. No pharma company is going to say kangaroo mother care is better than no care, right? Okay, Picots is a design, and in the design, so D Picots is what I call D Picots, and there are some more mnemonics we use in our journal clubs there. D is a design. First, find out what is the design. Dr. Vineet has taken you through multiple things. Observational, descriptive. Nobody is analyzing one to the other. Percentages are said, numbers are there, so many percentages were found. This was the median. But we are not comparing between them and finding associations. If that comes, it becomes observational analytical, or it is an interventional, and all of those intervention. So understand this. First question you are asking is it observation, is it interventional? If it is observational, if it is descriptive, or is it analytical? Most of the thesis is descriptive, and then you end up doing some analysis in the end to find out the R or R square correlation or something like that. Okay, some ecological studies. So here, under the observational study, you can do many studies, qualitative studies. So if you were to look into this and make a assumption, okay, intent was not there before, an MRU lecture was not happened in Faridkot so far. Number of trials which happened there in the past, last 10 years. 5 years down the line, number of trials happened. And you are saying that before this, only 10 trials happened in 5 years before this. After this workshop, in the next 5 years, 20 trials happened. Which would be the study you would take? Which of these studies would you be doing? No. So which is the study there in this group? What would be that study called? Is my question. Okay, cross-section list, one time. Today. So you can call it, so you need to use the correct word. So you need to now start thinking, before I did dieting, what was my weight? I did dieting for 1 month. What is my weight? So depending upon the study, some of them may be an overlap, and now you have to choose the most likely. So here I would say it is an interrupted time series study. Would be before I did one intervention, if we did an intervention in a workshop, we sat together. This helped us making relationship with Dr. Anjima and Anjima, right? Anuji, okay. So I'm trying to get Ruby, Anuji, Dr. Vikas is here. Vikas is here. Sorry. Okay, so I'm trying to get my names right. Okay, so after that relationship built, we thought about something and did a collaborative study, and therefore it is an interventional, an interrupted time series and seeing what happened. Quality effort which you do in the institution, it would be an interrupted time series study. Ecological study is again exposure. I mean, it is a prevalent study before and after. And like that you can have many. And case reports and case series are the most sought after because it's easy to do. But this should give rise to some of these as well. And cross-sectional study is most of the thesis generally is cross-sectional studies there. So this is some of the studies to show you various types of studies there and exposure this thing. Now in terms of direction wise, this is again a confusion in most people. What is a case control study? Can somebody tell me what is a case control study? Correct. So suppose this is the timeline. Okay. I want to study. I'm going to look there or I'm going to look here. I'm going to look here. I'm going to look at what? Okay, I have some pain in my leg. What was the potential cause? I am looking at, I want to look at how many people who came for this, attended the class for 1 year, how many of them got headaches? It's a retrospective. And sometimes we can stand here. Suppose I stand here and look there. Which cohort? So then what is looking there? That is prospective. So what is this and looking there? Huh? No. That is again a retrospective cohort. From etiology to cause. Etiology to outcome. That is outcome to etiology. So that was case control. So now at this point I want to know all the patients of MI, how many of them had hypertension? I'm not asking the question how many of the patients of hypertension developed MI is my question. So I take all the charts of MI, hypertension, and come here and say how many of them did MI. Or you will do an ambispective study, retrospective cohort and a prospective cohort. This can become a bit of a confusion at all levels. So it's important to get this clarification done. So that's some of the pictures, but understand where you are standing and where you are looking, from where you are looking, which direction you are looking. It's easy to understand that. Okay? So prospective, retrospective cohort. If you stood there and looked at the exposure, it was a case control. Okay, cross-sectional analytical study. Cross-section is today in this 1 month I am going to look at X kind of things. Some of the studies you can look at. You look at Google search it and see if you want to study some of the studies which you can come up with. So I will. Time series is something which is interrupted time series. Interrupted time series also give you a regression. Otherwise it is just a time series study. I will show you some examples of that. This is interventional study. RCT, factorial, we went to that. I wouldn't go to that. So also I will parallel trial design. Most of the trials are parallel group. He talked about factorial design that helps you to reduce the number of patients in the group. 2 drugs can be given together. One group will have no drug, one of them A, B or both. Adaptive he showed you. Adaptive is essentially keep the winning horses together. So you have a fleet of horses who are running together and some of them are winning and you drop the losers, go with the winners so that the good outcomes are more enriched. Okay. Pragmatics. Pragmatic study is essentially practical studies. The word pragmatism is being practical and not really because there is a inherent bias of choosing only the good fellows into the trial so that the results will look good. Non-randomized studies. So you can have an analytical interventional study without randomization. I have only one group of patients. I am giving isavuconazole or any drug to see if the infection rate is reducing or not. And I will compare it with the population mean or a population percentage proportion to look at if there is any change happened or not. Interrupted time series study, how it works. Examples in healthcare I will tell you, I will not talk. So, we had a problem of people not attending 8 hours of work in the patient. Everybody has a problem. So, everybody will come late, go early, nobody is fitting in time. Unless you put it on a attendance and the salary cut, it won't happen. So what we did, we sent an attendance marked red, green and orange to everybody. Red means you came early or late but went before fitting in 8 hours. Orange is you came early, late but you stayed along, finished the 8 hours and went. Green is came on time, went on time. Or late, no problem. So this is the red before the intervention. July is the intervention. The moment intervention happened, this is the intervention time and red started coming down, green started going up. time series only, not time series intervention there, and it is attendance compliance summary is this. Magnitude size, the feedback loops have a high impact, low cost intervention. That means we're just giving them, we're not doing anything, we are sending it in their email and WhatsApp saying this is your attendance. Up, response बहुत हो गया कि हमारा number आएगा. At least they are aware that it is done. Okay, design and PICOTS. Now we've finished the design part of it. PICOTS is extremely important part. What is the population we are dealing with? What is the intervention we are doing? And this should be your core for any study. If this is clear, the question is more important than the answer. Take all efforts to refine the question in such a way that there is no ambiguity in it. Okay, so I will give you so example. This ruxolitinib is a drug which you use in steroid refractory GVHD compared with the best supportive care for 24 weeks. Now, for example, what is the population? Population is steroid refractory GVHD. Intervention is ruxolitinib. Comparison is the best supportive care. Time horizon is in the next 24 weeks. Now this is clear. If you do not have, this is a problem. So you can say what of it. So this is the action plan. After the minutes you take the action plan and what is the plan. After the meeting, Dr. Faridkot will go back and write a research proposal in hemophilia, low cost intervention, low dose or optimal dose by the next 3 months. That is a time attached to it, a person is attached to it who will do 5W1HO. Now, from the PICOTS and also try and see what is the hypothesis you are looking for. Because you have something in mind, this may work, and you have a hypothesis. Based on the hypothesis, you make the question, or you ask the question and you make the hypothesis. Both ways it will work. And then it is that's a null hypothesis issue, and falsification of the null hypothesis then becomes the bedrock of all researchers. Internal validity. Internal validity is essentially the coherence and the consistency in which your paper is evolved. Okay? Now, there is an internal validity is looked at. We in our department we use the word spread. What is the selection bias? Is there a selection bias? In every observation study there is a selection bias. You cannot get away with it. If you want to get away with the selection bias, what is the best case scenario you can do in an observation study or a retrospective study? Retrospective study. There is something called propensity scoring. You have to do the grouping. So there is something called propensity scoring or multivariate regression you can do subsequently. So understand some of these terminologies, not that you would do that, but at least you can coordinate with the statistician to say that have you done this, have you done that. Matching. Propensity score matching. Okay. Then comes the performance bias, that is essentially what he mentioned about what is the patient's outcome. Is it blinded or not blinded? If it is not blinded, you come into the performance bias. Reporting bias can happen at various levels. One is the person who's analyzing can report it as better, or if it is not a positive study, you don't publish it. So then it comes into the publication bias as well. Attrition biases, you started with 100 patients, everything was done and sample size, everything calculated for 100, but the end of it is only 50 patients. Then comes a detection bias there are various types of biases you don't really, okay. Randomization, I wouldn't go into the details. Again, the allocation concealment is something which we will not, sometimes it's a problem. So one of the best way to understand it in my mind is, if you have 10 people, I want to randomize them into 5 each. I'll say first person come to group 1, second goes to group 2, third to group 1, fourth to group 4. Is it a good randomization? Why? Why is that? It's randomized. asking one person to go to group B because I know he is good. Where is the problem? Okay, why is there a selection bias? Correct. The second person does not have an equal opportunity to go to either of the groups. Therefore, the randomization will be A, B, 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 C, like that. We don't know. That is randomized. So various ways. So if you have to understand that concept, it is fine. I hope everybody understood the core principle of randomization. Every subject should have equal opportunity to go to either of the groups. Blinding we talked about, and confounders are something which you have not anticipated but it came out. In a randomized study with at least 100 sample size in each group, confounders are generally balanced out. But he said stratification if you have to do, you need 800 plus. Okay? So in a retrospective study, you cannot adjust for them. That is why the propensity score matching or multivariate regression of the confounders you thought may be influencing, you are taken into consideration, not the other factors. Okay, so that is the known knowns only we will take it, right? Okay, so now comes how do you assess the bias? There are certain tools available. This is something which you can look up and understand whether there are tools available. ROB2, Robbins, Javad scale, Newcastle, Ottawa scale, based upon which study you are doing. This is not a session to tell you how to do it, but giving a broad brushstrokes on these are available to look at the biases. Next comes the statistical significance, statistical and clinical significance. Very important as clinicians, not to be befooled by the p-values. You have to first say what is a meaningful clinical benefits. I can make p-value significant for a 1% significance, 1% difference between the groups by just increasing the number. Okay, so that can sometimes be a problem. If I want to prove my drug is better, the company wants to prove this drug is better, and I will make the p-value very significant by just increasing the n. Whereas that 1% difference is not too great to me. So if I ask a question, in multiple myeloma we have a drug called daratumumab. It costs 25 lakhs per year. First year, second year onwards 15 lakhs. Clinically significant, if you go to a conference, all over a conference, your industry will not sponsor you if you cannot talk a session on that. Okay? I'm not saying it's a bad drug, but how am I translating this to my patient? I will say there is a 15 to 20% advantage in terms of CR. Normally you will get 60% gets better. If I do this drug, there will be it may be a 15% better. Are you ready to pay this 25 lakhs for that 15%? That's the point which is coming to. We don't talk that language. Unless you know this is the clinical benefit. Now if the clinical benefit of 50, I would have definitely asked all the patients to do it. Here I might say 15% benefit ke liye 25 lakhs he has to sell his house and take mortgage may not be a good idea, and therefore I must tell that in so many words for him to understand that. So that is why in our setup this percentage and the benefits have to be linked to the cost. If insurance is paying or the government is paying, it's a different matter. So what are the things you need to look up? One, if the outcome is matching the test they had said to be. Sometimes they will say these are my primary aims and objectives, but the entire discussion is not secondary aims, which is not powered for the clinical benefit. So the sample size was calculated for the primary objective only. That is how the sample size is calculated. Secondary objectives are 10 or 5. Primary objective was not significant. OS was not significant. Overall survival was not significant. Now the entire discussion on the paper is on the secondary objective, and either they will refuse to talk about it, or they will one line frame it in such a way that it is slipping your mind. So unless you are conscious what was the Picot's, what am I looking for, you will totally be fooled by the p-values of the secondary objective, which is a post hoc analysis essentially. Okay. Assumptions of normality, independence are met. Now these are, sometimes it can, go over the head. But these we will discuss. Himanshu will talk about the confidence interval, the effect size, the hazard ratios, the odds ratios, the relative risk, etc. And the confidence interval and the number needed to treat. If you are looking for those, relative risk can be 1 becoming 2 is 100% or 50% more, right? But absolute difference is very less. So look at the absolute difference, look at the number needed to treat, which will mostly get you to what you want. Missing data, important to look at the methodology, what they are written. Now, this I talked about the absolute introduction, number needed to treat. MCPS is the meaningful clinical benefit scale in the ESMO guidelines for cancer trials especially. But there are other trials you can look at what is a clinical benefit. But I generally ask the question when residents probably present the journal club, in your minds, how much is good enough between these two? Is it 5%, 10%, 20%? And if you realize that everybody has a different threshold, it's like what car do you want to buy? Everybody has given the same amount of money, but how do you choose? Everybody has a different gauge in their mind. Percentage of difference, what you find is significant, is an average. By and large, you have to say it is somewhere between 10 to 15% has to be there an advantage, unless it is a life-threatening situation. Whether I should wear parachute or not parachute, it is a matter of lot of difference, right? It is not just a small difference there. Okay. So, hard endpoints versus surrogate endpoints, I want you to make a conceptual understanding. What do you mean by hard endpoint in any study?
Permanent disability, death.
General death. So, overall survival becomes the, so I'm starting a drug here, right? I want to reach there, that is the overall survival. Most studies are not giving the overall survival. So what do? You do a surrogate, progression-free survival. Cancer has not progressed or died. So not progressed is good enough. If you progress, you are assuming that you will die. So PFS becomes. But if the time from this to that space is very large, then I will say, I want a surrogate end marker. Surrogate end marker is I want to know if it is a complete hematological condition. Blood is okay, bone marrow I have not done. So I will come up to here and say this is a surrogate end marker, this is okay. I want to make it a little earlier. I'll say cytogenetic reduction, cytogenetic response. I want to make major molecular response. Now, this and that has become quite a bit. Surrogate endpoints can be taken up as, suppose I'll say, success in life. Surrogate end marker is, have you had 80% in the first standard? Is that a good thing? 80% in your grade. 80%. Now, A grade in first standard exam is a surrogate marker for success in life. Okay, I will say 5th standard. Slightly better, still not good. Okay, now I say the degree he has passed, the post-graduation he has taken. Not good enough, but still better. But there are other components which will make it. So therefore understand that the surrogate end markers can be, endpoints can be varied and you can be befooled. Now you can get a publication done, first standard 80% difference ho gaya. So what mein aa gaya na, wo khatam ho gaya. So what mein kare? Paper was fantastic. They went through all the rigor of scientific literature, but so what mein haar gaya na. Okay, so interpretation of survival curves is important in this situation. So hazard ratio. We'll come to that on a case-to-case basis on your study. I would not go into the details of that. Also look at the adverse events rate. Most often, adverse events जब आप देखोगे, there is no p-value there. And to make the interventional drug look good, they will combine all the bad things of the other fellow together, and whichever he is slightly better off is highlighted. Again, it is only your discernment which will help you to understand if the side effects are more or not. Otherwise, we are only talking about net benefit is not mentioned. Outcome is good, fantastic, he lived long, but with peripheral neuropathy, grade 4, not good enough. Okay. So now comes external validity. Now when you are looking at the inclusion-exclusion criteria, you are looking at the storyline of, is your race and ethnicity is represented there? If your race and ethnicity is not represented there, it is all Caucasians. Fantastic study. I will take it with a pinch of salt. Now the whole question of pinch or fistful of salt is the only question. There is no guideline for that. Whether how much will you say, I will not take it into consideration. That's your discernment there itself. This will tell you, the inclusion-exclusion criteria will tell you, is it possible? If they said I ruled out all creatinine more than 1.5 from that group, anybody with some side effect or some symptom excess I remove. That means it is not real world practical. No? I am seeing patients with more than creatinine myeloma. If I say remove all the patients less than 1.1 creatinine from the study, fantastic outcome came out. But for me it is not applicable. So what may wo nikal jayega. Evidence to decision is the next thing. This is something which is coming up very well but not talked about much. You have the evidence, fantastic evidence. It is highest level of evidence. How am I going to decide? That is what the bottom line is. Now for that, there are various tools. There is a grade and grade pro available, where inconsistency of the results, inconsistency, indirectness. So the result mentioned is not what you wanted, or the confidence interval is large, or it is imprecision comes there, and the publication bias. Now based on this, you can grade a study. In fact, Journal Club should end with this note. Now with AI available, you should be able to do it more or less and use your discernment whether it is right, going back and checking it. We use this mnemonic for our grading of the news, this thing. Resource use, that is, what is the equity, is it useful in our setting? What is the acceptability, patient preference? So basically, is it patient may prefer injection subcutaneous rather than IV, something like that, and is it feasible for use? And these are the setup of, when you see the guidelines, they will say grade, moderate evidence, moderate certainty, etc. Now comes the last part of it, the cost effectiveness, cost utility, or the pharmacoeconomics of it. That is something which we have to look at how it is good or not. And then ensure that the paper has some limitations and conclusions which is vividly mentioned. And it is extremely important to say the limitations about the sample size, a short follow-up, or a surrogate endpoints, as I mentioned, and poor generalizability, or the missing data, or uncontrolled confounders. Uncontrolled confounders can be a big problem. And again, when you see the title of the paper, you think in your mind, what do I want to know from this paper? Then you read the abstract, conclusion, and then go up. That's my strategy. Go look at the abstract, title, think, look at the conclusion. After the conclusion, go to the methodology and say, have they used the framework to reach that conclusion, if there is a correlation between these two. Okay, so this is, we put together with this 15 things. D, Faridkot, then CRPS, and MCBS is the recent addition to our discussion. Basically, D, Faridkot and CRPS. Checklist, any publication, you will come to that on the checklist has to be used on the guidelines. And then whether they have used a risk of bias, pharmacoeconomics, and so what. And the other thing is basically we start with this, what is my, your clinical benefit which you will say about. Okay. So do not be impressed by statistics, be convinced by the methodology. So the numbers may say whatever they want, numbers are important, extremely important, but look at the rigor of the scientific rigor there. Now, you have four, where are the, so can we stand up and go back to your own groups? Unfortunately, I would have wanted you to be here, and we will give you the introduction. During lunchtime, post lunchtime, we have time for you to discuss more. So go back to your, can you go back to your groups now? I want original institutional group, and we'll give you the lots to pick up on your research, I mean your journey. Okay, so I will start with this group. Can you give him the lots. Give. Pick up one. Blind. No, after that. You have to do. Don't then blame me after that. Okay, so that is your journal. And that सबको दे दो. एक लेना आपको. ना बस. बोलो उसमें क्या है? Okay, no. Okay, so you have the journals with you, right? We have some prints also. Can you, yes, why don't you read out? Okay, can you read out your journal? What is your journal? NEJM, that kangaroo mother care. Okay, NEJM journal, can you give that NEJM paper there? This one, that's a kangaroo trial, KMC trial, IKMC trial, kangaroo mother care trial. Okay, yours is carbitocin. Yeah. Okay, give this paper there. I hope you had a chance to read it. If you didn't have a chance to read it. And please access the Google Doc. Can one of you open the Google Doc which we had sent you? Right away, open it and save as your group name. Because that but somebody has to open up the laptop. Laptop open, go to the Google Doc. Okay, so I need to go to the Google Doc. Where is this laptop?
Can you please share it once again?
Sorry?
Can you please share it once again?
Yeah, so I will ask Richard to Richu. Can you call Richu? Okay. So where do I send it to? Richa has the group. Okay. So let me send it to the groups. WhatsApp groups I will send it to. Google Doc Manager. So I've shared it in the WhatsApp group. One is a journal critical review and the other is a draft research protocol. You may share it, then you have to now save as, so I need to show you how to do that. Take this. HDMI, you have to connect the HDMI.
I have to take it to
So what is this HDMI
वो आपको HDMI चेंज कर दो ना ग्रुप में डाला है मैसेज पे रिप्लाई नहीं है दोबारा डाल दो Your net is Google Doc is web. Have you been able to access the Google Doc? अपना चलाओ. Okay. अपना Google Doc चल किया. Okay. So share that and then you can start analyzing this. Your student who shared the doc with everybody. No, no. So we have to save as
Two of you will be presenting the journal club, and then use the same design and find a research question. Now for that, another two people can work on, but by discussing on the journal club, you will understand the design of the study and the pickots you want to make in your study. And those people will be presenting on the last day. And there are external judges who will give you some inputs on that. So that becomes a practical thing for you to do. You need to draft a proposal, at least go to my pickots, methodology and budgeting. Put somebody else on the job. So there is a framework for budgeting also given. So put your imaginary numbers, you can put it, no problem. Imagine you have all the resources in the world.
Excuse me, sir. Oh, sorry.
Okay, another 5 minutes, you assign responsibilities, who's going to do what, number one. And then once you assign the responsibilities, you have your lunch. During lunchtime, please sit together and discuss how you want to go about it. Okay. Okay, so are you through with the two activities? One is journal club presentation. This one, that is tomorrow afternoon. Day after tomorrow morning is your research proposal presentation. Same group, this group. Because at the end of it, they wanted you to go back and work together as a team. You need to mingle with them, that's a different matter, so that you can do collaborative studies. But the essence is to come back to the proposal. That is up to you. That doesn't matter. You decide what you want. This may not even translate to the final proposal. Okay? Doesn't matter. But at least you would have gone through the exercise. Yes. So the people who are working on the journal club will critically analyze it.
is ready, everybody. You can have lunch.
Through with your discussion, you can both have your lunch. So we can, next session was 2:15, right? So we'll have to catch up on the lunch break.
We will have lunch in 15 minutes and come back.
So ये तो हो गया अपने
मेरे बैग चल रही है ना नहीं दूसरा बैग इतने पे
हम लोग बर्थ रेट ले रहे हैं ना
It was stratified according to site and birth, date or division. The stratification was on site.
स्टेप बाय स्टेप पहले कैमरा सेटअप होता है। कैमरा सेटअप और यहाँ से ऑन ये जो है मोबाइल से ऑपरेट होता है। जब आप ऐप से ऑन करेंगे लाइव तो जब आप कैमरा कनेक्ट करोगे इससे और आप ऐप से ऑन कर सकते हो, नहीं तो आप ऑफ कर सकते हो। और ऑन करने के जब आपका इवेंट स्टार्ट होगा तो ऐप से ऑन करके तो आपका लाइव स्ट्रीमिंग होगा। तो कल हम उसी चैनल पे
for both primary and secondary outcomes. इसमें serial है, हमने statistical method कौन सा use किया? ITB method use किया। Missing data तो both the primary and secondary outcomes। मतलब 0 कर देते हैं। Overall risk of bias judgment।
Overall risk of bias. डबल ब्लाइंड नहीं है तो इसको
काफी सारे इटरेशन आगे काफी
साल के 2 साल के बीच की
ओवरऑल जजमेंट यही हुई कि उसमें काफी सारे
टेबल सही है ये तो पता नहीं देखो 72 बार उसके बाद EX ESC X C space rest is like PR 1.0
सेकेंडरी आउटकम के लिए वैक्सीन
आपको प्रेजेंट करने के लिए बोल देंगे ना हमारे पास CI तो कुछ उन्होंने मांगा ही नहीं 95% कॉन्फिडेंस है बट ऊपर लिखा हुआ से ऊपर ये आना था रिस्क रेशियो भी है बाजार
मतलब जो 28 days वाली
Okay.
Okay, brief attention from everybody. So, we will go through the next session briefly. One, we have given you each one journal article. Let me make it very clear because there's some confusion there. So, that journal article you were supposed to have critically analyzing it.
So whatever you are saying, it is getting...
Okay. So that is supposed to be critically analyzed on the framework which we have given on the Google Doc. Then one of you will have to convert it into a slide, and that will be presented tomorrow afternoon. There will be judges from outside who will come and do that. So that is exercise number 1. Exercise number 2 is similar to that design of the study. Formulate a research question of your choice. That means you are going to do your own research question.
That will be an individual activity or a group activity?
Group activity. Because
Same article that we have given or
no,
something?
similar design.
Something else that we
Something else.
Something else we'll think of.
Yes. So that is when the whole process of ideation comes in play. We will do that exercise now. Ideation. Okay? Ideation. How do you stimulate a thought process and make it asking well? So, okay, they already have a research question. What about you? Have you made your research question?
We were focused on doing the activity that we had given.
Which is the journal club. Okay, so let us put a pause to that right now. What about your team, sir? On the journal club. Okay, so at least you had the picot of the journal club is clear. You have done the design. Now you will get armed with the statistical understanding also tomorrow also initially. But tonight is the time you will go and critically analyze it if you have to. Let's go through an exercise called ideation. How many of you have heard the word mind mapping? No. Okay, so I will request Dr. Vikas. You have a whiteboard there. You take one of these. Okay, so you can give me one pen here. You can use that pen there. Mind mapping is essentially building on an idea which you have, asking other questions alongside. Okay, let's start afresh. What is in your mind, just like that? What is your try? What is the article on? What is the title? Mixed trial or what? What is the population?
The population is the 18 to 30 female population, 30 years of population.
What are they looking at? Intervention care.
The intervention, they are looking at the care of the mother in preconception and prenatal and postnatal area with the addition of the micronutrients.
Micronutrients. That is the intervention of micronutrients in the mother. What is the comparator?
So it is one with these micronutrients, other with the usual care.
Okay. What is the outcome?
The outcome is if the neurodevelopment.
Neurodevelopment, is it better or worse? Okay. What is the time frame they are looking at? How many months post 2 years?
Up to 2 years.
2 years. Okay. So your specialty is general surgery. Okay, wait, I need to get your name once more, please. Sit, please. Name. Sunny Chopra. And department? Biochemistry. Okay. Microbiology, biochemistry, general medicine. Animal medicine. Pediatrics. Can you think of a study which can include all of you? Or a topic which can include all of you? No, not same article. That is just to tell you that is an article. Antimicrobial resistance. Okay, what biochemistry component do you want to bring in there? Some biomarker. Okay. Can that be in surgery and medicine patients? Pediatric surgery. Okay, so what is the core thought process in your mind you want to work on? Antimicrobial resistance? So write AMR on the top, in the center. Center. AMR, okay. Try this. Okay, so all of you can keep helping them out, okay? This is mind mapping, ideation going on. So AMR, just put a circle. Small you can write also, no problem, circle. Now, what in AMR you want to check? What in AMR? In what kind of patients? Sit, please. Okay, general medicine and surgery. Okay, so you put an arrow here. General surgery, pediatric surgery, medicine. Okay. 3 ho gaya? Or now you build on it. Now build something on AMR in general surgery, pediatric surgery, medicine. What is the core issue in AMR? What is the reason? You want to do something in preventing antibiotic resistance. That's your impact. Now you will say, to prevent antimicrobial resistance, what should I do? That's the whole question. Now, this keeps developing as you discuss in the team. So one of you will look up PubMed, open up PubMed antimicrobial resistance. Look at other studies which are done in the lines of antimicrobial resistance. Right? So once you learn up antimicrobial resistance, you will find 2-3 articles on that. So then you will say, okay, I want to see, I will do a blood culture, I will send it. What is the fastest way in which I can identify a culture? What is the turnaround time for a culture? It's about 7 days. Early bleed can come in, but it takes another 2 days for the culture sensitivity to be ready. What are the ways in which you can early detection of the bacteria? Like bacteria. So that will alert you, but identification will be still another 2 days. No. What can you reduce? What can you use that? that alerted you, bleep it. Agreed. But after that, then how do you know it is E. coli or MRSA? It will take another 2 days. What are the earliest you can do that? Biofile. So write the word biofile. Write the word blood culture. You write whatever is being discussed, you write. It's just ideation. Small you can write. So blood culture, write MALDI-TOF, write whatever. You please write everybody. The whiteboard is for everybody to do. So you use a Google Doc to make as a thinking board. Okay? Hey, 1 second. Let's just see if it is a, I think this is a permanent marker. So go home. So, well, I also didn't look at it. So the one was not working. Okay, so essentially you can do it on a piece of paper. There are think boards available online. There are Google Docs, Zoom has this thing, whiteboards, whatever you want to call. Now you are sitting around a table, so it is easy. Suppose you are in different parts of the world, or parts of the country, different departments, still you can come to one place and start writing the small activities there. So that is your ideation board. Now you convert that into a research question. What is the smartest way to do that? Some of your ideas you put in ChatGPT and say, can you formulate a research question for me? Give me options. It will give you another 20 options. Then now refine it, come to one study, and now you take the study and say, I want to do a mixed method study, whatever the multicultural study based upon this kind of an activity. In this, can I do? So now you spend a lot of time fine-tuning your question. Spend at least half an hour to 40 minutes fine-tuning your question. After that, it is easier for you to define it better. So you got an idea. We just plowed the soil now with your journal question with you. Now we are sowing some seeds there. By day after tomorrow, it should sprout. Okay? So I will come to the research question. So for example, you can draft this research question. I'm just making it up. Either it can be a interventional study or a descriptive study. Okay, observational study should be the incidence of antimicrobial resistance among surgical patients. ICU or whatever, that is very straightforward. Second is escalation or de-escalation of antibiotics based upon your biofile. Now because that's the intervention coming in saying biofile versus blood culture, I can de-escalate faster, or I will do a non-escalation based on that. Otherwise, culture positivity means I have to start on something anyway, I will start hybrid antibiotic. And then you can say before the sepsis flow, biofire, how many of the patients were escalated unnecessarily and how many we could non-escalate because of this. So that's one way of comparing pre and post. Basically you have to sell the idea that we can be more data driven if you have biofile. That's one way of drafting the question. So modify that question a little better. What is your question? What is your research question?
Effective lifestyle intervention in school children related to NAFLD. Effect of lifestyle intervention on NAFLD children in schools.
In effect?
Schools.
What children?
NAFLD.
NAFLD, okay.
Non-alcoholic
Fatty liver in children.
In children, school-going children. Cluster randomized trial.
So your observation study, yes? Or how long?
For 1 year. Intervention है.
What are you intervening?
फैमिली बेस्ड इंटरवेंशंस देंगे उनको लाइफस्टाइल मॉडिफिकेशन इंटरवेंशंस देंगे और 2 आर्म्स में बनाएंगे इंटरवेंशन आर्म होगा एंड कंट्रोल आर्म होगा
And outcome, what are you looking at?
आउटकम की जिसको मैंने इंटरवेंशन आर्म में रखा है इंटरवेंशन दिए फैमिली लेवल पर स्कूल लेवल पर उसके जो आउटकम्स होंगे लाइक ALT-FD रेशियो है रेशियो है
After how many months?
1 year.
Okay, 1 year later. So basically before and after the intervention, 1 year later, what is happening to the ALT-ALT ratio?
And monthly visit.
What is the impact of it? So what?
To reduce the non-alcoholic fatty liver disease in children.
And there are studies to show that this is leading to cirrhosis in younger age.
Yes.
Okay.
As per them, 8 out of 100 children are facing this problem in urban and suburban areas.
Okay, good. So you got their study. Understand each other's study also will help you this thing. There is a population of children. In them, there is an existing problem of non-alcoholic fatty liver and hepatitis, which can lead to long-term complication, and therefore they want to give lifestyle modification in the children. 1 year later, they need to check whether they have improved. So almost like time interventional series study. Okay. So what about your team? Any ideation to be done? You were very, your this thing had gynae, lot of questions you had. So let's do one gynecology study based on this. So Harpreet was quite enthusiastically discussing in the notes also. You mentioned you wanted to do something in gynecology, right? So let us take that. No, not you. All of you will help you. So you can say what is in your mind and see if we can use a similar study using this methodology, this design.
We can use the hormone replacement therapy for the treatment of the vasomotor symptoms.
Vasomotor symptoms in menopause. Okay, so that's the thing. What is the design of the study here?
It is a randomized.
Open label.
No, that's fine. Randomized this kangaroo mother care versus no mother care. So it's easy. So tell me the research question. So ideation and outcome and this thing, you have to make a Faridkot. Everybody will help you.
They are not helping.
So okay. We will help you with that question. Okay. Look, Dr. Harpreet, this is where are we in this? Where do you think we are now in asking the question? Where are we in this 12 items? Number 1, ideation. We have an ideation. Idea is that in menopause, I want to do hormone replacement. What is this?
Hormone replacement therapy, HRT.
No, but hormone anyway you are giving it. But in vasomotor symptoms only. But so again, when you are asking the question, what is the word equipoise mean? No, equipoise. The word equipoise means there is no real answer whether to do that or do this. That is a research question. Whether I should stop antibiotics at 5th day or 7th day if cultures are negative. किसी के पास आंसर है? कुछ लोग बोलते हैं 5 पे बंद करो, 7 पे बंद करो, 10 पे बंद करो. That is a good question to ask whether to stop at 5 in the same cohort of patients. But is there an equipoise in your question? HRT देना नहीं देना?
Time scale of around 6 weeks.
No. So you would know better. I don't know that. So you will have to say, is there an equipoise between gynecologists? Between gynecologists, is there a differences of opinion on 6 weeks, 3 weeks, 5 weeks?
3 months.
Huh?
3 months.
My question is very simple. Is that what does your Foxy guidelines say about the HRT?
3 months.
That is the guidelines. Guidelines say 3 months, based upon some studies. So what does this study add to the knowledge? Evidence, yeah. Does it change any practice?
Probably in my area, where I'm doing the study.
People who are, it is a known fact that giving HRT is good for you. So I'm probing you to think laterally. Yes.
But patients are not using it. It is not being prescribed, and females are not taking it confidently.
So then comes the intervention would be not HRT, education. Offer education among the people
who are already prescribed HRT, whether if you do, everybody says आपने लेना है, prescribe करके दे दिया, उसके बाद एक group standard अभी होता है ना ऐसे ही. Now you should say one group I will train a nurse, I will put them, they have to report to her daily, they have to show me the empty strips at the end of 1 week, every week she will click a photo and send it to that nurse. Nurse says yes, we have taken the medicine, and then you will now evaluate their symptoms after 2 months and say this group is better or that group better and what is the compliant best group. Now the intervention is not HRT, education is the counseling. So that can be a study. That can be a study. But the other one there is no equipoise to it. Yeah.
Something just giving you an idea. You know, you can even do an intermediary medicine study also. Like the old practices blended with the new conception. Like HRT is documented. Some hormone in some fixed dose for a specific period of time. But then you may be knowing some people taking something else, like soy protein. Yeah. I've heard, है ना? So that is what is going on. So you can do a research like testing the old school thoughts or whatever is prevalent in the society against what is recommended. Like a Making to HRT with the instead of actually giving hormone, you give something else which is prevalent. People say there is a belief supplements or something which have hormone-like activity. It's just giving me an idea. Talking means intermediary medicine studies also on large scale which have a social benefit can be done, and that too on a large community based. Then multidisciplinary, multi-centered changes and lifestyle, something else that you take which has Ayurvedic like benefits or actions, something like a drug which is already proven to have something and then compare does it prove, like बहुत लोग कहते हैं कि डायबिटीज कंट्रोल करने के लिए people take कुछ खास तरह के उसको बोलते हैं शुगर प्लांट, उसके पत्ते खाना. I don't know what that is. I just heard. Trust me. मतलब उसको पता नहीं वो शुगर प्लांट है, उसके कितने लीव्स, कोई 2 खाता है, कोई 10 खाता है, 15. A study can be done. Does that actually help against a low dose metformin maybe or something? I'm just giving a very vague idea. These interdisciplinary studies can also be done.
So
and
look, you need to. So once you get an idea, then you have to see what are the ramifications of it. That is where the focus group discussion happens. Somebody will tell us why not this, why not that, be receptive about that. Or you come up with a totally different study also, no problem. All I'm saying is that this exercise is to make you understand the thinking process and the framework for a study. You can make any hypothetical, you can choose on Google and ChatGPT and you can get your PubMed में search करो देखो HRT का और कोई studies है कि नहीं similar. Okay, what about your team? Any thought process? No, I want to about community medicine. Okay. अपनी वारी कैसी होगी? वारी क्या? Okay, so community, OBG, physiology, anywhere physiology will fit, right?
You were actually trying to fit in dermatology.
Postpartum depression.
No, so even if dermatology may not come directly, you can take up certain areas of the 12 steps, no? There can be, you can do the ideation part, schematic review part, reviewing the study design, so many aspects you can cover. It's not necessary. The skill set is not only for your specialty, only one specialty can actually come in there. Not all will come. So, anything mind with your postpartum depression. Fantastic. That sounds interesting. So, listen to that group's Amritsar, right? Yes. Amritsar. So, postpartum depression. Okay. And what is the current evidence required? I mean, is there a question along with that? I mean, if you breastfeed, you get more depression or less depression? Is there anything?
Ladies who tend to end up into postpartum depression, they tend to refuse to feed, to lactate their babies. That is the evidence that
if
there
they
is.
lactate after
There is something called
more of the oxytocin will be released and
No. So you are saying in the postpartum depression, they refuse to breastfeed.
they are pursued
Okay, so you are saying the oxytocin release might help in rebalancing the hormones.
More attachment to the child.
Agree.
Okay, and other factors.
So the population should be post. No. One second. So the what is the population base? Postpartum women who have depression.
This is just an idea. We need to define our question.
Sure. So I'm just trying to help you in the process. I'm not saying it is this thing. Maybe you have to work more on that. So obviously we have had 4 discussions done, 4 themes. We have heard the questions. We discussed this matter. The question is more important than this thing. We discussed this. Okay. So I want you to write down this 4 items, 5 items in your research questions. Once you write down this picots, I'm telling you again and again, the word picots you take home, that will give you more yield than any other studies which you have learned again. Whether it is journal club review or whether it is answering a question in your life itself, picots will be very helpful. Okay, so population we mentioned, roughly, postpartum women with depression, that's very specific. Okay? And then the intervention is educated breastfeeding or coerced breastfeeding, you have to find the correct words for that. And intervention would be, a comparator would be non-breastfed postpartum depressed women. So you are going to randomize the women. Now the ethical question comes is it ethical not to persuade a postpartum women not to breastfeed? Or not to persuade to breastfeed? Is that ethically correct or wrong?
Ask the mother to refuse to breastfeed. If you think she is going to harm, then it could be the expressed breastfeed that can be given to the baby rather than putting the baby to the breast.
So now then you have to say, so we are not depriving both the groups of children, number 1. Number 2, we are not depriving the possible oxytocin in both the arms. One is suckling helps, one is expression is the other one. That's the only difference here. Both the children are not deprived. One mother is directly feeding, one mother is indirectly feeding. That's now the comparison. You got it? So again, that is when you ask the ethical question, is it when you compare one group, say HRT for example, if you were to take, one arm you say give HRT, one arm you cannot not give HRT, right? So that's unethical. Therefore you will say, one group I am educating heavily, the other one not educating, and we will say ultimately education is extremely important for everybody for any treatment, right? What about your study? Population. What is the population?
Admitted patient is medical and surgical ward.
Okay, is that suppose you have a patient admitted in medical and all patients admitted in medical and surgical ward. Suppose patients admitted for herniorrhaphy who did not have a fever. Do you want them in the group? Uska culture to gaya hi nahi. So you need to have a specification now. All the admitted patients who had a blood culture. Then only your study will go ahead. No. How many of them did? Now what is the intervention here? What are the 2 methods? Biofire cannot operate without conventional blood culture. So that is a problem.
Every method is interdependent that way.
So again there is some ambiguity there. You need to fine tune it. So you need to go back, search some other studies and all that. Quickly come to that question. Okay, so that's a modulation in this process is that. Okay, so your population was defined. Intervention is education.
8 to 14 years.
Yeah. How do you align that study with the methodology which we have given to you? Is it similar or different?
Similar.
Similar. Okay. So you are on top of it and you can prepare and come back with a proposal. Okay, so let me. So I just put a Google search on, PubMed search on antimicrobial resistance. So when you go to advanced, you will see that this is the queries which have come, and they have used up all the fields. So it basically automatically looked at all the fields. Now, when you do the search, also have you heard about the search strategies? Yeah, so if you do a search, it is good. So you need to say, I want to look at antimicrobial resistance among patients with, antimicrobial. The word antimicrobial resistance in surgical patients, suppose. So I will say surge and surgery. I want surgical. I will put surge. So surgical will come up. So surge and apostrophe this you put. It's a wildcard, so therefore surgery, surgical, everything will come. There are 5,36 lakhs of surgery, surgical, everything. It included all. So go back to the answer saying that learn how to do PubMed searches, how to do your search strategy based upon your Faridkot. Because that will allow you to get your review processes more enhanced. So I did that now, and so you can see various studies which are coming in. You glance through that based upon what the display options are, most recent first, and after that this comes. Do that. I just put in suggest randomized control studies regarding antimicrobial resistance to ChatGPT. Short versus long duration. Febrile neutropenia, early stop or conventional duration, that does not come. Community acquired infection 3 to 5 versus 7 to 10 days. Okay. De-escalation versus fixed broad spectrum therapy. Rapid diagnostics versus biomarker diagnostics therapy. Rapid molecular versus stewardship. So many options. Okay, you might get lost in there, but that will give you some ideation thinking. So I now refine this range in surgical patients.
You can side by side do both. While that is happening, you look at them PubMed and see what all you want to check. So that's antimicrobial. So single dose versus multi-dose surgical prophylaxis. Surgical prophylaxis seems to be a good option for you. Because surgical, it's more, when you ask me the question, try and ask a question which is doable. All of you have heard about the SMART goals, right? SMART objectives. Specific, measurable. So like that you say, surgical prophylaxis, done, not done, what is the outcome? Surgical prophylaxis is a common practice for you. Is there any unit doing not following surgical prophylaxis? Everybody is using. But is there a difference in the antibiotics used by everybody?
Yes.
That may be a good option for you. So you can say, I would now, even if you say I don't want to put a big study on comparison and clinical trial, it can be simple observational study we want to do on surgical prophylaxis, the current this thing, and I'm going to compare second generation versus third generation who is using it. That's one way of checking. I'm just giving some idea. Please refine it yourself subsequently. Even if it is an exotic study you want to plan based upon your trial, do that exotic study even if it is not possible in your team. But that will give you an understanding about how to define something. Okay, so anything on postpartum? Are we on? This is on menopause. Any other studies on menopause you could get?
Reframe your own title, sir.
Okay, what is the
So
title?
the comparison of the surgical menopause with the natural menopause on the bone health, osteoporosis.
Okay, that sounds good. So, you are not intervening, it's an observation study.
Right, sir.
Because you cannot randomize and say, I will do surgical menopause. Surgical menopause happens automatically, right?
मैम ने पहले सजेस्ट किया था उसमें हमने लिखा effect of intake of hormone replacement therapy versus lifestyle modifications for 3 months on relief of vasomotor symptoms in menopausal women. A single blind randomized control trial.
So one group is depriving hormonal
Yes,
therapy.
yes.
Is that ethically correct?
We don't
Right,
need to give.
yes, sir. It is not like it's not a life-threatening kind of a thing that we
So there
have
is
to be
equipoise.
Yes.
So say there is equipoise. Start with that word. If there is equipoise, good to do a study. Nobody is. It's iffy. That is no issue. It's observation only. Yeah? That is also good. See, look, the exercise is making you think through, write down the proposal, and get the rebuttal. If the judges say it's not a good study, sometimes they may say it's a good study, but you can continue with the study, no problem. After ethically right. Okay, so with this, what about this group again? Postpartum? Still working on. Okay, good. So everybody has homed into some kind of a question now, which you will develop over dinner now.
Sir, one question I had read that I will do a lifestyle modification on the CVD risk in diabetic patients.
Okay, in diabetes patients, one group you will promote lifestyle modification by whatever means you have, a personal trainer, whatever. The other group, no.
Other group is normalized.
Yeah. And then you are going to look at cardiovascular risk.
Risk I want to check HbA1c, CHAD score and all of that.
No. So again, when you say we have to be specific, I want to be HbA1c, at 6 months. Now, does cardiovascular risk manifest within 6 months? The chances of events happening in both are really less. Events कितना आएगा दोनों में? 6 महीने में कुछ नहीं होने वाला ना, तो उसके लिए 10 साल लगेगा. That is surrogate marker, yes.
That can be a short-term standard.
That sounds doable. That's a good idea. So now build on it. How do you standardize millet versus this thing? Will you ask them to buy
or will you pre-pack it and give it to them and monitor them? Because when you make the cut, so all that elimination has to be planned out. Whether you will pack and give it as a standardized manner, because they might eat more. He might eat cheese along with that. अगर वो और कुछ का डिजर्ट खा ले, उसके अलावा भी कुछ खाएगा ना वो, उसको भी स्टैंडर्डाइज करना है. No problem. So I'm just saying there are various confounders will be there. So that you have to control as much as possible. So it's putting the methodology, how are you going to control it? I mean, control those confounders. Absolutely do that. Okay. So when you do this, so okay, so this is your research proposal framework. So please access it, come here, make a copy and put your institute name here and then share it with everybody else. Make a copy. Okay, so utilize digital tools as much as you want to, but save as. So this is the other copy you will make, and from here it is easy for you to make the PowerPoint presentation. कर दिया था मैडम, ग्रुप में ऑलरेडी है, व्हाट्सएप ग्रुप में है, ये 2 गूगल फॉर्म्स थी. Okay, so we will make a pause here and imagine you have made the proposal and you have submitted it to the research committee. Dr. Rinchu will let us know how research committee will look at it. Okay, so you have an option to now prepare your research proposal based on what the research committee actually is looking at. Oh, gee.
Thank you for the cup of tea.
So,
can I take over informally by the time you teach?
Yeah.
Okay, so you know me. We'll skip all the protocols, the protocol of introduction of the speaker and all that, because you know me. All that you need to know about me is that I am a biochemist. And why am I emphasizing on that is, and I'm very proud to say that biochemistry usually in most of the institutes is the maximum earning department.
provided there are clinicians sending the samples.
That provided is a mandate for them. They can't do without us.
Yes, ma'am.
Okay, this is not to prove anybody down. This is just to say that when you think of the ideas, think that there is a biochemist ready to join you for an inter-institutional work. All set after this workshop, right? Think of good ideas. No, I personally think no study or research can get completed until unless you get some biochemistry done.
Ma'am, in my study, I have included biochemical markers.
Thank you. All right, so that's my introduction. Should we go ahead? By the time they picks up my presentation, I heard Dr. Joseph John while talking to you people again and again, repeatedly, and at this final level, saying that as long as that is ethically acceptable, as long as that is ethically acceptable. Any idea that you were talking about? Did he say that? I was counting so many number of times that he didn't realize. So how do we decide whether we are ethically correct or not?
All right. Agreed. Okay, I'll reframe my question. Okay, there are you may come up with some more things to it to define that. But is there some standard to it? Where do we stop? What is inclusive in the word ethically right? What all things get included? Who decides that? And what is the extent? That is my question. You will say something, I'll say something. For you, something might be ethically non-acceptable. For me, it should be ethically okay. It's a personal. Who decides? Just implements what I'm asking. Makes sure that you watch those standards. Where do the
perception ही हो गया मैम
वो तो perception हो गया पर
perception के according
तो objectivity से subjectivity में आना कहाँ से आएंगे? There are guidelines हाँ जी complete करो.
मैम follow the principles of the medical ethics.
All right. And who lays down those principles?
Accrediting labs.
ICMR is the one. So ethics committee follows the guidelines laid by ICMR, which gives us the ICMR ethical guidelines. और वो guidelines क्या है? Based on certain principles. जिसके आंसर आपका क्लिक देख चुके. What are those principles? किसने दिया था वो आंसर? Four ethical principles based on which ICMR has given the guidelines. Non-maleficence and justice. Is that all? Now my next question. मैंने अभी क्लास में भी और पहली क्वेश्चन पूछ रही हूँ. Is that the right way? That way I will come to know where you are already. किस पे build करना है, right? Okay. Is that all? Are these the only foundation or there is something else also? These are the basic core principles. But then ICMR guidelines जो वो release करती है उसमें जरा 12 अब वो 12 भी देखें पता होगी वो भी सब आपको. Because सुबह से मैं भी आप लोगों की तरह as participant I've been attending the session and I realized that everything that I wanted to say has already been covered. So it's just a revision. And I'm just going to share with you what is ethics committee, what is its role, responsibility and challenges. That's all. I'm just going to give a shape to it. That's all. Okay, my presentation is there. Okay, can we just count out which are the 12 थोड़े से out of those? They have been mentioned by the speakers before me a number of times. Let's see if you can recall. Which are these more ethical considerations beyond the 4 basic principles? Tell me. Informed consent. Okay. That is a component of taking the informed consent. Privacy. Absolutely. Confidentiality. Yes.
Risk benefit.
Okay, that comes under justice.
Ma'am, all of these are mentioned in participant information sheet only.
हाँ, पर हम सिर्फ वो components देख रहे हैं क्या क्या है. उसमें हम mention इसीलिए करते हैं ना because these other than the 4 other components.
We give the participant information sheet as well.
तो उसी के components हम देख रहे हैं कि कौन-कौन से हैं. Which other than these 4 are the components of ethical considerations.
Yeah, the compensation you have to give.
एक आधा कुछ ऐसा जो अभी तक mention में नहीं आया. If anyone of you knows.
Adverse drug, any adverse reaction or any adverse.
Okay, documentation of anything that goes wrong. Okay. Anything else?
Like guides should be available if somebody is coming to work.
Cost benefit.
Facilitator should be there.
And
benefit risk assessment. That is called as benefit risk assessment, as such mentioned by Dr. Ranjit also, and sir ne bhi mention kiya benefit risk assessment also. Anything else? One thing that I did not hear from the previous speakers, मतलब by chance reference में नहीं आया.
Alternative options. The informed consent.
we need to take care of the vulnerable groups especially.
Yes, absolutely. That has also come. Absolutely.
And the participants.
Special considerations for the vulnerable groups so that they don't get exploited. Absolutely.
All treatment should be on intention to treat basis.
वो beneficiaries में ही आ गया. Institutional preparedness is one. We should not come like that. Chaliye, we'll go through this formal presentation hurriedly because you will realize that most of it has already been covered. There's nothing very specific about it, yet we'll go through it. So by this time we know why ethics matters in medical research is. It protects the participant rights and dignity, right? It ensures scientific validity and transparency. It is mandatory for publication and funding, and now you must be knowing that quite a number of index journals now even reject the submission if the IRC approval is not there. Agreed? So this has become a mandate now. So this is not only about compliance actually. When we talk of ethics in medical research, it is about building faith in medical system as such. So historical basis was very well covered by Dr. Ranjit. I will not go through that. That he has already taught you in detail, right? ICMR guidelines were given in, anybody knows? 2017. And there is one, okay, this covers just one thing before I move ahead. ICMR gives us the guidelines for ethics. And who gives us the guidelines for the legal part?
Advocate in the IEC.
Scientific review.
No, you've not got my question. IEC actually makes sure that we conform to the guidelines laid by ICMR and one more regulatory body that takes care of the legal part of any research. What is the full form? Wait till you get the answer. Oh, sorry. Okay. So what is an ethics committee? EC, or also called as Institutional Ethics Committee. It is also called as, there is one more name for that. IRB, and the fourth one. Institutional Review Board, EC, IEC, and one more. REC. What is R?
Institutional Review Committee, Research Committee.
REC. The answer has already given by Dr. Joseph Chan. And he said, you'll come to know. He said that there. This tests your presence. Registered Ethics Committee. Now my question. He asked the same thing, you'll come to know. It has its regulation. It also has to be registered. He said that. Does anybody know why the word Registered Ethics Committee? At least something is there, which is new for this session. Because every ethics committee has to be registered. It itself has to be registered. Now, what is it? It has to be registered with? Is there one body? Multiple? What is it? Before I commit a mistake, I must be doing that. Thank you. Thank you so much. If an institutional ethics committee is actually dealing with the non-drug trials or the observational studies, it has to be registered with National Ethics Committee Registry Board for Health, Biomedical Health and Research. This is NECRBHR. Yeah? National Ethics Committee Registry for Biomedical Health and Research. If it deals with observational study or a non-drug trial or a non-clinical trial. But if the Institutional Ethics Committee is dealing with some drug trials, clinical trials, bioavailability, something like that, then it has to be registered with, and the full form is CT. No, I heard CD, sorry. CDS. That is Central Drug Standard Control Organization. Alright, we move ahead. Now, Ethics Committee. So, 4 names, all synonymous. Right. So, important part in this is that Ethics Committee should be independent, multidisciplinary body. These two terms are very important. What do we understand by independent? Every institutional ethics committee has to be an independent body and it has to be multidisciplinary. It can vary from 7 to 15 members. So what do we mean by these two terms? We say our judiciary system is independent. What does that mean? The same. Absolutely. Absolutely. That is definitely there, but independence does not, someone coming from outside does not entail its independence.
So in other words,
Please.
So unless it's there, principle is there, they want to do, and she is the ethics committee chair or ethics committee. The ethics committee can overrule the decision of the industry. That's essentially the independence means.
So I am answerable and in submission to the director of our institute as Professor Department of Biochemistry, but if I am a chair of IEC, I am not answerable. I can give an independent decision. Right? It has to be multidisciplinary, otherwise it is not acceptable, it cannot be registered. What does that mean?
Usually the chair is not from the institute.
Yes, not usually. It's a mandate for the chair to be the external person. It's a mandate. It's a mandate, sir. You're absolutely right. Okay, multidisciplinary means departments, and that includes, okay, let's see. You need to have medical scientists, you need to have clinicians, and you need to have basic scientists also, that is also a mandate. Other than that, you need to have non-scientific persons, non-medical scientists, and that include a legal advisor, which can be a lawyer, okay? One layperson. Yes. Layperson is social worker only, right? Or a community person, and then an ethicist. These are the minimum. So that is why the number can vary from 7 to 15, and this is how it is multidisciplinary.
Why do you think that is made? Why do you think that rule is made like that?
To ensure independence.
One is independent, but why do you want multidisciplinary there? Independence can be made separately, like judiciary. Why is it multidisciplinary?
It is the intervention should be viewed from the point of the public.
That to remove the bias.
Absolutely. We are default pathway is me, I, me, myself.
Medicine people will think in one line.
Yes.
They will have one skewed way, their way. Surgical people will have different way. A biochemist will think differently. So that is that. So in general, just summarizing, it reviews scientific and ethical merits of the protocol and protects the vulnerable populations. Okay. What is an institution review board? The same name, except that this is a US-based equivalent of ethics committee. This is a very strong regulatory framework, and it is adopted by many international institutions. Otherwise, it is the same, and this IRB applies to India also when doing an international collaboration.
So one more question. Why do you think they made earlier the ethics committees were not registered? Why do you think why it is registered now?
Because all are working in the same framework. And there will be
standardization. But people were even violating ethics committee. So you will choose the guy who is a yes man to me only into the ethics committee. So whatever I say, he will agree. Usko pay karo. That, to avoid that, registration has come in and the guidelines have come in, and you have to submit their CV and other things to the registration board.
There is a protocol for IEC to get registered. Again, reconstitution sabse pehle. Again, it should be independent body. It should be multidisciplinary. Then it should have laid down SOPs as directed by ICMR, and then only through a specific form, online, the registration process can be initiated and completed. Okay. Now, what are the objectives? It ensures ethical compliance, monitors the consent and risk-benefit ratio. This is just overall summarizing. We just discussed, there are so many of them. And oversees the participant safety. So, why medical faculty must care for an IEC approval? This is a legal requirement before any study now. So this is a mandate. It's a legal requirement. Neither can a researcher do without it, nor can an institution afford to have a ongoing research in the institute without having a proper approval. It protects the researcher as well as the institution and determines the publication acceptance that we all want. So research requiring the EC or IRB approval, like which are the researches which actually require this approval? Clinical trials, biomedical studies, student dissertations, community studies, studies with humans or human data involved, or any samples of humans which involve these. So what should be the composition? Sir, only told the external person, chairperson, it's a mandate. Internal person cannot be the chairperson. Member secretary is an internal person, and then we have a combination of scientists as well as non-medicals. Clinicians, basic scientists, lawyer who is a legal expert, ethicist, a layperson, community representative, and a social worker. These are minimum to say. Now, what is the role? Ethics Committee reviews the protocol, that it is scientifically sound, and approves it. It monitors the adverse events, brings its documentation as you pointed out. It ensures participant safety and reviews the amendments and continuing studies. So, Institutional Ethics Committee is not only concerned with giving the initial approval, it follows through and ensures that throughout the study conforms with the laid down ethical and legal guidelines. So this follow through and overseas is a very important one. It's not only the initial part, the IEC ने approval दे दिया, now it's done for me. अब researcher जाने या उसका काम, it's not like that. So Institution Ethics Committee has a responsibility to make sure that till the time the study is completed, it conforms to all the laid down guidelines of ethics and legality. Again, same thing. For science, if scientifically any protocol is scientifically poor on conception, it is equivalent to being unethical. So the methodology must justify the risk. There should not be undue risk to the participants. So Ethics Committee evaluates both what? The scientific methodology as well as the ethical component of the protocol. Ethical principles we have seen on which it is based, does a risk-benefit assessment, takes informed consent. We've seen that. We'll just rush through that. Data protection and confidentiality is maintained. This can be done by anonymization, secure data storage is important, and DPDP Act compliance. This DPDP, what is this DPDP? You gave me the answer. I said wait till the answer comes. Digital Personal Data Protection. This act was given in year 2023, and this is the one which gives the guidelines for the legal components. So this is DPDP Act year 2023. Because I can see everyone is sleeping now. Chai kahan gayi? Chai ko bhi sula diya aap logon ne hum to.
So ask questions, chocolates can be given. Khatam?
That one is for me. Because I deserve 1 minute for लोगों के साथ डील कर रही हूँ. Sorry for that. So okay, what types of ethics committee or IRB reviews are there? 3 types. Broadly, I think Dr. Ranjit, you talked about it. है ना? The types of reviews मैं वही देख रही थी. Everything has been covered. Exempt, expedited, and full board. Exempt, no intervention, absolutely. Expedited, minimal risk. Maybe slight, but hardly any. Minimal. तो वहाँ पर थोड़ा सा the board can review and pass it on. Full board, where there is good amount of risk involved and a lot of intervention is there. Usually surgical procedures or कुछ इस तरह के procedures. So 3 main different categories. So what is the workflow? Sir, मुझे याद है. He mentioned submit, review, solve all the queries, refer to you by the IEC, rectify them, resubmit, and take the approval.
Which number is it on? On the cycle में कौन सा number पे आ रहा है? For it to keep activated. In that sheet, in that laminated sheet, कौन सा number में है अभी अब? Number 6. So I want you to go through that in between to be active so that we know that where we are. Map.
So again, what I said, EC is not only about the initial approval to the proposal, that is about the continuing review that all through the study till it gets concluded conforms to all the ethical and legal guidelines. It also monitors deviations and is concerned with the safety of the participants throughout.
So once pause one slide before. So I want you to understand that DPDP. So look, this is something which I want you also to read a little more, because now the digital platforms are coming. Government is coming up with the number Abha ID coming up, right? So Abha ID means that the details of a patient whom you have seen in your hospital from Faridkot, if they go to Patiala, by accessing the Abha ID, they can get this details there. But that also comes with a risk that somebody might access this data of this particular patient who has a survival of only 6 months probably, and his brother may access the data for its plot case and all that. He may misuse it. Or the data can go somewhere else for them to do mapping of the people, etc., insurance companies, whatever. It can be sold. That's why this comes in a lot of cyber security along with that will come. And that is the reason why if you want to do studies across the institution, you must get knowledge on how the data will be managed. If the data goes abroad, it has to go through another set of compliances are there which has to be done. So no identification can be put in there. It has to be anonymized data only transmitted for the studies. Which means it is also a challenge for you. So patient Malkiyat Singh with hospital number this, with date of birth this, we will be able to map all the three and find out this is the map. If I have to send it to somewhere and I have to do a data query, how will I know is this Malkiyat Singh or somebody else? So I have to have a separate code generated for each of these patients, and then only I can send it. So understand the nuances of sharing data when you have to send it somewhere else. So if somebody says share the data, ask him to give an email asking clearances from that place, and especially government to government, any name, data, other number, about details. Be careful about sharing the data. It's not a no, but be careful. Traceability has to be created.
There are two slabs under this DPDP. Actually, there are multiple components. I just mentioned it here. One is how much of the data. Second is for how long. And there are conditions to that. How much data means the bare minimum that you need for the research. No extra information to be taken. And any information that you take for the patient has to be justified in accordance or compliance with the study that is being conducted, that it is needed for that study. Number two, for how long can you have the access and you can store the data, that also has to be justified. And the data cannot be stored beyond that. So both the things under DPDP Act have to be justified. How much data and for how long it can be stored by the researcher.
Yeah, deviation and safety. When you are doing a drug trial or anything, and the data, as I said, so our data clinical coordinators will have to file a deviation to the ethics committee saying that this happened one day before. Repeat sampling has to be done. Even if you have to take one extra sample on another day, there has to be a report to the ethics committee that we have to do.
Responsibilities of the PI. Who is PI? Principal investigator. Alright. Accurate submissions. Safe conduct of the study. Reporting the adverse events and maintenance of the records. What is there? Which adjective when you maintain the records? There is a specific adjective which has been used before in the slides. Transparent. Absolutely. It's not that while documenting I change it because ये थोड़ा सा very अलग हो गया. I wanted the study to show this, so I changed the data. I cannot. Transparency has to be maintained. Absolutely.
So what is ICOA?
Legible, accurate, legible, contemporaneous, original,
Complete.
ICOA C it was. Attributable. Whatever you so residents को ये सिखाना है. So what is record maintenance? What is the difference between falsification, fabrication, and forgery? So falsification is if it was 10, you wrote 20. Fabrication is there was nothing, you wrote 20. Forging is the sign. All the three are bad. Okay? So this happens sometimes in thesis. Beware of the students who come overnight on thesis ready kar diya. So that is 100% fabricated. So if you ask ChatGPT, it will fabricate for you also. So please be aware of it. And that is something which we have to take home, saying that even if you don't do a study, it is fine. But don't do these things.
So what are the common reasons for any proposal getting rejected from the IEC? Poor methodology, scientifically low. Incomplete documents, very weak consent process. Consent process has to, there is a proper way of administering consent. First, the consent form has to be designed and then how to administer it. And unjustified risks. Again, a proper assessment of benefit to risk assessment has to be done. So unjustified risks are absolutely not acceptable.
So once again, I need to answer a little bit on the consent form. So consent form ऐसा नहीं है कि आप ये ना इधर sign कर दो. वो नहीं है सारा consent. Consenting has to be sat through and discussed in full length. It is difficult for a clinician. I am telling you for myself. So you need to have somebody who can help you with it, and at least you can go there and say, have you understood these? In fact, we have for transplant patients, we are now doing audio-visual consents. Because it's such a complex procedure, we have to give the consent forms even for trials, we give it 1 or 2 days before in their language. So ask them first, typically should be Punjabi, and they say whatever language, and then you send it to them. AV consent ke liye separate consent, and then you sit down and go through each paragraph and ask them to sign at each paragraph. In the consenting process, always mention what are the alternative options they can take. And if you don't go for treatment, what is the outcome? If you go for treatment, what is the outcome? Like inform, this has to be there in the country. So abroad it is very easy to implement because 9 out of 10 people can actually implement it. Our problem is little extra because an uneducated patient is an extremely difficult, and therefore audio-visual, if you have an option, that is going to be mandated very soon, but otherwise it's a good thing.
So that reminds me to ask, actually these are only the pointers. What are the challenges? Sir has mentioned 1 or 2. Can we come up with some more challenges for taking an informed consent? Language barrier. Main. Then? That meant me to ask you, what is the difference between consent and assent? Assent, sorry.
Assent is done after 7 years of age when you have to take it separately from the child, and the child is able to understand the risks.
Does it have a legal value?
Might have to. The consent of the parents.
Legally, it is not considered consent, but it has a value that the patient understands what has been explained to him, has a mental capability of understanding and giving her own consent to it, but it is not considered consent because he is not legally not an adult. So in that case, the protocol has to be finished. In case there is no guardian, parents available, only then it has a value, otherwise it doesn't have a value. Yet, there is a procedure of explaining it to the minor who has a mental capability and can understand and can involve himself into the procedure.
What if the parents get consent and the minor does not consent? I'm the only one to whom this is relevant.
Yeah, this can happen in cases of also where the child is
It can happen the other way around also, ma'am.
Then it is clear, the parents are not giving consent legally, you can't do it. But what if the child does not give consent, the parents give consent?
In that case, if there is no non-beneficence and there is beneficence in the decision, and medically the clinician also thinks that it is in the benefit of the child, the minor, and the parents also want it, then it there are many who are teaching phase 3 part 1 students. We are teaching this in ACTOM nowadays.
So you have to document with
in the file that as a patient, if the child has been explained, she is not understanding the process. She is unable to say yes or no. But the parents have understood and they have agreed upon. So in the larger benefit of the child, we are going ahead with this. So one paragraph documentation has to be there.
So that means for every minor, assent has to be there on the
Assent
file
and
along with the
consent, both.
consent.
Both assent and consent.
Have to be documented.
12 years and above, it should be written assent.
Yeah. 7 to 12 is only a verbal, haan, kar raha, but that is enough. We document that, patient and child agree. But 12 to 18,
it is written.
they have to sign or write. So that means 12, the 7 years to 12 years, only one consent by the family, parents. 12 to 18, it will be both
consent
and assent. 18 above, it is only consent. So only one document for 7 and 18 above. 12 to 18, two documents.
So now Ethics Committee and IRB in Punjab, what is the local perspective? We know that it is growing research output nowadays. It is really growing. That is why we are here. Just because there are stringent NABH requirements and NMC requirements also for research which have become a mandate. So there is a increasing output now. And at the same time we are strengthening the IECs also across. But then there are certain challenges also. One, there are limited ethics training. How many of you have already gone through any of these ethical training? Anyone? That is a major problem. Then variable EC function. Though there are guidelines, we've seen ICMR guidelines for the ethical component and for the legal digital personal data protection, DPDP, right? 2023. And there are guidelines for the registry also, which was mentioned. Every study has to be done. Despite that, there is a variable EC functioning across the institutes. And all the more because of the increasing interest and the pressure on the researchers, now the committees are also getting overburdened. So we have to cope up with that, conduct more trainings, involve more people, register proper ethical committees so that more of the authentic research can be carried out. So ICMR Guidelines 2023 के key points क्या हैं? It defines the ethics committee responsibilities, it defines the vulnerable group and lays down the guidelines for their protection and lays down the standards for biomedical research, all the ones. Then we have international collaboration. Sir mentioned most of them. How? This is more regarding the data preservation and ensuring the transparency and the confidentiality. Now, ethics in publication. There are well-defined authorship norms. We all know that. Who can take the first authorship? Who can be the second author and all that, right? And we should avoid plagiarism. There are apps which can detect, you know that, and especially for students ke thesis ke liye to. And they are the ones who are facing the brunt actually. And trial registrations should be actually properly done. What are the future directions? Again, what we mentioned, digital ethics platforms are coming and the control and privacy, confidentiality of the data, their documentation will be very soon become a huge challenge. And there will be stringent guidelines coming up which we will have to follow. AI research oversight is a challenge. For this, we need to have improved ethics committee training. That is the only answer. Making people aware what are the norms, what are the guidelines, what has to be conformed with, what we should not make a compromise with. So conclusion, ethics committee or institutional review board. Can it be called institutional research board also? Yes or no? Yes. That is not the right term, though it is very frequently but mistakenly called as institutional research board also, but that is not the right term. It should not be called institutional research board, it is called institutional review board.
So in our center we have research committee and ethics committee.
both expertise committee, research committee sit together. Here this EC subject RC subject. RC is essentially looking at the methodology and the process. This committee is looking at the individuals on this.
Actually, as I pointed out, we have divided the roles. So clearly pointed out. We have one research committee which looks at the methodology only, and the ethical component is looked after by another committee. But then our research committee cannot be called as IRB. IRB, when we say, means Institutional Review Board. That is the only acceptable international term.
A single body is called an IRB,
Actually, these are synonym words. It protects participants and researchers and is critical for credible publishable research. Thank you so much for patient listening. Time for either your questions or my questions. Of which one? Okay, DPDP Act year. And ICMR guidelines? 2007. Alright. And then what is the name of the regulatory body with which any clinical trial has to be registered? Full form? Clinical Trial Registry of India. Okay. Does Ethics Committee have to be registered? Yes. With which body? Two different bodies depending upon clinical. बहुत basically याद रखो. Clinical trials with one, non-clinical trials अगर बाकी और studies हैं जिसमें clinical trial नहीं है या drug trial नहीं है, वो किसी और के साथ. तो अगर clinical trial नहीं है या drug trial नहीं है, observational study है या biomedical research ही है, तो it is National Ethics Committee Registration Biomedical Health Research. Okay. और अगर drug trial है तो full form CDSCO.
Clinical Drug
Standards Control Organization. Thank you so much. Thank you for your patient listening.
Thank you so much, ma'am. It was great discussion, great session. Now I request Professor Dr. Shashikant Dheet sir for the next talk. Sir is professor and head in the Department of Pediatrics, GGS MCH Faridkot. He is MD in Pediatrics with Fellowship and DM in Neonatology from prestigious institutions like GMCH and PGI MER Chandigarh. He is the recipient of multiple awards including FIAP 2025 Fellowship, and there are many more awards to his credit. He is having 62 publications and 27 chapters, and he has been there in many presentations, and he has fetched many grants as a project investigator from ICMR. His topic of discussion is Critical Trial Registry of India. So please, sir, over to you now.
So very good afternoon to all of you.
Good afternoon.
So in this workshop, we all are going to discuss about the research proposals, right? Yes or no? Yes. So what are you going to do with the research proposals? We are definitely going to start working on them, and we are supposed to start submitting somewhere, and ultimately we want to do it. So as of now, as per the mandate of Government of India, any trial, whatsoever it is, has to be registered with CTRI. So next 30 minutes or so we'll be discussing about a topic which will be mandatory to all of you. अगर आप कोई भी स्टडी करना चाहते हैं, कुछ भी स्टडी करना चाहते हैं जिसमें ह्यूमन पार्टिसिपेंट हैं, एंड आई नो हमारे भी ह्यूमन पार्टिसिपेंट होंगे ही होंगे, then we have to register it on CTRI. So CTRI is Clinical Trial Registry of India, right? So we'll just be going about a brief about this. So right now WHO has inducted कि जितने भी clinical trials all over the world हो रहे हैं, they have to be registered somewhere. और इंडिया के अंदर that authority is CTRI, right? तो आपको just CTRI नाम याद रखना है. Commonly जब हम छोटे थे, when we were doing MD, so जब भी CTRI का नाम आता था, तो हुआ सा आ जाता था दिमाग पर CTRI register करना पड़ेगा. And we were bit डाउन हो जाते थे, यार छोड़ो रहने दो, ऐसे ट्रायल नहीं कर रहे. At that time it was mandatory कि we only do RCTs registered in CTRI. So अभी ऐसा नहीं है. So RCTs के अलावा भी बहुत कुछ register करना पड़ता है. तो it is not a I hope and I am trying कि by the time this discussion is over, you people get a feel of what is CTRI and you are not afraid of this term. So अगर वो आपके दिमाग से निकल गया तो मेरा पर्पस सॉल्व हो जाएगा, right? Okay. So देखना टिपिकली माइक की जरूरत है. I think I'm audible. So if we
not audible here.
अच्छा इसके लिए So if we see about this slide, so this CTRI is mandatory for all interventional clinical trials, right? So this is also required for PG thesis, जिसके अंदर भी human participants होंगे. So any PG thesis involving human participant, why it is required? It is required to ensure the transparency and ethical accountability. And if we want to publish this to any ICGMA journal, any journal which is accredited, which is in published literature, so one of the mandate is it should be CTRI registered, right? So it also helps us कि two studies are not being done simultaneously or they are not being duplicated. So also whenever research is registered in CTRI, it increases the trust both in the publishers as well as the patients and as well as the researchers. So this is the mandate of CTRI. So which studies need CTRI registration? So this is very important. Like you can see that all drug trials, all vaccine trials, all biological trials, surgical trials, device trials, IUF system trials, behavior, lifestyle intervention, diagnostic accuracy, any prospective observational intervention study, and all PG thesis, all ये हम नहीं करते अभी. So all PG thesis with human participants. So हमारे लिए जितने भी हमारे PG thesis हैं, ideally they should be registered in CTRI, right? So अभी के लिए suffice here would be कि आपने जो भी research करनी है, आपको CTRI में register करना है. So just let's start doing this.
So there was one doubt here. If I want to do a prospective study, then it has to be registered.
Yes, it has to be registered as of now. So all the as of now, so all the studies which we are doing prospective registration is important. If I am going to do any study, it has to be registered. Then it has to be started. By retrospective study it means कि आज मैंने register किया, तो मैं आज उस पे काम कर सकता हूँ. लेकिन बैक डेट में नहीं कर सकता. मैं ये नहीं कर सकता मैंने 2010 में एक स्टडी की थी और मैं आज उसको रजिस्टर करूँ. दैट इज नॉट
हमने डाटा पिछला वाला लेना है.
डाटा ले
स्टडी आज ही करनी है.
हाँ स्टार्ट आज की है ना. स्टार्ट आज की है
तो कराना पड़ेगा.
हाँ यस. रेट्रोस्पेक्टिव डाटा है बट डाटा है ना. स्टडी हम आज कर रहे हैं. मैं वही कह रहा हूँ ना. स्टडी हम आज करना शुरू कर रहे हैं तो कराना पड़ेगा. देन वी हैव टू रजिस्टर कि हम वो डाटा उठाएंगे. लेट्स से कि मेरी थीसिस थी एमडी की 2005 में मैंने स्टार्ट किया और मैं उसको कहूँ कि आज मैं CTRI रजिस्टर कर देता हूँ. दैट इज नॉट पॉसिबल.
स्टडी आज करनी है पीछे वाली.
मतलब रजिस्टर जो आज करेंगे उसके बाद करेंगे आगे. यानी आज से पहले जो स्टडी एनरोलमेंट शुरू हो चुका है वो नहीं रजिस्टर करना. तो CTRI हैज डिनाइड इट कि ये पॉसिबल नहीं है, राइट. सो बिफोर एनरोलिंग द फर्स्ट पार्टिसिपेंट इज वेरी इंपॉर्टेंट. चाहे वो रिट्रोस्पेक्टिव डाटा हो. सो बाय रिट्रोस्पेक्टिव जैसा सर ने बोला कि मैं आज डाटा देखना चाहता हूँ, बट आफ्टर डूइंग दिस देन आई स्टार्ट लुकिंग फॉर दैट डाटा. यू गॉट इट. पुरानी स्टडीज जो हो चुकी हैं वो नहीं. But if I want to start, that's a separate thing, right? ओके, रिट्रोस्पेक्टिव रजिस्ट्रेशन का मतलब है कि जैसा मैं बता रहा हूँ कि मेरी थीसिस को मैं आज रजिस्टर हूँ, दैट इज नॉट अलाउड। तो CTRI अलाउ ही नहीं करता, जिसको काट देता है।
तो जैसे मेरी थीसिस मैंने कर ली, 3 साल आज खत्म हो गई, तो आई कैन रजिस्टर
No.
दैट एज अ रेट्रोस्पेक्टिव स्टडी। उसमें एम सब्जेक्टिव और वो सब तो मैं स्पेसिफिक कर सकता हूँ।
आप कर सकते हो, बट उसमें चीज अलग है कि आप वो डाटा देखोगे अकेला, आप फाइल्स दोबारा देखने के लिए करवाओगे। आप ये नहीं कहोगे कि मैं प्रोस्पेक्टिव कर रहा हूँ, चेंज हो जाएगा आपका।
नहीं, मेरा थीसिस होगा, सपोज कोई भी है कि मैंने मान लो कि CBC लेवल देखने, HB लेवल कितने हैं, मैंने
ठीक है,
थीसिस कर लिया,
ठीक है जी।
उसके बाद तब अप्रूवल लिया नहीं, थीसिस खत्म हो गया,
हाँ,
अब मैं कहूंगा मैंने CTRI पे रजिस्टर करना है,
हाँ,
तो आई टेक एट
हाँ,
रेट्रोस्पेक्टिव स्टडी
ये
ऑफ रिकॉर्ड देखना है CBC पेशेंट कमिंग टर्शियरी केयर हॉस्पिटल जो पुराना 3 साल का
ये यही बोल, हाँ, ये दिस इज ओके,
दिस इज ओके,
दिस इज ओके। आपको उसके लिए दोबारा एथिकल कमेटी का अप्रूवल लेना पड़ेगा, लेकिन वो डाटा सेट आपका बेकार नहीं जाएगा,
वो डाटा यूज कर सकते हैं।
हाँ, बट वो यू गॉट द पॉइंट कि वो उसको थोड़ा सा काम को इधर से घुमा के करना पड़ेगा। हमारा सारा काम दोबारा, प्लस वो टाइप ऑफ स्टडी भी चेंज हो जाएगा, सब कुछ चेंज हो जाएगा, स्टडी डिजाइन भी चेंज हो जाएगा, राइट? यू गॉट दिस पॉइंट। सो प्रोस्पेक्टिव रजिस्ट्रेशन इज द ओनली मैंडेट एज ऑफ नाउ, जब तक, एंड इट हैज टू बी डन बिफोर एनरोलिंग द फर्स्ट पार्टिसिपेंट। ठीक है।
One of the challenges we had was it was taking a lot of time to reply, and in between they said nobody was being paid and therefore nobody is there to approve the study. How do you?
We can't start before the CTRI approval comes out.
No, that's what. Now, what is the gap now? What is the turnaround time for the
7 to 10 days जैसे, हाँ, 7 to मैक्सिमम 1 महीना हो जाता है। मतलब हमारी स्टडी के विदिन 1 month आ गई 3
So is the approval required for this or
Yeah, I'll be going through this. नहीं, अभी मैं आगे बताऊंगा। Just wait, let's see the next slide. So just note these points. These are very important points. So पहले आपका फाइनल स्टडी प्रोटोकॉल आपके पास होना चाहिए। Unless until you have study protocol, आपका आगे पास नहीं, मतलब आप आगे कुछ चीजों में स्टक हो जाओगे। Then ethical committee approval, I have put in bold कि ethical committee approval आपका साथ में होना चाहिए। जब तक ethical committee approval नहीं है, आप CTRI में रजिस्टर नहीं कर पाओगे, ठीक है? So if you want to do something, you have to have an ethical committee approval. Informed consent document, details of PI, co-PI, funding, study design, sample size, outcomes, intervention. I'll be going through all of these, but just summarize करने के लिए ये चीजें आपको prerequisites हैं. If you are going to enter into CTRI, ये चीजें चाहिए, ठीक है? So now CTRI कैसे फॉर्म भरते हैं, मैं आपको ऑनलाइन दिखाऊंगा अभी, राइट? So सबसे पहले जो फर्स्ट सेक्शन आता है वो आता है पब्लिक एंड साइंटिफिक टाइटल। जब हम भरना शुरू करते हैं तो जब फर्स्ट आता है उसका कॉलम दैट इज पब्लिक एंड साइंटिफिक टाइटल। तो जैसा कि नाम से पता चलता है पब्लिक, सो पब्लिक टाइटल का मतलब है कि जो आम दुनिया को हमने बताना है, उसमें ज्यादा साइंटिफिक जार्गन नहीं यूज करना, पब्लिक वाली चीजें हम ज्यादा यूज कर सकते हैं, राइट? सो इसके अंदर इट इज बेस्ट इफ वी राइट, जैसा हम सिंपल टाइटल के अंदर लिखते हैं ना, रेंडमाइज कंट्रोल ट्रायल, स्टडी डिजाइन में मेंशन कर सकते हैं उसके अंदर। देन अदर थिंग्स वी नीड इज टारगेट, सैंपल साइज, एलिजिबिलिटी, ये सारी चीजें अभी आपकी आगे आएंगी। सो ये सेक्शंस हैं। देन यूजली आफ्टर सबमिशन CTRI टीम उसको 7 to 40 डेज में रिव्यू कर लेती है, मैक्सिमम 1 महीने तक आपका फर्स्ट रिस्पॉन्स आ जाता है, अनलेस अंटिल देयर इज सम इशू, राइट? एंड दे रेज सम क्वेरीज की उनके हम आंसर दे देंगे तो जल्दी हो जाएगा, एंड देन वी गेट अ CTRI रजिस्ट्रेशन नंबर। तो ये स्टैंडर्ड है इसके। सो पोस्ट रजिस्ट्रेशन हमें क्या करना है? सो वंस आवर स्टडी इज कंप्लीटेड, सो वी हैव टू अपडेट द रिक्रूटमेंट स्टेटस कि कब हमारी स्टडी खत्म हुई, हमें CTRI के ऊपर जाकर रिक्रूटमेंट स्टेटस डालना पड़ता है कि खत्म हो गई, टर्मिनेट हो गई। एंड देन इफ वी आर अमेंडिंग समथिंग बाय एथिकल कमेटी, देन आल्सो वी हैव टू डू दिस, अमेंडमेंट्स आल्सो हमें डालने पड़ते हैं। एंड इफ द PI co-PI चेंजेस, तो वो भी डिटेल्स डालना पड़ेगा। आफ्टर द स्टडी कंप्लीशन वी हैव टू अपलोड द समरी ऑफ द रिजल्ट्स आल्सो, एंड कब आपकी स्टडी बंद होगी वो भी डालना पड़ेगा। देन दिस स्टडी फुली फिनिशेस। Supervisor can act as a PI in for CTRI purpose. For CTRI purpose.
Sir, can you just tell me something? Once the PG student submits the plan for the thesis, it's only after over Padi University getting the CTRI, then the enrollment of the
Yes, obviously, yes.
Not before.
Not before.
Which means 2 to 3 months.
Usually भी सर 1 साल लग जाता है स्टार्ट करने में। एथिकल कमेटी का जब अप्रूवल आ गया ना सर, एथिकल कमेटी का अप्रूवल होते हैं हम CTRI में डाल सकते हैं। नहीं, I'll show you. मैं एक करके दिखाऊंगा। हाँ नहीं it will take 10. अगर आपके पास डॉक्यूमेंट्स हैं तो it will take 10 minutes. If the time permits, I will show you live है कि अभी क्या रजिस्टर करूँगा hypothetical. हाँ मतलब अगर टाइम हुआ मैम तो hypothetical हम कर लेंगे, right? So this is mandatory को जैसा मैंने बताया कि this is mandatory. Now it starts कि हमें क्या क्या उसके fields होते हैं. So the first field is public and scientific titles. So public title के अंदर लोगों को बताने के लिए we should always avoid the jargon. So written for community understanding. So लोगों को क्या बताना है उसके लिए डिजीज क्या होगी, इंटरवेंशन क्या होगी, पॉपुलेशन क्या होगी, इतना काफी है, ठीक है. सो एब्रिविएशन अवॉइड करेंगे लोगों को बताने के लिए कि हाँ बंदे को समझ आ जाए. सेकंड पार्ट इसका साइंटिफिक टाइटल है, जो आपका प्रोटोकॉल का टाइटल है एज सच कॉपी पेस्ट करें. अगर कोई एक्रोनिम है तो एक्रोनिम डालना है, टाइप ऑफ स्टडी डालनी है तो बीच में टाइप ऑफ स्टडी डालनी है, एंड इट शुड अवॉइड ब्रांड नेम्स. सो टाइटल के अंदर प्रमोशनल नहीं होना चाहिए. सो द फर्स्ट सेक्शन इज द टाइटल. सो उसके दो पार्ट्स होते हैं, पब्लिक टाइटल एंड साइंटिफिक टाइटल. सो दिस इज हाउ वी एंटर दैट, ठीक है. उसके बाद सेकेंडरी आईडी मांगता है वो. नेक्स्ट सेक्शन इज एडिंग ऑफ सेकेंडरी आईडी. तो हर ट्रायल का एक नंबर होता है, एक ट्रायल नंबर. अलग अलग डेटाबेसेस के अंदर, अलग अलग सिस्टम्स के अंदर हम उसको रजिस्टर करते हैं. सो दिस जस्ट टेल्स कि इंस्टीट्यूट का प्रोटोकॉल नंबर क्या है, रेगुलेटरी फाइल का नंबर क्या है, जो भी हमने सेकेंडरी आईडी हम डाल सकते हैं. हमारे आईईसी का नंबर क्या होगा. सो दिस जस्ट एक उससे रिलेटेबल हो जाता है कि फिक्स हो जाता है कि ये है, डुप्लीकेसी नहीं होती. सो कोई सेकेंडरी आईडी नहीं भी है तो भी चलेगा, हम नहीं लिख सकते हैं, राइट. ओके. सो नेक्स्ट इज अगला सेक्शन इज डिटेल्स ऑफ पीआई. सो हमारे जो पीआई की डिटेल्स उसके बाद भरनी पड़ेंगी. सो इम्पोर्टेंट ये है कि पीआई शुड बी बेस्ड अपॉन द ट्रायल साइट. मेडिकली क्वालिफाइड होना चाहिए, वहां पे स्टडी की इसकी रिस्पॉन्सिबिलिटी है, पूरा डेजिग्नेशन, इंस्टीट्यूशनल एफिलिएशन, वी ऑल नो दिस हैज टू बी डन. सो पीआई के अलावा नेक्स्ट पार्ट इज कंटैक्ट. सो कंटैक्ट के अंदर भी दो कंटैक्ट आते हैं इसके अंदर. तो फर्स्ट कंटैक्ट इज साइंटिफिक क्वेरी कंटैक्ट एंड सेकंड इज पब्लिक कंटैक्ट. सो पब्लिक अगर क्वेश्चन पूछेगी तो कौन कंटैक्ट आपने करना है. अगर साइंटिफिक क्वेरी किसी ने पूछनी है तो किसने कंटैक्ट करना है. इसके अंदर कॉमनेस्ट होता है कि अगर हम पीजी के नाम से रजिस्टर कर रहे हैं, यंग फैकल्टी के नाम से रजिस्टर कर रहे हैं, दे हैव जस्ट एंटर्ड एंड दे आर नॉट क्वाइट वर्सेंट विद द टेक्निक. सो आपका जो सीनियर पर्सन है दैट कैन एक्ट एज ए साइंटिफिक क्वेरी कंटैक्ट. क्योंकि अगर कोई साइंटिफिक क्वेरी पूछना चाहता है तो सीनियर पर्सन कैन एक्ट. सो सेम पर्सन भी हो सकता है, बट जस्ट मीनिंग इसका यही है कि इफ समवन आस्क फ्रॉम द पब्लिक इंक्वायरी तो पब्लिक क्वेरी कंटैक्ट, इफ समवन आस्क साइंटिफिक क्वेश्चन तो साइंटिफिक क्वेरी कंटैक्ट, राइट. सो एथिकल कमेटी के अंदर जो नाम वगैरह वो सेम ही होना चाहिए. एंड देन स्पॉन्सर्स आल्सो वी हैव टू टेल कि आईसीएमआर से स्पॉन्सर्ड है, डीएसटी से स्पॉन्सर्ड है, एमआरयू से स्पॉन्सर्ड है. सो वी हैव टू टेल द डिटेल्स अबाउट द स्पॉन्सरशिप एंड द फंडिंग, राइट. देन कंट्रीज ऑफ रिक्रूटमेंट, जितनी कंट्रीज में स्टडी हो रही है, अगर ये मल्टीसेंट्रिक स्टडी है एंड इट इन्वॉल्व्स मल्टीपल कंट्रीज तो ऑल कंट्रीज के नाम हमें इसमें डालने हैं. एक कंट्री में तो एक, राइट. सो नेक्स्ट इज ट्रायल साइट्स एंड एथिकल डिटेल्स. तो जितनी ट्रायल साइट्स हैं, जितनी भी, इफ इट इज ए 3 ट्रायल साइट, बाईसेंट्रिक स्टडी, ट्राईसेंट्रिक स्टडी, तो हर ट्रायल की साइट का इसके अंदर डिटेल डालना है. सो नेम एंड डेजिग्नेशन ऑफ ऑल लोकल पीआईज, सबके फोन, ईमेल, एथिकल कमेटी का नेम, फुल पोस्टल एड्रेस, हर कमेटी का डालना पड़ेगा. ऐड की ऑप्शन आती है उसके अंदर, एंड दैट वी हैव टू ऐड, राइट. सो ये बहुत इंपॉर्टेंट चीज है. तो जो एथिकल कमेटी इसकी है, इंडिपेंडेंट एथिकल कमेटी इज नॉट ए प्रीरिक्विजिट. इंडिपेंडेंट एथिकल कमेटी नहीं हम इसको डाल सकते. पहले क्या होता था कि कोई प्राइवेट हॉस्पिटल के अंदर स्टडी हो जाती थी, वो डाल देते इंडिपेंडेंट एथिकल कमेटी ऑफ एक्स हॉस्पिटल. सो नाउ इट इज नॉट. सो ओनली द आईईसी विच आर रजिस्टर्ड विद सीडीएसको. सो आईईसीज विच आर रजिस्टर्ड विद सीडीएसको, सिर्फ उन्हीं का सर्टिफिकेट चलेगा. एंड डेट ऑफ अप्रूवल उसके ऊपर होना चाहिए, वर्जन ऑफ प्रोटोकॉल भी होना चाहिए. सो अगर मल्टी सेंट्रिक है तो हर साइट का इसके ऊपर होना चाहिए, एंड इट मस्ट मैच द प्रोटोकॉल वर्जन. एंड इट एथिकल कमेटी अप्रूवल मस्ट प्रीसीड द सीटीआरआई रजिस्ट्रेशन, उससे पहले नहीं कर सकते, राइट. सो इंडिपेंडेंट एथिकल कमेटी नहीं चलेगी. मतलब हमारे लिए चलो नहीं इम्पोर्टेंट, बट कोई प्राइवेट हॉस्पिटल्स अपने लेवल पे करवा देते हैं कि हम इंडिपेंडेंट से करवा देते हैं, तो वो नहीं चलेगी. सीडीएस को अप्रूव्ड ही चाहिए, राइट. या नॉट रजिस्टर्ड विद सीडीएस को. So
suppose a private they have a provision to submit a request committee.
Yes, that's fine. That's fine. So any IEC which is the university, let's say कि there are many good hospitals जिनकी अपनी नहीं होती, and they are let's say किसी के साथ, या तो अगर उसके साथ affiliated है तो चलेगा. सो हेल्थ कंडीशन नेक्स्ट कम, अगला जो सेक्शन है उसमें है कि हेल्थ कंडीशन हम क्या डिस्कस कर रहे हैं. सो वी हैव टू स्टेज द कैटेगरी ऑफ ट्रायल पार्टिसिपेंट एंड देन वॉलंटियर्स ले रहे हैं या पेशेंट्स ले रहे हैं, और ICD-10 के अकॉर्डिंग वो बोलता है आगे आगे कि क्या क्या पेशेंट्स होंगे, दैट वी हैव टू फिल, एंड उसके आगे डिजीज कैटेगरीज हैं, ये सब हमें डालना होगा, राइट. देन स्टडी टाइप, स्टडी डिजाइन, ये सब कुछ आपका अगर प्रोटोकॉल आपके पास होगा ना हाथ में, तो सब कुछ उसमें लिखा होगा. तो जैसा मैंने पहले स्लाइड में बताया था कि इफ यू हैव दोज पर्टिकुलर डॉक्यूमेंट्स विद हैंड, तो इट विल टेक हार्डली 10 मिनट्स. आपको कॉपी पेस्ट करना है उसके अंदर. सो ये सब चीजें आपको पहले ही मतलब रेडी होनी चाहिए. तो स्टडी टाइप, स्टैंडर्ड स्टडी टाइप्स है, स्टडी डिजाइन, उसके अंदर एलोकेशन कैसा होगा, इंटरवेंशन कैसा होगा, कितने आर्म्स है, कैसे मास्किंग करेंगे, क्या पर्पज है, ये सब कुछ हमें पता होता है. तो दीज ऑल थिंग्स हैव टू बी एडेड इन दैट. देन इंटरवेंशन क्या कर रहे हैं उसकी सारी डिटेल, अगर ड्रग है तो डोज कैसे देनी है, ड्यूरेशन कैसे देना है, रूट कैसे देना है, वट एवर इंटरवेंशन यू आर डूइंग उसकी डिटेल्स. एंड इफ इट इज एन कंट्रोल ट्रायल देन व्हाट इज द कंपैरेटर, you have to give details of that too, right? So ये हो गया. And as inclusion exclusion criteria, ये भी हमारे पास होते हैं. तो what are the inclusion criteria, we have to copy paste there, उनके particular points बने हुए हैं, so that we have to add. Then exclusion also, then participants का details, ये भी सारा details, जो भी participant की detail है वो we have to fill in that section. Then randomization details, then how are we going to randomize. Not only randomization, we also check for allocation concealment, जो भी methods है, standard methods है, blinding में उनको इस तरीके से कर रहे हैं. तो this also drop down menu बने हुए हैं उसके अंदर. So there are drop down menu, just click upon that and just select that drop down menu और ये हो जाएगा, right? And then outcomes. So what are the primary outcomes, what are the secondary outcomes, हमें just अपने protocol में से copy paste करके वहाँ पे लिखने हैं, right? Then sample size, ये भी उसके अंदर options होती है. मतलब I am just telling कि these things you have to fill. तो आपके प्रोटोकॉल में ये सब चीजें होंगी तो कॉपी पेस्ट करते रहना है, मुश्किल नहीं है, यही चीजें होती है उसके अंदर, राइट. देन डेट ऑफ फर्स्ट एनरोलमेंट, राइट. सो अगर अभी स्टार्ट नहीं हुआ प्रोजेक्ट तो वी हैव टू राइट एन एंटिसिपेटेड डेट. फॉर एग्जांपल मैं 1 जनवरी 2026 से एक्सपेक्टेड है कि आई विल बी स्टार्टिंग. सो इफ द ट्रायल विच डेट यू आर प्लानिंग टू स्टार्ट, सो दैट डेट वी हैव टू मेंशन, राइट. सो एंटिसिपेटेड जो आप डेट डालना चाहते हो वो यहाँ पे डाल सकते हैं. कितना ड्यूरेशन है ट्रायल का, वो अगर मल्टीपल जगह पे ट्रायल्स हो रहे हैं तो इंडिया वाला इंपॉर्टेंट है. देन वी हैव टू डेफिनेटली मेंशन स्टार्ट डेट इन इंडिया आल्सो. दूसरी कंट्रीज का तो है ही, इंडिया का भी आपको कब स्टार्ट हो रहा है वो बताना पड़ेगा. देन व्हाट इज द रिक्रूटमेंट स्टेटस ऑफ द ट्रायल. तो अभी के लिए एज ऑफ नाउ तो नॉट येट रिक्रूटिंग ही आएगा हमेशा. बाकी है ना, ये तो कुछ पहले वाले चीजें हैं, बट अभी के लिए तो बाकी बैन ही हो गए हैं. सो वी हैव टू स्टार्ट द रिक्रूटमेंट आफ्टर द CTRI. तो स्टडी जब खत्म हो जाएगी देन वी हैव टू ऐड द डेट ऑफ स्टडी कंप्लीशन, फाइनल एनरोलमेंट कितना हुआ, एंड वी हैव टू अपडेट इन CTRI. सो ये है. एंड देन इन द एंड ब्रीफ समरी ऑफ द स्टडी, जो भी हम करना चाहते हैं. वी हैव टू एंटर हाइपोथेसिस क्या था, रेशनल क्या था, वहां पे पूरा बॉक्स बना हुआ है, उस बॉक्स के अंदर जस्ट एंटर करना है. सो दीज आर द पॉइंट्स विच यू हैव टू एंटर देयर. एंड देन द पब्लिकेशंस जो इसके अंदर से आपकी पहले सेम टॉपिक पे हो चुकी हैं वो मेंशन करनी है, नहीं है तो कोई बात नहीं. IPD कितने बने हैं वो सब कुछ करेंगे. एंड एज आई टोल्ड अर्लियर, पोस्ट रजिस्ट्रेशन के बाद जब खत्म हो गया हमारा, देन चेंजेस अगर कोई होते हैं या कुछ भी, मतलब बाद में प्रोटोकॉल अमेंड होता है, रिक्रूटमेंट स्टेटस चेंज होता है, ट्रायल टर्मिनेट हो गया, खत्म हो गया, तो दीज थिंग्स हैव टू बी एडेड. एंड आफ्टर द स्टडी एंड्स वी हैव टू प्रोवाइड द कंप्लीट डेट, कब खत्म हुआ, कितने पार्टिसिपेंट्स आपने एनरोल किए. तो ये सब चीजें खत्म होने के बाद भी आपने डालना है उसके अंदर, ठीक है. अब रिजेक्शन होने के चांसेस भी हैं. सो व्हाट आर द चांसेस ऑफ रिजेक्शन. सो इफ द प्रोटोकॉल इज नॉट अटैच्ड और इफ इट इज अ रॉन्ग वर्जन. वो जो CTRI में ऊपर हमारे बंदे बैठे हैं ना ICMR के अंदर, दे सी, हम कॉमनली करते हैं कि उनको वो देखते नहीं है, ऐसा नहीं है, दे रीड. एंड जो हम लिख रहे हैं और जो साथ में प्रोटोकॉल अटैच कर रहे हैं, इट शुड बी सेम. सो अगर वर्जन अगर हमें कॉपी पेस्ट करने में गड़बड़ है तो ये हमें ध्यान से करना है कि सेम वर्जन जो हमने फाइनल किया है वही, उसी में से इंफॉर्मेशन हम वहां पे लिखें. इफ द एथिकल कमेटी लेटर का जो डेट्स हैं दे आर नॉट मैचिंग, स्टिल इट विल लीड टू रिजेक्शन. अगर ICD-10 कोड मिसिंग है तो भी रिजेक्ट हो सकता है. अगर आउटकम्स टू वेग है फिर भी ऐसा ही रहेगा. अगर कोई ब्रांड नेम हमने डाल दिया तो भी ऐसा हो सकता है. If there's a mismatch in the sample size or randomization is not described, and retrospective registration, this is important. अगर हम attempt करें retrospective का तो वो सीधे reject कर देंगे कि ये तो आपके already start हो चुकी है. So these are the important errors जो अगर हम ऐसे errors करेंगे तो वो reject हो जाएगा आपका. And once it is rejected, फिर बहुत मुश्किल है उसको दोबारा से री रजिस्टर करना, इसलिए पहले 5 मिनट लगाकर हम ध्यान से उसको करेंगे तो वो हो जाएगा. सो दिस आई हैव ऑलरेडी टोल्ड. सो ये हो गया है. देन लास्ट वाज दिस, द CTRI रजिस्ट्रेशन इज वेरी वेरी इंपॉर्टेंट फॉर ethical, legal as well as a research point of view, and it should always be done before the first enrollment. So data entry should be crucial होती है, and हमारे लिए important ये है कि PG thesis is also important. So ideally speaking, so PG thesis has also to be done this. So I think that was all about CTRI registration. कोई question? Otherwise इसके बाद मैं hands on if there is a time 5 मिनट में दिखा दूँगा. So any question? Hands on दिखा दें चलिए. इसमें easy हो जाएगा. So this is my account. तो मैंने अब तक एक RCT किया था, उसके अंदर ये मेरा पर्सनल जिसमें मैं PI था, राइट. सो यहाँ पर सी दिस इज द होम पेज. सो वी जस्ट हैव टू क्लिक हियर टू एड न्यू ट्रायल. तो जैसे मैंने बताया था, देखो सी द पब्लिक टाइटल, राइट. द फर्स्ट स्टेप इज पब्लिक टाइटल. साइंटिफिक टाइटल, ठीक है. नहीं मैं जस्ट एक वैसे कर सकते हैं अगर इफ यू थिंक योर साइंटिफिक टाइटल इज इक्वल टू पब्लिक टाइटल. आपका साइंटिफिक टाइटल ही पब्लिक लिंक, फॉर एग्जांपल इंसिडेंस ऑफ टाइफाइड इन फरीदकोट. हाँ मैं बता रहा हूँ इंसिडेंस ऑफ टाइफाइड इन फरीदकोट. इट इज सेम फॉर पब्लिक एज वेल एज साइंटिफिक, राइट. सो लेट्स से फॉर दिस आई विल राइट, आई विल डू दिस चेंज, राइट. सो एक्रोनिम नहीं है, नहीं, ठीक है. अब अगर आयुर्वेदिक नहीं है तो proceed.
आपका 2 हो गया ना, नंबर ऑफ रजिस्टर्ड ट्रायल्स 2 हो गया काउंट. ओके. तो ये बात करके वो आपका ट्रायल क्लैरिफिकेशन मॉडिफिकेशन रजिस्टर्ड ट्रायल्स को
सो दीज आर द पॉइंट्स जैसा मैंने बताया, राइट. सो पार्ट 2 पे अगर हम जाएं, this is the part 2. So if I want to add secondary ID, I can add here, right. आगे इसके बाद PI के डिटेल्स, जो भी डिटेल्स है.
It's a similar entry if you have to write for ICMR portal.
Right, like this. आगे मैंने these are simple things. Then contact person of scientific query, right. So same as above. If I write yes तो ऐसे ही आ जाएगा. So contact person for public query, if same as above, I mean इसके बाद चला जाएगा. Then we can go to next part, right. So next में जाके, so name of primary sponsor, address. So secondary sponsor होगा तो यहाँ पे हम ऐड कर देंगे, right? So number of selected country, let's say I do Algeria. I'll just proceed. Go to next part. So this is about sites of study. So here we enter the sites. All the sites here we enter, right? And then if I want to add, we can add here. So DCGI approval required, not required, we can go with this, right? So ये सब कुछ हम यहाँ से ऐसे यहाँ पे आपका ethical committee की details है ना. So here we have to add the details of ethical committee. तो ये सारे ethical committee के यहाँ पे details डालना पड़ेगा, right? So then study type. यहाँ पे देखो point है post graduate thesis. Yes. Type of trial. You just have to add from here, right? So suppose it's a drug trial. So these are all drop-down menus. So this is the intervention and the comparator agent. So inclusion criteria भी हम एज डाल देंगे, जेंडर डालेंगे, डिटेल्स डालेंगे. यहाँ पे हम सिंपल कॉपी पेस्ट कर सकते हैं उसके अंदर, राइट? मेथड्स के अंदर भी ऐसे जैसे ये सारे नीचे जाकर. So these are the comparator agents. These are the primary outcomes. We can simply copy paste from there. Directly copy paste हो जाते हैं. So primary outcome, the time points, the secondary outcome. If I want to add, we can add from here, right? कब स्टार्ट होगा, कब खत्म करने का प्लान है. सो इसी तरह टारगेट सैंपल साइज, ये सब कुछ, फाइनल नंबर ऑफ पार्टिसिपेंट्स, फेज ऑफ ट्रायल, ये सारे पॉइंट्स जो मैंने डिस्कस किए थे, दिस वी कैन ऐड हियर. सो कॉपी पेस्ट करना बेस्ट ऑप्शन रहती है. आपका स्टडी प्रोटोकॉल अगर आपके पास होता है. देन रिक्रूटमेंट स्टेटस, ग्लोबल स्टेटस, राइट, नॉट येट रिक्रूटिंग, राइट, इंडिया का स्टेटस, नॉट पब्लिकेशन डिटेल. ये जो समरी का पॉइंट है, सो दिस इज द समरी. So this is a summary of all, जो सारा कुछ है हमारा पूरा summary हम क्या कर रहे हैं ना. So summary हम यहाँ पर करेंगे and then this was the last part. बस इतना ही है. That's it. So this is how we go with it, right? So not difficult. So any question you want to ask? हाँ जी मैम, अगर आपने प्रोटोकॉल ठीक लिखा है ना तो रिजेक्ट नहीं होता है. ये हमें वैसे ही लगता है कि मुश्किल काम है. एंड इफ यू वांट टू बी ऑथेंटिक, जस्ट नहीं होता रिजेक्ट. सारी दोबारा बोलो. क्लिनिकल ट्रायल मेटा उस टॉपिक का, उस टॉपिक पे. हाँ ठीक है, नहीं जरूरत नहीं है. हाँ मतलब वो So any other question? Otherwise I think we are already running behind time. All right. Any other question? चलिए, thank you.
Felicitate sir with the
Well done.
Very good presentation.
Thank you so much, sir. Our next speaker is Dr. Damanpreet Singh. He is Clinical Research Coordinator, CMC Ludhiana. He has expertise in oncology and rare disease trials. And his topic of discussion is standard operating protocols.
Good evening, everyone. So I'm Damanpreet, and I'm working as a clinical research coordinator in clinical hematology department in CMC Ludhiana. So today I'll give you an overview on standard operating procedures. So standard operating procedures are the written instructions to standardize the procedures and the quality in the clinical trials or in any research unit. So what does everybody, I know everybody now is familiar with the ICH GCP. So what does ICH GCP says about SOPs? So it says it's a detailed written instruction to achieve uniformity of performance of specific function. In simple terms, SOP is a written process, a way for the clinical side to perform a task the same way each time it is completed. So as we know that quality is not a destination, it's a continuous process. So to standardize the quality, we need to give us standardized instructions. So before starting any clinical trial unit or research unit, SOPs should be made so that everybody knows what to do, when to do. So SOPs are used to identify the responsible person for each task. So it is a very important thing, because in the end, if any task is not performed, then anybody can say it's not my part of job. So to delegate everyone, we need to give a set of instructions. Describe actions, what to be completed. So हर टास्क को हमें बताना है कि क्या और कब करना है, और जो भी टास्क करना है वो एक रिटन इंस्ट्रक्शन और SOPs बना के रखनी है. So on the basis of that instructions, all staff, research staff should be trained. The SOPs are used to monitor the site performance. So are SOPs required by law or regulations? So FDA does not mandate SOPs, but if the quality of any clinical unit is not up to the mark, if any inspector came to the site, he will definitely ask to verify what are your SOPs, how it is written. So whether it's a fault of the SOP or a policy level, or the people who is working in a clinical unit is not doing their job properly. So, but ICH GCP says that systems with procedures that assure the quality of every aspect of the trial should be implemented. And next, we know that we now are familiar with the NDCT rules 2019. So these rules are given by the CDSCO and everybody is following. So in NDCT rules, there are mentioned the responsibilities for sponsors, there are mentioned the responsibilities for institutes, and similarly it is also the responsibilities for investigators are also mentioned. So in responsibilities of investigator, it is written that standard operating procedures are required to be documented by the investigator for the tasks performed by them. So benefits of SOPs. So SOPs ensure that all the research conducted within the clinical site follows federal regulations of FDA, ICH GCP and NDCT rule. And it also ensures that it follows the institutional policies. For example, some institute ethic committee mandates that EC fees should be paid before consideration of the protocol in the meeting. So it's an institutional policy. So these policies, these SOPs ensure to protect the rights and the welfare of the human study participants.
So one second, like to discuss, how many of you have SOPs in the research process? In treatment process.
You have SOPs to how to frame the project.
So if you set out to write SOPs, there will be hundreds and thousands of SOPs. There is no limit. So our usual practice is when in doubt, make an SOP. If there is a process which is unclear by people, or two people are doing different things or same thing, make an SOP and keep it. When it takes. So even when you use any drug, to dispose a drug, there is an SOP. To shred the papers which are given to you, you cannot tear and throw like you do or like this and throw. It has to be shredded as mandated by the clinical trial protocol. So all of this have a SOP written down so that there is no violation. So we have clinical trial clear when they come and do the site inspection, we have to do this big file will be there with multiple SOPs written down and kept there so that there is no confusion. So start 2-3 things you need to go back and do immediately. Get everybody get a GCP certificate. That's one. Second is by the mandated by medical colleges, we are supposed to go through a research methodology process which takes about 6 months. Which all of us thesis guides have to do that. So do that. And then start making SOPs for your department so that every aspect is done. And a job description for the clinical trial coordinators or biostatisticians, whoever you are hiring, so that there is a standardization of who comes in, what week. It's a lot of paperwork, but it helps you to align your thought process and streamline your actions.
So it provides autonomy within the clinical sites, improves the quality of data collected, thereby improving the science of the study. So utilized as reference and guidance as how research will be conducted within a clinical site. Excellent training source for new employees or new fellows. So whenever the staff changed, so we don't have to, so that we don't lose the quality of our clinical trial. So these standardized guidelines should be made. So now writing a SOP. So development a template for SOP. So what it should have. Potential elements for SOPs. Header. In header it should clearly state the title, original version date, revision date, effective date and approved by. Usually in the clinical trial units, PI is the one who approved the SOPs. Purpose, why one has the policy, like if it's an informed consent, so why we are making it, so we have to clearly mention it for informed consent. Responsibilities, who the policy pertains to, in this we write that who are included in that, we will define the responsibilities of the person. So if it's a SOP on the informed consent, then we will write that PI should take all the informed consents. Instructions, procedure, how to accomplish the items of the policy. And references. What are the policy based on? Usually for clinical trial units, we reference ICH GCPs and NDCT rules. And appendix. Appendix is the source document or the case report form if it is required. If we are making a SOP on the delegation or we are giving the roles to the research unit team, then in appendix for delegation SOP, we can give the appendix form, delegation log form. Tips for writing SOPs. Use clear and concise language, use active voice, and avoid name. Use titles inside, like CRC will do this, PI will do this, rather than pointing out any name of PI.
So when you are doing a clinical trial, you cite the sponsors or the CRO, entire organization, and the delegation law. So I have to say, so-and-so is authorized to do action number 1, 3, and 20 only. You can take samples, you can courier the samples, you can put the documents together. So-and-so can get the vital signs, so-and-so can take immediate. It's like the doctor can do this, nurse can do this, whatever. But here every task is spelled out. It is a pharma sponsored delegation law. So it comes with the SOP and the JD and only the delegation law involved in that. And also you have to have regular training processes for them. So you have to show so-and-so got trained on so-and-so item on so-and-so date with signing dates. So a lot of paperwork, it is a pharma driven trial. PI initiated ICMR trials may not have that stringent paperwork.
So implementing and monitoring SOPs. So SOPs should be introduced gradually. Sometimes if there are no SOPs, then if we can introduce all of the sudden all SOPs, then it can make a mess also. So prioritize the SOPs which are important first. Like informed consent, these SOPs are very important. So prioritize their SOPs to implement first. Now, principal investigator should approve all SOPs and designate an effective date. SOPs should be revised on a regular basis. Usually it mandates to revise SOPs annual basis, but if PI does not have so much time, so 2 yearly is also acceptable. Previous version of SOPs should be retained. So we have to make, if the SOP is revised, we have to make a new file for the SOPs which are obsolete by writing out that this SOP is obsolete and not in work, rather than destroying or discarding them. SOP training. So all staff should have SOP training. Training should be documented. SOP should be accessible to the staff. Now as told, like there are sponsored trials also. So in sponsored trials, they will provide us manual of procedure. In manual of procedure, they will give us the certain instructions, and these are study specific. For example, if they are providing us IP, any investigational product, they will say dilute in this volume, use this range. Every instruction they will provide in a very detailed manner. But that are manual of procedures, and they are only study specific. That are not site specific, and SOPs are site specific. I'll show the difference. So, what are the difference in the SOPs and manual of procedures? It's compliance and custody, uniformly implementation of trial scope. It's organization wide. It's a institutional based, and manual of procedures are only study specific. For example, if we are having a SOP on informed consent, then what we will write as an institute that use this room, use this camera, and what all needs to be done. But for if manual of procedure, if for informed consent is of study specific, then they will write what to told about IP, what are the risks, what are the benefits. That will be a study specific one. So regulatory, it is required by GCP. MOPs are supportive documents. Longevity, it's a long term, and MOPs are only trial durations as it's a trial specific. Users, so SOPs users are all institutional staff. MOPs are only study team. So there can be if there are many clinical trials on the site, so few CRCs are part of one study and few are of the one study. So manual of procedure are applicable only to the study team. Now these are the few SOP tables or titles which we are using on our clinical site. These are not all. If you want, I can share the PDF. I can share the PDF of the title. So these titles says like we have a SOPs on pre-study, review of protocol. We have SOP on review of investigator, informed consent, end of trial reports, start, and all like randomization, how to do. So in these type SOPs we have written everything and in detail what to do. So it's an example of SOP preparing and submitting amendment in IRB. So as I said, that title is there, date of original version is there, approved by, and effective date is there. So now title of the studies SOP is preparing and submitting amendment IRB document. We state purpose clearly, we state responsibilities, and there are instructions, set of instructions, like who will control, do this. Study coordinator will be responsible for keeping log of all the documents, complete the IRB documents, copy of all the instructions are there.
So imagine when, if and when you are audited for a study you will do, you have all the processes in place. So there is some important aspect called source data verification. So you have submitted certain data into the system. Somebody will come and say, okay, last year on 8th of May you wrote an hemoglobin of 8 for this patient. Show me where did you get that number from. So you should be able to show in the file somewhere it is written, or the report in the computer, that it is HB was 8. Or they would have said, okay, this patient had a headache after taking this medicine. Show me in the source note where the doctor saw, when the doctor saw, and it is written headache started on so and so date after 10 days of medicine, resolved after 5 days. That source data verification has to be there. That is one of the difficult parts of doing studies, apart from seeing patients, is to do this alcohol on the document, so that at the end of it somebody comes and evaluates you are looking into it. So SOPs will ensure that you have gone through this process and done that rigorously.
So here is another example of SOP. It's a SOP on the staff responsibility, duties and job description. So we have a SOP title, SOP number, version and location. So it also has a clear purpose, background, and then there is a procedure. Then whenever the new staff join, what are their roles, we have defined everyone, current post and continuous, and references there are available, and appendices. So this is another SOP example on investigational product. So it also has that, as I said, the template is there, the SOP title is there, SOP number, version, date, approved by, purpose, background, procedure, receipt of IP, how to receive IP, there are instructions. Then how to dispense IP, there are whole set of instructions which we followed on our side. Now, there can be many SOPs, it can be 100 and all. If there is no change, so it's not mandatory for PI to review all and each and every SOP. So we can have a revision history template in which PI can write like these SOPs, no change needed, and revision date, revision version, and can return that no change is needed and it is reviewed.
So we ever apply for NMA certification? This is a big process which we go through.
So this is one website where you can download a sample templates for all the clinical SOPs. There are few, but you can download few templates and refer to as it is very helpful.
Okay, so thank you, Daman, and it has been a tiring day this morning. Site likhana global health methodologies. If you put in, ChatGPT is also very good. It will give you have to make your prompt engineering very well. Don't just copy paste whatever it comes out, and you can get good data from there as well, good SOPs easily. It's like your research assistant who will come and give you some documents and modify that and take it. Okay, so tiring day since morning from 9 onwards, I think we've been here.
packed, but you have lots of work to be done at home. So you decide how you want to do it, sit here, or you collaborate.
Recharge for tomorrow.
Recharge, yes. We are already sit tired.
Yes.
So please work on your Google Docs online if you can, and at least assign some responsibility so that person will go do up that work and come back. As I said, it's an immersion experience. At the end of it, we should come out with something gold. Gold is always refined in fire, right? Yeah, I want Dr. Nitu, please.
And there is going to be a healthy competition.
Yeah. So we'll mark and we'll give you prices also. Then I will get the best team for JC and tomorrow morning there is recap and quiz. Okay. So tomorrow morning we'll have recap and quiz. We will have some quiz there to understand what is happening. So some of you will have to burn the midnight oil tonight. Okay. So tomorrow afternoon there will be a JC presentation and the judges will be there. They will be marking your presentations. So it is your pride to fight between Hoshiarpur, Faridkot, Amritsar and Patiala. It should be a healthy competition. From the trial perspective, that is the ideal.
This was not a question, this was an answer, this was a suggestion.
You are going to analyze all this for some study purpose?
No, I'm not doing any study. I have not taken a consent for that, right? But Dr. Nitu will be doing 5 years from now as what is the quantum of change happened in Faridkot in number of studies.
Yeah, I like to do it.
Okay. So over to you for the instructions for the evening and the morning. 9 o'clock. So if anybody wants a wake-up call, Dr. Nitu will arrange that. Okay. So.
Okay, we'll close the session here. Tomorrow we'll meet at 9 o'clock. But assignment which sir has given, you are supposed to complete that. Now we'll have a cup of tea and you can do the discussion.
You have to keep your institution's flags high.
Yeah.
If you have to keep your institution's flag high, you have to prepare well and come for tomorrow and day after. There will be independent judges.
Sir, there should be first, second, third and